Great pleasure to kick off this scientific program of our Jubilee Conference for the 35th anniversary of the Institute of Bioorganic Chemistry, Polish Academy of Sciences, and 30th anniversary of the PCSS affiliated with our institute. And today in the morning, we are going to kick off with the session on life at the molecular level. And so we will go through the scales both in size but also in time throughout, and we start from the smallest one. And today we are having a great selection of speakers in the first session who will talk about different molecular machineries and their dynamics as well. And so I'm very, very glad and excited to introduce to you the first speaker for today, Professor Jonas Ries, who is currently working at the EMBL in Heidelberg, but he is moving soon to Max Perutz labs in Vienna. And Jonas is also an ERC investigator, and he actually is one of the very few power users of the min-flux microscope. And he will talk to us a little bit about the application of the super resolution microscopy for structural biology, but combining both biology, computational work, but also a bit of engineering, optical imaging, and so on. So, Jonas, the floor is yours, and we are very excited to have you here, and please.
Thank you so much for this wonderful introduction, and also for inviting me here to this amazing meeting. And it's an honor to kick it off with the first talk. I'm at the moment still at the EMBL, and in January, actually, my group will move to the Max Perutz labs at the University of Vienna. So my group is really interested in understanding how complex molecular machines are organized in cells, and for this we are developing new tools for super resolution microscopy. Maybe I wait just 30 seconds for everybody to come in, and then okay. So, you all know about the recent resolution revolution in structural biology that allows us to now solve structures of proteins even inside cells, and that's with an amazing resolution of a few angstrom. This is incredible progress that we have here, but there are still some limitations connected to electron tomography. Okay, maybe I will leave it a second.
Let's give a moment to the people to come in. I think the coffee was too tasty today, so we still have some people fluxing in. So sorry for that interruption, Jonas. But
no problem.
At least we can see that there is quite a lot of people interested. So
it's more fun with a larger audience.
Yes, that's right. So, ladies and gentlemen, I would like you to take your seats as quickly as possible, because we have already kicked off at 9 o'clock to be right on time, and Jonas has already started to talk about his exciting work on the super resolution microscopy. So without further ado, I give back again the floor to Jonas, and please, if you feel more comfortable starting from the beginning, feel free to do that. But,
sure. I think now with twice the audience, I can start again. So what I was saying is that my group is interested in understanding how molecular machines are organized in cells, and to investigate this, we are developing super resolution microscopy technologies. You all heard about the resolution revolution in structural biology, especially electron microscopy, that allows us now to solve structures in proteins even inside cells and with angstrom resolution. And I find this absolutely amazing, the progress in this field. However, there are still some limitations in electron tomography. To reach such a high angstrom resolution, we need to average, as you know, over many identical particles. And this is not so easy to do if you look at dynamic and less regular protein structures. But also we need such a high resolution to identify specific proteins in the electron densities. So this is why I think that optical super-resolution microscopy can be the ideal complementary technology to structural biology. Of course, we'll never have angstrom resolution. In the best case, we have the resolution of a nanometer or so, limited by the size of the label. But we have the advantage of molecular specificity. We know precisely what proteins we are looking at because we put a fluorescence label on it. And we can now see these proteins with a very high contrast that allows us to look at individual particles without any averaging. And also, at least in principle, we can do this in the living cell to directly look at dynamics. So the kind of vision of my group is to develop new super-resolution microscopy technologies that allow us to measure the structure and dynamics of molecular machines in fixed and ultimately in living cells. So I would like to start with a brief introduction how we want to do this, how these super-resolution microscopy methods work. And for this, I start, of course, with the resolution limit of light microscopy. You all know that light is a wave, and because of this, we cannot resolve structures smaller than about the wavelength of light. So this resolution limit, so as a result, even if you look at a tiny object like a single fluorophore, it won't appear point-like in your detector or in your eyepiece, but as this washed-out spot. Now, if two objects get so close together that these spots overlap, we cannot tell them apart anymore. We cannot resolve them. So for more than 100 years, this resolution limit seemed to be absolute. It is literally set here in stone it's not the gravestone, it's the memorial of Ernst Abbe, I was told. People really thought that's the best we can do with light. It won't be possible to resolve finer structures. But then, in around 2006, the so-called super-resolution microscopy techniques were developed, which of course could ignore that light as a wave, but they used smart tricks to kind of circumvent the resolution limit. And today I'll talk about the technique developed by Eric Betzig and others, and it was awarded with the Nobel Prize in 2014. The technique, you might know, is called PALM or STORM, or nowadays we call it single-molecule localization microscopy. To understand how this works, we have to understand the difference between resolution, resolving two objects, and localization, finding the precise position of a single isolated object far away from all other objects. This, of course, is much easier. You can imagine that it's much easier to determine the position of this one blurred out spot than distinguishing the two overlapping spots in the top. We do this by fitting a mathematical model of the Poinsett function to our data, and how well we can do this is described by this equation here, r, the size of the Poinsett function, over square root of number of detected photons. So if we plug in a typical values for a single fluorophore, we see that we can localize it with a precision of a few nanometers. This is, of course, great, but it doesn't help us for biology, where we have hundreds or thousands of proteins within one of these diffraction-limited spots. So what can we do? How can we resolve such a structure that just looks blurred in our microscope? Well, as I said, we need to use a trick. And the trick used in single molecule localization microscopy is to not look at all fluorophores at the same time, but only to look at very few fluorophores at a given time. And to be able to do this is absolutely essential. We need to use fluorophores that we can switch between a bright state that gives us many photons and a dark state that lives for a long time. Because then we can switch off all the fluorophores, switch on very few of them, determine the position, switch on another subset of fluorophores. And after many thousands of cycles of activation, localization, and deactivation, the positions of the single fluorophores will create an image of the structure. So this is, I think, quite nicely illustrated in this movie here. It's a movie measured by my colleague Ricardo Enriquez. And this, of course, shows the Eiffel Tower. And as some of you might know, once an hour, the light bulbs on the Eiffel Tower just start blinking. So he took this movie with a cell phone, then went home. And, okay, it's not playing. It's playing on my screen here. Let's try again. It's just slow. Fine. Yeah, now we see the bit. But what's important is, so he now took the software used for super resolution microscopy to determine the positions of the single light bulbs, and then you would see how on the right-hand side these positions slowly fill in the image of the Eiffel Tower. And now that the movie stopped, it's actually quite nice, even better to see. No, well, we can look on the other. It's even better to see that the Eiffel Tower looks more sharp on the right-hand side than the light bulbs on the left-hand side. So we already have some super resolution here. But of course, on spatial scale, it's a bit too large for biology. Now, of course, the same thing also works in cells. Here you see on the left-hand side a camera image of blinking fluorophores, and on the right-hand side how these positions of the fluorophores fill in the structure of interest. Here, nuclear pore complexes. After tens of thousands of camera frames, typically half an hour to an hour of measurement, we get the super resolution image, a reconstruction that has a resolution between 10 times and 50 times better than the resolution limit of our microscopes. So basically, we could overcome the resolution limit of optical microscopy and replace it by this new super resolution limit R over square root of n. So now I would like to tell you about a few projects in the lab where we use super resolution microscopy, and I will start by telling you how we try to push this technique towards structural scales. And what I will start with is quality control, because it's something that is, I think, really important and something that also really has been missing in the field of super resolution microscopy. And that makes it very difficult to decide, let's say, if our microscopes are still as good as they were a year ago, or if our microscope is better or worse than, let's say, the MinFlux you bought here in Poznan. What you see here are two data sets publicly available that show the same system, microtubules, an image with the same technique, but clearly they don't look the same. And the reason for this can be manifold. It can be the microscope, imaging conditions, the labels we use, sample preparation protocols, or even the software we use for analysis. And now to test all these points and to optimize them, what we like to have is a so-called standard sample. That is a sample that is the same everywhere and every time we look at this. Therefore, we developed such standard samples, and our idea was to use nuclear pore complexes, because here nature puts proteins at well-defined 3D positions in the cells and it defines their geometry. We chose as a target protein NAP96 because it forms two rings in the nuclear pore complex. Each ring contains 2 × 8 copies, and these 2 rings are in register, so that in the top view we can nicely see the 8-fold symmetry of the pore. We then generated cell lines using CRISPR, where we labeled every copy of NAP96 with a tag that we can use for super resolution microscopy. And we use these cell lines really every day to optimize all aspects of the data, of the sample preparation, imaging, and also data analysis workflow. And just for example, you can use it to ask what is the resolution of the microscope. If you can distinguish the 2 rings in Z, you know that you have a Z resolution of at least 50 nanometers. So these cell lines are now freely available, and I would say have become the gold standard for quality control in super resolution microscopy. So this is how we in my group can image these nuclear pore complexes now in 3D and 4 colors simultaneously. Below you see a single nuclear pore complex, and above an average calculated from about 1,200 particles. Okay, thank you. So we can now take these beautiful images using super resolution microscopy. In my lab, we put a lot of effort to do this automatically, so we can put the sample, and 24 hours later we can come back and have many of these images acquired. And then the big question is, what do we do now? How can we analyze this data in a meaningful way, in a quantitative way, to investigate our biological hypothesis? Because just looking at it only takes you so far. There were really not many approaches out there that could help us, so we had to come up with our own. And our idea was to fit a geometric model directly to the super resolution data, and these are the coordinates of the single fluorophores. So the idea is to first calculate the likelihood that such a parameterized model describes the data, and then we optimize this likelihood, maximize it, and as a result, we get the most likely parameters of the model that explain the data. We can also use it to test different models. For example, to ask if this specific nuclear pore is better described by an 8-fold or a 6-fold symmetric model. We just have to do the optimization and then compare the likelihood. So this approach turned out to be quite useful because it allows us to extract meaningful and quantitative parameters from individual particles without any averaging. For example, here for the nuclear pore, we could extract the ring diameter, the ring distance, but also the twist angle between the 2 rings. When we then compared the experiment, this is here, with realistic simulations, we found that the spread of the experimental parameters was quite a lot larger than from the simulations. And this tells us that there is indeed some biological heterogeneity, that not all nuclear pores look the same. Actually, it's something that people know in the field. That's nothing super surprising, but this gives us now a tool to really investigate and quantify this heterogeneity. We can also use this framework for single particle averaging, now in super resolution microscopy. Yeah, you know, it's very common in electron microscopy, but here we applied it to average, again, nuclear pore complexes. On the left-hand side, you see a 3D average that we calculated, again, from about 1,400 sites. And if you now look at the upper and lower ring individually, you see some quite interesting features. You see that these corners are not point-like, but they're elongated and point a bit outward in a spike-like fashion. And this directly reflects that each corner of the nuclear pore complex is occupied by two proteins with a slightly different radius. I should say that we can calculate these averages without any assumptions on the symmetry or even geometry. So finally, we can use this approach to integrate many measurements into one common coordinate system. What we did here is to image our reference protein, NAP96, always together with one additional other protein that we labeled usually using antibodies. And by then registering all the reference proteins on top of each other, we first of all register all the nuclear pores on top of each other from a given data set, but also we register all the other proteins from all the different data sets on top of each other. And we can get these kind of multicolor protein distribution maps. So I think in the future, something like this could be useful to determine the positions of proteins in these complex structures that might be too small or too flexible to be visible with electron tomography. So now I would like to change gears a bit and talk about an application of single molecule localization microscopy to cluster-mediated endocytosis. So what is endocytosis? You all know endocytosis is the process by which cells take up molecules from the surface and from the environment. It's kind of how cells eat, and therefore is a very important process for any eukaryotic cell. So during endocytosis, more than 50 different proteins in many copies assemble at the plasma membrane. Then within seconds, they pull in the membrane and pinch off a vesicle. So endocytosis has been quite difficult to study because of this huge complexity, the fast dynamics, and the small size, way below the resolution limit of optical microscopy. And as a result, we don't really know too well where all these different proteins are throughout the endocytic timeline. So we wanted to change this, and we performed super resolution microscopy. I should say that we used here budding yeast as a model system, because that makes it very easy for us to label the target proteins endogenously with a super resolution dye, in this case, a photoconvertible fluorescent protein. By focusing on the bottom pole of the yeast cells, we kind of get top views, top projections on the endocytic structures. And on the right-hand side, you see a single endocytic site with one of the proteins, LAS17, that forms beautiful rings at these endocytic sites. In this slide, you see the result actually of a lot of work. These are the average distributions of 23 different proteins, half of the proteins involved in endocytosis. These were constructed from more than 100,000 images of endocytic structures, and these are huge numbers. Remember, single molecule localization microscopy is a very slow technique. It takes minutes to hours to acquire a single super resolution image. So this work was only possible because we completely automated the entire data acquisition and data analysis workflow, so we can put our samples and our microscope run without any user intervention and produce a lot of data, like in 24-hour runs. So the results are actually quite interesting. You see that the different proteins really occupy very different zones during endocytosis, but also that this organization kind of reflects their function. So cold proteins form small structures, small blobs. Actin regulating proteins form larger rings, and interacting proteins something between rings and patches. From measurements like this, we could get a lot of information of how the yeast endocytic machinery is structurally organized, how it's assembling, and also how it's force generated during endocytosis. So now we are very interested in understanding the last part, the very dynamic part of endocytosis. And ideally, we would take a movie in 50 colors with millisecond temporal resolution, single nanometer spatial resolution. But even with MinFlux, that's not really possible yet. So our approach is to instead try to reconstruct the dynamics from thousands of snapshots that we take in fixed cells, under the assumption that all of the images that we see come from the same underlying process. For this, we use features in these images to sort them according to the progression throughout endocytosis, so pseudotime, and then we'll align them in space to calculate these kind of composite images. And on the right-hand side, you see one of these dynamic reconstructions. This is not a real movie, this is just the calculated movie, but it runs in real time, and at the moment it would be quite difficult to get similar spatial and temporal resolutions with live cell super resolution microscopy. Recently, we also were very interested or became very interested in mammalian endocytosis. And the reason is that there is an ongoing controversy how the clathrin coat forms and deforms the membrane. There are basically two opposing models out there. The so-called constant curvature model that postulates that clathrin forms with a constant curvature around the growing vesicle. And the other is the constant area model, where people have observed using actually electron microscopy that many structures are very flat, and then the hypothesis is that clathrin first assembles as a flat lattice and then slowly changes its curvature and forms the vesicle. And since then, every year a few papers are published supporting either one or the other or none of these models. So we thought we could make a difference here and shed some light on this question using super resolution microscopy, and specifically the high 3D resolution that we have combined with a high throughput compared to electron microscopy. So we can now use 3D localization microscopy to precisely map out the shape of thousands of clathrin coats in a cell, and then we analyze it with the framework I showed you and extract two important parameters. The first parameter is how far does the clathrin go around the sphere? And no matter what model you believe in, this parameter increases over time. So we can use it as a proxy for pseudotime. Now the second parameter, and we can use it then to sort all the individual experimental structures according to their progression. So the second parameter is the size of these structures. And this now allows us to distinguish between the two models. For this, I plotted for you here the size, or more precisely the curvature, 1 over the radius, versus the angle theta, the progression along endocytosis, and on the right-hand side, the surface area of the endocytic structure, again, versus time. Now we can compare these experimental values, and each dot is a single endocytic site, to predictions from the models. Very clearly, the constant curvature model doesn't fit. We see a clear increase of curvature with time. But also the constant area model doesn't really work well, because we do see that the area increases during endocytosis. So we teamed up with physical biologists in Heidelberg, Ulrich Schwarz and Felix Frey, and developed a new model that describes clathrin growth and the so-called cooperative curvature model. Our kind of assumption here is that the growth of the clathrin structure, that of course happens at the rim, that this appears with a constant rate. So it's kind of the overall area growth is proportional to the rim length. The other assumption is that clathrin has a preferred curvature, h0, and is driven towards this preferred curvature. If we solve these equations, we get a solution, and that we can then fit to our data, and as you see, it fits quite well, and really gives us some additional information on how the clathrin coat forms during endocytosis. But we are also trying to do something similar in mammalian cells as we do in yeast cells, and this is we try to reconstruct the dynamics from all these snapshots. Here we use clathrin as a spatial and temporal reference, and by imaging it together with a second protein, we can align in the future many proteins again into the same coordinate system. This is just some proof of concept that shows clathrin in red together with the protein dynamin. That's a GTPase that in the end pinches off the vesicle. And here you get the first idea how this protein kind of arrives at the place at the rim where it then does its action. So in general, I think that this approach of reconstructing dynamics from many snapshots can be useful, because it gives kind of time information for all these techniques, all the super resolution techniques that are not life cell compatible. And that's not only our high resolution single molecule localization microscopy, also DNA paint will never work inside living cells, expansion microscopy neither. And for all of this, to have such tools ahead might be useful. But of course, it's also very dangerous, because we really have the assumption that all the images we see are representative of a single underlying process, and we can easily get artifacts here. So I think you would agree with me, it would be so much nicer if we could take these kind of movies directly in the living cell on individual endocytic structures, on individual molecular machines. And this is what we hope to achieve at some point using MinFlux. So, but let me start by briefly introducing to you the principle of MinFlux. So, again, after the super resolution limit was developed, people thought that's it. We will never be able to localize fluorophores better than given by the Poinsett function over square root of number of detected photons. But then Stefan Hell, who already held during that time the Nobel Prize for development of stat microscopy, came up again with a smart idea to even circumvent this new super resolution limit. So here's the idea, and that's the underlying principle of MinFlux, is to look at single fluorophores, not with a camera, but with a donut-shaped beam that he scans around it, and then by knowing the positions of the donut and measuring the intensities, we can precisely determine the position of the single fluorophore, and that much better than if we had looked at the fluorophore with a camera. And of course, MinFlux, as single molecule localization microscopy, also really relies on having these single isolated fluorophores. Again, that's important. So, how well can we localize single fluorophores? It's given by this equation here, and compared to our super resolution limit, we gain twofold. On one hand, L, which describes the size of the scan pattern, can be smaller than the Poinsot function, and then we have the 2's in the denominator. So now if you look at this equation here and ask you, what value of L would you choose in order to get the best possible sigma, the small sigma? What would be the answer? Now, clearly L equals 0, no scanning at all. Of course, it would be nice if it was true. It would be much easier to build these instruments, and they probably would be a bit cheaper. But unfortunately, this is an approximation, and one problem is it's only valid if the fluorophore is within the scan range L. But when we start our measurements, we don't know where the fluorophore is, and therefore we cannot use a small L. A solution to this is to first scan with a large L, determine the approximate position of the fluorophore, then reduce the size L, scan it again with and like this zoom into the fluorophore by always adjusting the position around the estimated fluorophore position. With this, it's even possible to beat the 1 over square root of n dependence, because now, if one does this kind of iterative MinFlux, the resolution scales with L over square root of n to the k, where k indicates the number of iterations one uses. Maybe a brief word, why is the resolution so high with MinFlux? Where does this high sensitivity come from? So imagine for a second that you know where the fluorophore is, and you position the donut precisely on top of the fluorophore. So the fluorophore is now in the dark center, doesn't see any light whatsoever, doesn't emit a single photon. But if you move the donut ever so slightly, let's say by a single nanometer, then suddenly the fluorophore will start seeing a bit of light, we will start detecting a bit of signal, and there's a huge difference we can measure between no photons, a few photons, and a few more photons. And this is kind of what gives us this high sensitivity of this MinFlux approach. We were very lucky at the EMBL Imaging Center that we received the first prototype of a commercial MinFlux instrument by the Aberrio company. And here you see how we can now image a single nuclear pore complex with this instrument in 3D with a quite amazing resolution. But what is also for me still quite fascinating, how well MinFlux works in living cells. And this is data that we acquired together with the inventors of MinFlux, with a group of Stefan Hell, on our nuclear pore complexes, this time with a quite dim but live cell compatible label, the photoconvertible protein M-maple as a marker. And compared to single molecule localization microscopy, also in the same sample, we indeed get a much higher resolution. So what we would like to establish in the next years is to use MinFlux for dynamic live cell imaging, to use it to take movies, time-lapse images of molecular machines like the nuclear pore complex, like endocytic structures, to really directly see how these structures deform. And that might be possible with single nanometer resolution in 3D and a resolution of less than 1 frame per second. But it's still quite a long way to get there. So it's not that it's possible at the moment. Instruments are still too slow, and also we need good fluorophores that we can switch on and off many times with light. And that work in the living cell, of course. But what already works now quite well is to use MinFlux for single fluorophore tracking. In fluorophore tracking with MinFlux, we determine the position of our single fluorophore, and then in every iteration we recenter the scan pattern around the fluorophore. And by doing so, we kind of follow the fluorophore through the cell with our donut. Compared to camera-based tracking, we gain twofold. First, we have the very high localization precision of MinFlux. Second, we get this precision with few detected photons, meaning that we can be quite fast. And third, the fluorophore spends most of its time close to the dark center of the donut, and therefore doesn't see much light, doesn't get easily bleached, so we get very long track lengths. We now wanted to use MinFlux to track the motor protein kinesin, and as you see here in this beautiful animation, kinesin walks on microtubules in this kind of hand-over-hand manner, and thereby drags vesicles, cargo throughout the cell. Every step corresponds to a displacement of the motor domain by 16 nanometers, and this leads to an 8 nanometer displacement of the cargo binding domain. So this stepping motion of kinesin has been studied actually quite extensively using many biophysical techniques, but all of this was done in vitro on purified proteins, because up to now we lacked the resolution, the spatial and the temporal resolution to look at this in the living cell. So we wanted to change this using MinFlux, but before we went to living cells, we optimized our imaging in fixed cells. This has the advantage that we can precisely adjust the concentration of the labeled motor proteins that we add from the outside and dilute it down to these ultra-low concentrations that we need for MinFlux. Also, we can change the ATP concentration in the buffer and thereby slow down the motor proteins. Here you see one track that we measured in these fixed cells under reduced ATP concentrations, and in the movie down there you can actually quite nicely see these 8 nanometer steps of the cargo binding domain, and you see these steps even better if you look at the position versus time plot. Now we can ask the question, what is the resolution we obtain with these measurements? And we can answer this question by looking at the standard deviation of the position measurements in each of the steps. If you look at the raw data, these are the gray curves, it's the gray data, we found a standard deviation here of around 3 nanometers. But because we can measure hundreds or even thousands of single localizations in each step, we can determine the step position with a much higher accuracy or precision better, and in this case with a standard error of the mean of better than 1 angstrom. But of course, this is not the final resolution of our instrument. In the end, we are limited by instabilities of the sample. Let's say the microtubules might move by a nanometer or so, or also by the microscope that is not more stable than a micrometer or a nanometer. And also the label itself is larger than 1 angstrom, but all together these are technical problems that we can hope to reduce in the future. And I think this illustrates that indeed MinFlux can be useful to study, on one hand, conformational changes of motor proteins, but also to map out microtubules in fixed cells with down to protofilament resolution. So next we turn to living cells, and for this we expressed kinesin with a halo tag at now the motor domain, and labeled it with a live cell compatible dye. And after really zooming into a very small part of the cell, we obtained many tracks. This is now one of the tracks rendered as a super resolution image, and you can nicely see the clusters of localizations corresponding to the 60 nanometer steps. And steps, of course, you can also see here in this movie. The localization precision here is about 2 nanometers, and that combined with a temporal resolution of better than 1 millisecond, which is quite good for live cell tracking of fluorophores. It allowed us now to look at the steps in many of these tracks and to extract 2 parameters. One is the step size, how far does the step go? The other is the dwell time, how long does the step take? We now could measure this parameter kind of for the first time in the living cell under realistic conditions. So all the data I've showed you so far was 2D data. But biology is 3-dimensional, and to really understand how protein moves and undergoes conformation changes, we would like to monitor these changes in 3D. As I showed you earlier, and as you might know, MinFlux has been used for 3D, but up to now only for imaging, but here we then extended it also for 3D tracking. The idea is to now not use the standard donut, but the so-called 3D donut, and you can think of it as a sphere of light with a dark center in the middle. And by positioning this 3D donut at 7 different positions around the fluorophore, we can then extract its X, Y, and now also the Z position. Here you see some results of, again, image in the living cell. Compared to 2D MinFlux, we lose a bit in temporal resolution and also spatial resolution, but for this we get this beautiful 3D data, which is still good enough to map out the 60 nanometer steps. So this is actually all quite nice, but if you look only at a single color, the applications we can do with MinFlux tracking are quite limited, because we cannot distinguish conformational changes of the proteins from external motion. So at the moment, we are very hard to establishing dual color MinFlux tracking, co-tracking, and the idea is to put, let's say, a red and a green label on different parts of the protein, and then measure with nanometer precision in 3D how these positions change with respect to each other. And we hope that at some point we will be able to do this down to, let's say, 100 microsecond temporal resolution. And with this, we would really be able to see proteins undergoing conformational changes during the action in the living cell. This is the first dual color, and I think up to now one of the very few dual color tracks that we could measure with MinFlux, and what we labeled here is the cargo binding domain with 2 different dyes. Doesn't tell us too much about the motion yet, but it tells you that we are not so far away from being able to monitor conformational changes with dual color code tracking in MinFlux. So I'm very excited about these developments. And this already brings me to the end. I want to thank my wonderful group at the EMBL and all the alumni that contributed to the projects I showed you. And we have wonderful collaborations and quite generous funding. And thank you for your attention.
Thank you, Jonas, for this extremely illuminating and exciting talk. And I wanted to open the floor to questions. We have already a few people who would like to ask those, so please go ahead.
I just noticed this very long tail of when you showed the dwell time of the kinesin. So do you think that's because they stop moving, or you lose the ability to track them?
Yeah, so this model that we fitted is actually a 4-state model, and this is what expected if you look at what happens there. And that fits quite well even to the long tail. But you observe correctly that there are some outliers that go beyond the fitted curve. And indeed, this could be morphos that, for instance, hit a roadblock and then are stuck there for a while. It might be other ways that they get immobilized. It is something that we actually, I did show the data, observed quite a lot, that a lot of these motor perform some sidestepping. And often the sidestepping is after a long time that they are stuck at a certain position. So we really think these might be roadblocks that are circumvented.
Thank you. Any more questions? We have a question out there, and there. I saw 3 people. We still have a moment to ask questions.
Thank you so much for your very interesting talk. I want to ask like a few short questions. First of all, okay, sorry. So how many localizations you need to get this nuclear complex image in 3D that you showed? So like in numbers, and how you recalculate resolution from the amount of localizations and localization precision. Thank you.
So in principle, in single molecule localization microscopy, it's enough to see a fluorophore only once. So single fluorophore for each of the corners is enough to get all the information you need. And then the question is the resolution. And this is determined by the localization precision, which is determined by the number of photons, but also by instabilities of the setup and other stuff that can go wrong. Of course, if you see the same fluorophore several times, you can average the position and then increase it. But in the end, it doesn't really matter. You look at, in the end, the total number of photons you detect and can calculate the precision from this and compare it to the precision that your instrument allows.
Thanks. We have a question out there, and then here.
Yes, so the MinFlux for sure have amazing results with imaging of some discrete, sparse objects. And just the question, what are the most recent advances in its usage with imaging of some kind of more dense structure, like let's say nucleus or chromatin, that are challenging for some other super resolution techniques? So if you could like...
Yeah, good point. I'm not sure if what we gain with MinFlux. So MinFlux is even more sensitive than single molecule localization microscopy to fluorophores close by, because the fluorophore you're interested in is in the dark center, and then you have this huge, very bright rim of light around it. If anything gets close to this rim of light, of course, this will overshadow the signal you're interested in. But having that said, we have very good fluorophores that have a very high switching contrast, and that even for relatively dense labeled structures, you can go into this very fine single molecule regime. One limitation is, of course, that you have to then still image every fluorophore one by the scanning process, which takes time. So the throughput of MinFlux is actually very poor compared to single molecule localization microscopy, which is intrinsically parallelized over all the pixels. So you would have to measure a long time. And only if you look at very, very tiny regions of interest, let's say a single endocytic site, you can speed things up because it scales with the size and beat the speed of single molecule localization microscopy.
And how good is it? There was like the first examples was about the imaging of the nuclear pore complexes, also presented. So how like challenging for MinFlux is just to go to the level, like let's say to the middle of the nucleus, where you just need to go 10 micrometers from the cover slip.
Yeah, so I think this is not so much an issue because we can use silicon oil objectives or even water objectives and then go deep in. You can give in all MinFluxes, I know some way of correcting for aberrations and still get a good donut deep in the sample. And you use a pinhole that rejects out of focus fluorescence. So in the end, MinFlux is maybe even better suited than single molecule localization microscopy to go into the nucleus or a bit deeper away from the cover slip.
Thank you.
Yeah.
Thanks for a very interesting talk. I have a general question about the dyes that are compatible with the technologies. Because what is the limitation of the size? I guess you cannot use antibodies for staining, or can you? Or do you have another solution if you want to study a particular protein that doesn't have any probe?
Yeah, of course, very good question. I think many people still use antibodies even with MinFlux. But of course, if you have a probe that is primary, secondary, let's say 50 nanometers in size, you don't really gain from having single nanometer precision, because then standard single molecule localization microscopy, DStorm, also gives you a resolution that might be better than the size of the antibodies. So it's then up to you. What we like to use, if we can do genetic engineering, is something like Snaptag or HaloTag, because it allows us to use bright dyes and have a small tag. What we would love to use are these fluorescent proteins, these photoconvertible, photoswitchable proteins like in EOS. The problem is that with the commercial MinFlux instrument, they seem to not work yet. And we haven't really understood this, but it would be a big advantage of MinFlux to localize them with this nanometer precision, which it should be able to do so.
Thank you.
Thank you very much. So we have a time for one more question if there is anything urgent. Otherwise, I don't see it. So maybe I'll ask you one more. So we talked a little bit about this before, but you showed initially, the cryo-EM, and then you showed how kind of like a single molecule super resolution microscopy can kind of like complement, let's say, the cryo-EM information. So how do you see the future of, correlating the two together? And
yeah, I think this will be a quite useful development to bring these two worlds together and to combine this molecular specificity of the fluorescent signal to identify specific proteins in cellular tomograms, where it's very difficult to identify specific proteins. And one can use it to, on one hand, for segmentation, and then one can do averaging, or it's in the future maybe even during model building as additional constraints for some proteins that are not visible in electron tomograms. I'm not sure if I would do it with MinFlux, because under this, if you do anyways imaging under cryo conditions with cryo temperatures, these fluophores tend to live very long, they blink very slowly, so you get enough localization precision even with standard single molecule localization microscopy and have to wait a long time anyway, so you would gain from this parallelization. But this is something that we and other groups are really working on, and that we hope that it will become standard in a few years.
Thank you. Thank you so much. Please join me again in thanking Jonas for the great talk. And now I would like to invite you and invite here to the stage Professor Magda Konarska. So we are very fortunate to have her here with us. She comes from Warsaw, from IMOL, and she is particularly well known for her contributions to an operations of another very exciting molecular machine, which is the spliceosome. So please, Professor Konarska, join us here, and we are very looking forward to the talk.
Thank you very much, and happy birthday to this institute. 35 years is a beautiful age, already with demonstrated achievements and yet open for the future, so congratulations. So today I want to, I forget that I have no pointer. I will talk about the spliceosome. And in particular, so this is one of the largest enzymes in the cell, the most complex. It's a ribonucleoprotein machine, which uses its RNA core as the catalytic center to catalyze not one, but two different reactions. And so what I want to focus on today is how does this enzyme select substrates, and specifically, how does it do that for the second step, for the second of these two reactions? And so let's remind ourselves what are these two reactions, and without a pointer, so look to your left. So, my goodness, this is difficult. It's not easy and there is no pointer, so you will have to use your imagination. So the first reaction is the attack of this branch, so-called branch site adenosine, an adenosine in the intron that attacks the 5' splice site and becomes linked to the 5' end of the intron, forming down below this characteristic lariat structure with the branch, and cleaves off the 5' exon. That's the first step. The second reaction is almost a perfect reversal of the first, but now this 5' exon, rather than attacking the branch site, it attacks further down to the 3' splice site. And so below you can see that it attacks, becomes linked to the 3' exon, forming spliced mRNA and cleaving off the intron. So these two reactions, they are catalyzed, now remember, in a single catalytic center, and therefore it's obvious that the spliceosome now to the right, after it catalyzes the first reaction, it needs to rearrange, reposition substrates before it can catalyze the second reaction. So this is obvious, but the second part of the process is much less understood because the cryo-EM structures don't see that very well, and genetics to analyze this requires much more complex tricks. And so what I want to tell you today is how does it happen in specifically how does the 3' splice site is recognized for this whole process. And so, back. If you think about selection of splice sites for catalysis, it's obvious for many of you that alternative splicing is really when you see how important that is. And there is many alternative splicing factors that facilitate selection of this or that site. But this all happens at the early steps of spliceosome assembly. And it predominantly, I mean almost exclusively, refers to selection of 5' splice site and branch site, so substrates for the first step. But the second step, how is it selected? It's much less clear. And so here we go. This is again the spliceosome with the RNA core in color, and here on the right is the catalytic center. But I will be using a simplified schematic to illustrate these things, and specifically we'll focus now, review how these sites are recognized for catalysis. So we start with the 5' splice site in the red size, at least that's a good pointer. It is recognized by pairing interaction to the conserved Akaga motif in U6. And this was described 30 years ago. The next substrate, it's delayed, is the branch site, which is recognized through pairing, this time to a U2 sequence, a very highly universally conserved sequence in all eukaryotes. And this is a tandemly repeated triplet, GUA. This was described, as a birthday present to your institute 35 years ago. But then what happens with the 3' splice site? Well, that was much more difficult to establish, even though the 3' splice site motif is very strongly conserved. It's always pyrimidine AG, in yeast, in humans, and everything in between, of course. So how is it recognized? And so we, together with Charles Query, actually did figure it out, but it required developing a whole separate system of building an orthogonal spliceosome, so a second spliceosome in the cell, which we could manipulate it and show it. And so we formulated a conceptual model and then tested it and proved it genetically. So here are the results. On the left is the first step reaction, and you see in the nucleotides immediately flanking the branch site in this blue box are positioned in the first of these two triplets, GUA. But we know now that after completion of the first step, this whole newly formed branch structure, branch lariat, translocates along U2 by 3 nucleotides, opening the space of this first triplet to bind the 3' splice site. And now this becomes clear because now both substrates for both reactions are positioned at the same site, the first triplet of this GUA repeat on U2, and both are pointing to the active site in the same way. And in both cases, the branch site A and the terminal G nucleotide of the intron are single-stranded for this reaction, and those are the requirements. So that's great, but Ishani, a graduate student in my lab that came just 1 year ago to the lab, was still asking, so where does this 3' splice site come from? Because of course it's very improbable that it would just land in this open space by itself. So where is it and how is it recognized? How is it positioned? Or specifically, sorry, something happened. And specifically what happens when the spliceosome completes the first step but cannot find the functional 3' splice site. And so this again required development of a new system to ask these questions where these events become rate limiting for the process. And she did that. She developed a new system by following this peculiar observation that we had for quite some time but never had enough time to look into it. And this is that, so in the middle below you see this schematic of our reporter system, which is based on actin intron, into which we put all kinds of mutants to test in these reactions. And on the left we can test efficiency of splicing of these different mutants. Always in red you will see the wild type intron and its splicing. And so this is all very well known and established. But what was peculiar is that when we test 3' splice site mutants, they inhibit cell growth. And remember, this is just introducing reporter into wild type strains of yeast using normal media. Of course, these cells don't need splice product of this reporter. So what could be going on? And we speculated that most likely these 3' splice site mutants allow the spliceosome to proceed through assembly and first step of splicing, but then they cannot complete the process and they jam the spliceosomes at some stage. And by doing so, they deplete the pool of available spliceosomes such that there is a shortage of spliceosomes for endogenous introns. So that was the interpretation. And in fact, this is what Ishani showed, that this is, and I will be showing you today, that this overproduction of mutants defective in the second step results in jamming of these spliceosomes in the first step catalysis, and that basically the cell runs out of free spliceosomes. So how could we prove this point? She considered 2 options. One was to reduce supply of these reporters. So, and she did that by introducing mutation into promoter of this reporter, and that completely alleviates the growth defect. And the second option is to increase production of spliceosomes. And again, she did that by overproducing 2 of snRNAs. So far, we have results for U2 and U6 snRNAs, and overproduction of these snRNAs, again, corrects all these growth defects. So that means that we have a system to analyze this process, and now I will show you what we learned about, so that first of all, that these spliceosomes accumulate, that they are jammed in the first step conformation, and I will show you that it happens because of 3' splice sites forming either incorrect, like in this UAC on the very right on top, or that they bind inefficiently, so very weakly. These are very unstable interactions, like for this UUG, where it just doesn't stably bind to the appropriate binding site. And this binding site I'll show you is also on U2 snRNA. This is this GUG motif. So this is so that it's easier for you to follow how did we do this. So Ishani took, again, this one of those reporters, UAC. She did it for many of them. I'm showing you for UAC, which binds, we say that it binds inappropriately and hyperstably, and in particular, the problem is this forbidden pairing of the terminal intron nucleotide, because when changed to a C, it now pairs to the G in GUG, and that creates a problem. And in this context, she now overproduced a variety of ATPases in the boxes in the top schematic that normally facilitate transitions of the spliceosomal conformational changes along the whole pathway. And she tested several of them, but only one of them, PRP16, when overproduced, overexpressed, completely corrects the growth defects. And so this is very important because PRP16 marks a specific point in this whole process, and it facilitates exit from the first step conformation. So without PRP16, cells would tend to, spliceosomes would tend to occupy the first step conformation, have difficulty entering this transition state. PRP16 overexpression helps this and moves things forward. But of course, it does not fix the problem of these 3' splicate mutants. They are still mutated, they still do not participate in splicing, and so you see that splicing efficiency is still down. So what happens is that these introns, the spliceosome exits the first step conformation and unloads this cargo, discards these introns, and they are debranched and degraded, but the spliceosomal pool is replenished. So Ishani also did the opposite experiment by overproducing mutant form of PRP16. And in this case, of course, these mutant PRP16s will further stabilize the first step conformation. And this results in an even worse growth of these strains. They really are pathetically slowly growing. This is amazing. And so this, done on several different reporters and all that, leads us to two very important conclusions. First is that the 3' splicate is recognized already in the first step, and we really know that it is the first step because of this PRP16 result. But the second is perhaps more stunning for me, and this is that this recognition of the 3' splicate during the first step is strictly necessary to even move on forward. So this is a perfect example of a quality control checkpoint that determines the availability of a properly recognized and positioned substrate in order before it can even move on towards the second step. So now the last thing I want to show you is how do we know that where do these 3' splicates bind during the first step of catalysis? And so this GUG, so if you look at the bottom right corner, it is located immediately upstream of this branch site binding site GUA, and in fact overlaps by one nucleotide sharing this G, which is used in both of them. And we know that because Ishani now tested various combinations of mutants in this GUG binding site on U2 with different 3' splicate mutants in the reporter. And on the left you see the results for UAC, and in the green box the G in the GUG is now mutated to a C, therefore eliminating this inappropriate pairing to C. It's really bizarre, the pointer is very difficult to, I hope you can follow this. So disruption of this terminal pairing, of terminal G pairing, fixes the whole problem. Now these strains grow fine. So again, that means that introns are rejected, discarded, and relieve the block of spliceosomes at this stage. But conversely, when the central pair in this GUG motif is mutated, Now we have a situation of two bad things happening to this binding site. We have the incorrect binding of the terminal intron position, and we don't have the position that would hold it stably. And that makes it even worse. And again, this is one of the dramatically inhibited strains. And what I find particularly convincing is the opposite situation where the green mutant of U2 is used in the context, so the one that for UAC helped so much, tested in the context of UUG reporter, now crashes because now it creates this inappropriate pairing of the terminal intron position, but still leaves the poor binding of the central one. So in conclusion, we showed that the 3' splice site is recognized twice during the catalytic phase of splicing. The first is by binding to this, in the first step, binding to the GUG site located immediately upstream of the GUA branch site binding site. And then the whole unit is translocated as one along U2, positioning the 3' splice site in place for catalysis. And so this again demonstrates that the two splicing events, two splicing reactions really are very close to being a perfect reversal of each other. It demonstrates, it explains many things, but perhaps in a broad sense, it is very important to understand this mechanism, because clearly the same mechanism has to operate in all other eukaryotes. We are talking about absolutely conserved positions, both in snRNAs and the splice site. And so therefore, this opens a possibility of potentially developing strategies to alter splice site selection in some kind of diseases caused by inappropriate alternative splicing choices. So I think that this is what I have time for. So here is my group. Ishani is nearly in the center, and I talked about her results. This is, I should stress, this is an early point in work because she just joined the lab, but she's fantastic. And many things need to be still added to the story to make it really final. And of course, all the other people are also working on interesting other projects, which I hope to tell you about some other time. Thank you very much.
Thank you so much for this very exciting talk, and also very fresh results also shown here.
Yes.
So the floor is now open to questions, and I see already the first volunteer.
Volunteer, yes. If you assume that splicing is co-transcriptional process, so whether the same mechanism can operate during circular RNA formation, or it needs to be somehow changed?
No, I think, no, it is the same concept. We are talking about catalytic events, right?
Okay.
So whether it's, so the splice in circular...
If first place which is recognized is 3', yes? Which is a substrate in a second.
Oh, that's what you mean. I don't see, okay, I didn't think about it this way. I don't see a problem, because this is, you know, okay, when I say that the 3' splice site is recognized during the first step, what I really mean is that it's recognized prior to the exit from the first step conformation. So it could be earlier, but it certainly must be there in order to proceed further. And whether this is, you know, 3' splice site and branch site are basically always...
But I think that the whole RNA molecule is somehow interacting with entire transcriptional complex also, sure. Yeah? So whether the same, okay, we need to think, because we observed previously when we study circular RNA in plants that by removing by studying mutant with cap binding protein, yes, it completely changed circular RNA formation. So it means that somehow positioning of RNA within transcriptional complex can influence also splicing.
So I think that we really should sit down and look at it more closely. But I would say that perhaps you're talking more about the assembly phase and recognition in this global sense. I'm talking about recognition of particular nucleotides for catalysis. So this is really high resolution, what happens at the active site.
Okay.
Right. But the same, I don't see a conflict
You
between
don't
these
see
two.
Thank you very much. Do we have any other people? Yes.
Thank you so much. I have a question which is also related to co-transcriptional and post-transcriptional splicing. Because the mechanism that you are showing, I think it works well with co-transcriptional version. But I think there is this phenomenon, especially in plants, where some introns are co-transcriptionally spliced, but some of them remain. And I think it's related to the timing of removing the 3' end because it takes more time. But then the second round of splicing appears where those introns are removed post-transcriptionally. So do you think there is like a, when the first attempt fails, whether there is any factor that could facilitate the 3' removal of your intron? Or do you see any mechanism that could facilitate this part?
From the point of view, from the point of view of the mechanism of, this mechanism of catalysis per se, it doesn't really make much of a difference. Remember that actually in yeast, supposedly, I'm not sure if I believe that, but supposedly a large fraction of introns are spliced post-transcriptionally. And yet it just doesn't make much of a difference. At the same time, remember that this ability to slow down or block progression into the second step may be very useful for some specific introns. For example, telomerase RNA that's in schizosaccharomyces pombi that ends the whole process after the first step. This is quite likely just using this type of, not necessarily that these sites are mutated in specific way, but that slowing down or stopping or delaying the second step may be just a normal part of the game for many introns. So I don't see a conflict, so of course many different introns will play different.
No, it's not about the conflict. It's about more asking why it happens, why it doesn't happen in the first stage, but it happens in the second.
Why it happens?
Yes, because if some introns are removed co-transcriptionally, but some of them stay and they are removed in the second round of post-transcriptional processing, and they are removed, and this is not. As you say, this is related only to, to the ability of removing them. So why they could not be removed in the first thing? Is there?
Well, because, for example, transition out of the first step conformation is slowed for this particular. There are many, and we already know a lot about what might regulate and facilitate this. But then, then they do the rest post-transcriptionally, and that's. I'm sure that that's how it is. And for many introns, that may be, their flavor.
Okay, thank you.
So we have a last quick question here.
Yeah, so I was just wondering about this amazing reporter that you have in cell line that reacts to the problem in the 3'. So if you think from a stoichiometric point of view, that must mean either there are enormous amounts of this thing in your system, and so you titrate that down. And so my question is, at which point, where does the sensitivity to this really start?
Right. So this is done in yeast, which have fewer spliceosomes because they have fewer introns also. And we are actually, this reporter uses a powerful promoter. This is a GPD promoter which just pumps up massive amounts of these reporters. And reduction of transcription by about 40%, which is what we do in this experiment where we titrate it out, already eliminates this process. So we are really dancing around the edge. I would say that even the wild type intron is already pushing the system, because even though it doesn't block splicing, but it still uses so many spliceosomes that the cell is already becoming, it's hard. But in the mammalian cells, I imagine it would be difficult to titrate it out because the number of spliceosomes is enormous. But the mechanism and the ability to modulate it catalytically is, I'm convinced that it has to be the same.
Thank you so much, and thank you again, Magda, for the great presentation. And please join me in thanking Magda for this one. And we talked about the transcription, so now we are moving to translation, and we have another speaker with us, so Professor Sebastian Glad. He is originally Austrian, but came to us from Krakow, from the Małopolska Center for Biotechnology. And Sebastian has also been contributing quite significantly into the story of the tRNAs. And we will hear a bit more about that today. So Sebastian, please, the floor is yours.
Thank you very much. So first of all, thanks a lot for the invitation. I also should say that I recovered from a cold, so I hope my voice can catch up, so you get the sexy part of structural biology today. So I will try to explain and move from splicing to the next step of translation, but I will start with acknowledging the people that actually do the work. So I'm really happy that I have a fantastic team in Krakow, and also all the funding agencies that support the work here. I will not talk today about, and I will try to use this, but so I will not talk about our ubiquitin research and our photosynthesis research, but I will focus on translation today, and I'll show you a bit of what we try to understand how this process works. I've been fascinated throughout my whole career about how a cell makes the right protein in the right cellular context at the right time. And so I spent a lot of time on transcriptional control. So I did work on transcription and chromatin remodeling for some time. But then we moved to understand how translation works at the ribosome. And so more particularly, we are focusing on tRNAs and how chemical modifications of those tRNAs can shape the otherwise relatively boring translation elongation step of the ribosome. And so let me explain this a bit more in detail, what I'm talking about, chemical modifications. So tRNAs are kind of hotspots for modifications, and these modifications are quite widespread, not only in tRNAs, but also in general in RNAs. So you will find RNA modifications in basically all types of RNAs, including tRNAs, mRNAs, snRNAs, ribosomal RNAs, basically any kind of RNA. And just to highlight this here is that, and this slide should show this, is that I think that's what we understand as an RNA here. So you have 4 bases, and that's what an RNA is built out. That's what the textbook says. But when we consider all the modifications, and that's the universe that we're talking about. So each of these bases can be modified by many different modifications. So every RNA molecule is in fact heavily modified. And I should also say that each tRNA molecule that only has 73 nucleotides carries between 8 and 15 modifications on it. So talking about RNA in just 4 RNA bases is really simplifying the whole story. The lab has been interested in 2 main questions, and I will try to explain this a bit more in detail. Is that initially we want to initially we want to modify a specific RNA site, right? And then we want to study what are the functional consequences of this modification. And I draw this picture here because these 2 questions are really interrelated. Because if you think of regulating a function by a modification, what you want to come up evolutionary is in fact a very highly specific modifier that is modifying that site specifically. And then you can regulate the functional consequence of that by this modification site. Just to highlight something that we have been particularly interested is the hotspot of hotspots. It's the anticodon region of the tRNAs. This is where we decode our genetic information and translate it from RNA into proteins. And here I just highlighted all the eukaryotic modification enzymes that specifically modify these sites. And so you can imagine that all of these modification enzymes also have to talk to each other because they come together to shape this very small RNA region. So how does this work, or how we at least think this works, is that if you have a modified RNA, the ribosome will recognize the correct tRNA and it will reject the wrong tRNA at the right speed. And the ribosome itself, it's not traveling along an mRNA at a constant speed, but it actually promotes the folding of the nested protein chain by changing that speed. And so when you have a modified tRNA, you're going to have an optimal translational decoding, but you also have an optimal translocation speed of the ribosome. When you don't have these anticodon modifications, you will slow down the ribosome, and you will not only make less protein, but you're also going to make it at the wrong speed, and therefore the proteins will not fold properly. And just to illustrate this here is that typically the proteins, so the proteins start to fold exactly when they are produced. So proteins fold from the N terminus to the C terminus. And so now you can imagine by changing the speed of the ribosome, you will affect this co-translational folding speed, and therefore you will have aggregated proteins. So when you don't have tRNA modifications, you make less protein, and the problem is the protein that you actually produce is going to be unfolded. And this has quite strong consequences, because most of the tRNA modification enzymes are associated with severe human diseases, and especially neurodegenerative diseases. So what we see there is problems with translation. We have proteotoxic stress, we have protein aggregates, all that we know from other common neurodegenerative diseases. And so we have been studying in the lab for quite some time the most complicated system in that sense that actually adds a modification to uridine bases in the wobble base position of tRNAs, obviously having a direct consequence for decoding. And it's called elongator. Just to highlight this here is that when you want to acetylate a protein, you just need a single protein. It's called GCN5, it's histone acetyltransferase. You need acetyl-CoA, a peptide, and this protein. To acetylate an RNA is much more complicated. So here you really need actually a large complex, 6 subunits, 850 kilodalton. You need the radical chemistry, and you need 5 regulatory factors to recycle this complex. And this was nice because it gave me a job for some time. It gave us also quite good kind of ideas of how to study this. And so basically this is just to summarize all the things that we have been working on over the last years. Basically trying to show how the naked tRNA, this is a challenge. The naked tRNA comes in, it gets modified, it gets released by a subcomplex, and then that protein basically has to recycle. And these regulatory factors, these KTI proteins, are used for this recycling. One of the intermediates has escaped our observations for a long time, and so we published this year. So this is the fully assembled complex with all 6 subunits. And I will not go too much into detail what this structure means, but we finally understand how these 2 complexes are assembled, and also we understand biochemically how this tRNA is bound and it's released. By now we have not only studied the yeast complex, but we have actually obtained several different intermediate structures of the yeast complex, the mouse complex, but also the human complex. And today I will show you what we have observed for the human complex. And so this is also the best structure that we have. I hope you can appreciate the high quality of the tRNA bound in the active site here in Sion. And we can obviously understand a lot of things. We have also found that there is acetyl-CoA that is actually necessary for the modification reaction is bound in the active site. We have determined several structures of this complex with different acetyl-CoA hydrolysis states to understand how the complex is actually reacting during this reaction, during this hydrolysis state. And we have used active site mutants to confirm our findings. But one thing that really kind of surprised us a lot in this finding was that, and I'll try to do this here. So here's U34 that gets modified. But what we found is that there is another base of the anticodon, not really anticodon, that is positioned in a very prominent position, which is U33 here on the right. And U33 is very interesting position because it's basically a universal base in all tRNAs. It's always a U. And second, what is interesting, it's never modified. And so what we did then biochemically as well as with yeast genetics, we can actually show that this surprising finding of U33, that U33, and I will skip to the next slide here, U33 is as important for the U34 modification as the uridine 34. So now we seem to have an explanation why this universal base is recognized. So elongator recognizes only tRNAs by this UU motif. But second of all, it also explains why U33 cannot be modified by other bases because it takes part in this modification reaction. And we have also some data for some other anticodon modifiers that this U33 is actually a unique motif that is recognized by these modification enzymes. So to summarize and to conclude, we have understood how this complex works, also in humans, and we're happy to move on to other complexes. And so here I want to highlight one thing, and this is from if you want to know something about tRNAs, we just published in JBC a very nice comprehensive review about the structure of tRNA. And I think I want to highlight here is that you can see that all of these different proteins are recognizing the same small tRNAs. And they are recognizing those tRNAs in very different ways. So if you now think about your nucleic acid binding protein, you can imagine how many different ways one can actually recognize these very small building blocks. And so we have contributed to this, and I will show this only very quickly, I don't have time to this. We have now structures, high-resolution structures, this is all unpublished work, of basically all of the clinically relevant human tRNA modification complexes. I should also say AlphaFold is great. The basic structure is always most often correct. But for all of these structures, we have actually 4 or 5 reaction intermediates that allow us to track this, and this is really what has changed with cryo-EM. But the last few minutes, I want just to show you and focus you on the other question that we tried to answer, and this is, what are the functional consequences of this modification? And so here I will not go too much into detail, but we have had a very nice collaboration with Marina Rodnina. This was recently published about M6A, which is one of the most prominent mRNA modifications. We can actually show by solving 10 different cryo-EM structures that the anti-conformation of the M6A is the one that is able to be recognized. But because M6A is in this other conformation, it actually inhibits the translation in the ribosome. And Marina has done fantastic single molecular thread studies to complement our structural work. But so please come to me and talk to me about this work later. What I want to talk to you about is popcorns. So what Ting did, a postdoc in the lab, she found had this amazing scientific observation when she was eating popcorns. And she found this very nicely shaped tRNA popcorn. And that gave an inspiration for not only the project, but also for writing an ERC grant. So what do we want to study? So tRNAs, what we actually want to understand is how the different modifications shape the tRNA molecule during its maturation step. The problem with tRNAs is that they're really small, so it's very hard to put them into a microscope. But they're also, the modifications are very heterogeneous, so it's very hard to crystallize them, and you need a lot of material. So what we did is we still tried very hard for some years to put these tRNAs into the microscope. And that's what we ended up with, is we tried to produce 10 different human tRNAs by in vitro transcription. We checked that they are actually folded, and we also determined the thermostability of these tRNAs by a new method that we developed. And I just want to highlight here already, you can see that for folded RNAs, the GC content is completely irrelevant for the stability. And I will come back to this later. And so what we managed over many years is to actually come up and study tRNAs by cryo-electron microscopy. So what you see here is 2 reconstructions of human tRNAs by single particle cryo-EM. They are 24 kilodalton. If you think about 24 kilodalton, you can imagine a lot of different RNAs that you can image with this technique. And I also want to highlight here that these are the best 10 models superimposed over each other. And so this is, even if we are at 4.85 angstrom, we actually can come up with a very high confident atomic model of RNAs at this resolution. But this allows us, and we wanted to take this a step further, we wanted to check the modification status. And so here we've initially tried to use the trick of Moderna and BioNTech that use the N1-methylpseudouridine to stabilize the mRNA vaccine. That's why all of this actually worked in the end. And we tried to incorporate this modified uridine into our tRNAs, and not surprisingly, this doesn't work. So when you have an N1-methylpseudouridine incorporated, you would destroy the RNA, the fold of the tRNA. So you cannot use the same trick that we used for the mRNA vaccines for folded RNAs in the future. But if, and now I really need to point this down, if we use the pseudouridine that is incorporated naturally in the tRNAs, we actually stabilize the tRNAs. So when we exchange all uridines with methylpseudouridine or pseudouridine, we lose the fold. But if we put the pseudouridines in a very particular space, we stabilize them. And so we purified all the human pseudouridine synthases and active site mutants of it in vitro, and now we can actually use any kind of pattern that we want, and that's what you see here on the right. So we basically detect the different modifications. These modifications are stabilizing the tRNA, and we developed a new method to do this here, and I will give up on this pointer now. But so what we can see is that these modifications are really conferring additional stability to these tRNAs, and that allows us to actually also image them after the modification stage. So what you can see here is the two structures before and after modification. It's a challenge. Okay, from unmodified to modified, and I hope you appreciate that there are differences from the left to the right. So there's not many, but it's obviously still a tRNA, but the T-arm and D-arm interaction is compacting. For other tRNAs that we couldn't get the structures before, it made a huge impact of introducing the modification. So you can see here on the left, we're talking more about blobs, but on the right, we can actually get very nice tRNAs, and we can also see that they have actually a huge impact on the stability and thermostability of these RNAs. We do find different examples. I won't go too much into detail of this, but there's some tRNAs that actually depend on some pseudouridine synthesis more than others, and sometimes the pseudouridine site actually is quite unimportant for a tRNA. So this is a very complex system. Can we see the modifications per cryon? We can't, but these are pseudouridines, so they are isomers of uridine, so we wouldn't even see them probably at 1 angstrom resolution. But what we can see is in our local resolution maps, we can see a rigidification. So the blue one is here, it means that it's more compact. This rigidification really happens really close to the modification sites. And so last but not least, we teamed up with Janos in Warsaw to simulate the whole thing, and maybe I would try to still highlight this here, is that one of the very surprising things for us was that when the tRNA, just to highlight this here, this radius of gyration, so this size of an RNA, never corresponded to a fully unfolded RNA. Yeah, so RNAs don't fully unfold even at 95 degrees. And so what we can actually conclude from this is that the RNAs, when we unfold them, they still keep some secondary structure elements. So these are not like proteins that completely unfold. And we can detect that only very unique single nucleotide interactions are crucial for the fold of a folded RNA. And just to highlight this here, this is just a short video, and I hope you can appreciate that. I'll try to do this again. So here's an interaction that is really crucial for the fold, and there will be a moment in this video when this small interaction is broken, and that really unfolds the RNA. So it's not like a DNA where you have a GC content where the nucleic acid really depends on all of the interactions, but RNAs are probably really depending on very, very, very precise contact points, and then, not surprisingly, and I will focus this focus point for tRNAs is exactly where we find these pseudouridine sites that stabilize the tRNAs. And so with this, the conclusions are manifold. The sequences are different. The T-D-arm interaction is the most important one. The pseudouridines are much more complex than we thought. And they act very locally. And then we can study tRNAs, but also other RNAs. So we have also structures of riboswitches. You can think of guide RNAs or any kind of small RNA that you can study. And the last conclusion is to eat more popcorn for our scientific ideas. And with this, I'm finished. And I'll try the last time to acknowledge the lab. There are many people that that really tried to that contributed to this work. Obviously, fantastic collaboration partners all over the world, and the funding agencies. And thank you for your attention. Thank you.
Thank you, Sebastian, for another really exciting story about the complex molecular machines, and for one of, I think, my favorite dietary advices I've been given in the last few years. So that's great. I'll open the floor to questions. So, yeah, we have someone there.
Thank you, Sebastian. It's Miłosz here. In case you don't see me. Fantastic talk. Thanks a lot for that. So I have a question, because I have to admit that I have a problem buying the fact from your introduction that all RNA modifications, they accelerate the rate of translation. Mm-hmm. Do you really think this is absolutely a universal feature, or we don't know so far of any that actually decrease the rate of the reaction of the translation?
No, you're right. So the main fact is that the studies that we have on the biophysical level, they are the ones that show the slowdown in the absence of the modifications. Mm-hmm. But I think it's because it's easier to detect. Mm-hmm. In terms of ribosomal footprinting as well as in the biophysics, it's easier to detect the thing. There's obviously also the rejection state that it seems to be quite important for this. So tRNAs without the modification are more, even the right one is more often rejected. So it's not that trivial that it just, the accommodation phase of the tRNA. But I'm pretty much convinced that some of the modifications that stabilize the tRNAs, they would actually contribute to an accelerated one by stabilizing the tRNA. So I fully agree that this is just to tune. You have, with the modifications, you have a system to tune the elongation speed of the ribosome in a very precise manner.
Okay. Thank you.
Thank you. Do we have any other questions? Yeah, we have one more.
Thanks a lot for an excellent talk. I have a question concerning the link with diseases, because you were showing that there's various enzymes that are mutated and they cause intellectual disability or whatnot. And my question concerns now the relative dosage of those modification diseases. Can you expand on that?
Yeah, so this is a very good question. So this has kept us busy. It was also a reviewer too that kept us busy on this question as well. So what we can show, and I don't know if you can go back to the presentation, and this is maybe just to, it's possible? No. So what we can show is that when we modify one side and the other one, it has an effect on the stability. But then when we mix these samples, we don't have an increased stability, but only when you incubate and introduce both modifications, we get an additional stability. So it means that we are probably 100% modified, and we did a lot of titration experiments to figure out what is the rate of modification. To quantify these RNA modifications is not that trivial as it might sound, because we use a semi-quantitative primer extension method to measure this, but some of the transcripts are still going through even with the modification. So it's a tricky business. Nanopore sequencing is going in this direction to allow us to quantify these detect and quantify these modifications, and we were obviously very happy to implement this in the lab.
Thank you. Do we have any other questions? If not, then I wanted to thank you again, Sebastian, for the great talk. Please join me in thanking. Thank you. And now we move on to two remaining presentations. So we start with Dr. Maxim Serdyakov. So Maxim Serdyakov, who came here from the International Institute of Molecular Cell Biology in Warsaw. And he will talk to us about the molecular machines that interact with the RNA.
Good morning, dear conference participants. My name is Maxim Serdyakov. I'm a first-year PhD student at Laboratory of Single Molecule Biophysics in International Institute of Molecular Cell Biology in Warsaw at Professor Ewelina Majecka. And I'm very grateful to the organizers for the opportunity to speak today. And I would like to tell you about the dynamics of RNA targeting complexes in bacteria. So, our main hero for today, it's HFQ. HFQ is RNA binding chaperone. And HFQ usually facilitates interaction between small regulatory RNA and their target mRNA. So, since mRNA is regulate the many stress response pathways in bacteria, HFQ is required for adaptation of bacteria to some environmental changes. And also in some bacteria, HFQ is known as an important violence factor because deletion of HFQ impairs to the ability of these bacteria to invite epithelial cells and secrete some another violence factors. So it's extremely interesting to understand how we can regulate HFQ and prevent it from carrying out their functions to possible development of new antibacterial drugs. And it's also interesting that HFQ can bind directly to the mRNA, not to without presence of regulatory sRNA. And chaperoning function of HFQ, it results of binding small mRNA with three parts of hexamer structure of HFQ. It's called proximal surface, distal surface, and rim. And here you can see one of their crystal structures, it's proximal surface. And so usually mRNA bonds with only one side, so only with rim or only with distal surface, and sRNA bonds in two binding sites. So, as HFQ facilitates interaction between sRNA and mRNA, ternary complexes, sRNA, mRNA, HFQ usually formed in this process. And the next steps in regulation, it's known that it's usually actions like RNA degradation by degradosome or inhibiting of assembly of ribosome on RNA, and HFQ plays a significant role in all these processes too. So all the questions about stability of ternary complexes are very important for successful mRNA regulation. And in this particular study, we think that all the stages of formation, dissociation, these ternary complexes are very flexible, and their occurrence depends on many factors. And we want to elucidate these factors, and first of all, it was the binding of order of RNA to HFQ, because it's a very rare situation when mRNA can bind to HFQ first. So they think there's differences between efficiency of formation of ternary complexes when sRNA is pre-bound to HFQ or mRNA is pre-bound to HFQ. Also, we want to understand which factors which depends of dissociation of ternary complexes, and how can we regulate the stability of ternary complexes? So, for example, can we modulate their lifetimes? And of course, we want also interested in further fate of ternary complexes in downstream regulation. And for this purpose, we made single molecule experiments using TOF. TOF is total internal reflection microscopy. And here you can see a sample scheme, but shortly it's based on the phenomenon of total internal reflection. So then light beam is totally reflected. It's no longer possible to pass it to the medium, but it creates the electromagnetic field, and it's helped to visualize a very thin layer of medium, and to reduce background fluorescence. And with this method, it's possible to visualize 100 single molecules in parallel. And using, for example, 2 laser beams, it's possible to visualize molecules bound with 2 different fluorophores in single experiment. And in our experiment, it was H of Q, or H of Q with pre-bound RNA complexes mobilized on the microscopic slide. And next, we injected the opposite fluorescently labeled RNA molecule, so it was Cy3 labeled sRNA and Cy5 labeled mRNA, and thus we could observe the formation and dissociation of ternary complexes because we get time trajectories from individual molecules from the CCD camera. And here you can see what we exactly have in our experiments. It's a movie from CT camera, and this one from experiment with pre-bound small RNA, so Cy3 labeled. So before the start, yeah, you see a lot of molecules. It's complexes H of Q with pre-bound small RNA, but sorry, not this one. So, yeah, and if we started. We injected the Cy3 labeled mRNA, and it's hard, especially without a laser pointer. But from some moment, it's possible to see the binding of mRNA molecules with our immobilized complexes. And from this movie, we obtained traces like this one for each individual molecule. And here you can see a signal from Cy3 labeled sRNA and signal from Cy5 labeled mRNA. So from these traces, we can do some conclusions about formation of ternary complexes, dissociation, and all processes which happens. So we could observe several possible events on H of Q complexes. Of course, first of all, it's overlapping, so overlapping of signals from sRNA and mRNA at the same time, and we interpreted it as a lifetime of ternary complex. But of course, sometimes we observe dissociation of resident molecule before the binding with injected molecule, and sometimes injected molecule didn't bound with particular H of Q complexes. And also, it was interesting event, it's displacement of residents by incoming RNA. And if we observe if we watch on the fractions of different events on H of Q, it's possible to see that the fraction of overlapping events, it's always bigger than sRNAs per bound to H of Q. So with per bound sRNA, the complex formation is facilitated, and it was our first observation, and it was confirmed by next experiments, and I will describe it in more detail below. Because I want to start to comparing between binding single RNA molecule to H of Q and formation of ternary complexes. We want to compare these processes. So we have experiments with only H of Q molecules and just watch binding of mRNA and sRNA with it. And we compare the experiments with formation of ternary complexes, so binding of 2 H of Q molecules. And as you can see, we plotted RNA arrival time, distribution of RNA arrival times, and binding of sRNA and mRNA to H of Q, it's always quite a fast process. But if we compare it with the formation of ternary complexes, it was always slower. So to bind, a second RNA molecule, it needs some additional energy. And that's why the formation of ternary complexes is slower than the binding of RNA with H of Q. But also, you can see here that these light green lines, in comparison with light red lines, it's formation of ternary complexes with pre-bound mRNA comparing with complexes with pre-bound sRNA. And formation of ternary complexes with pre-bound sRNA is always faster. And this fact is consistent with our previous result. So we can interpret it. It's because as I said, the sRNA have 2 binding sites with HFQ always, and mRNA has only 1. So for binding of sRNA to complete the formation of ternary complexes, it needs more structural rearrangements, and that's why we saw it both in the fraction graph and in the RNA arrival times. This is a conclusion. So after that, we want to compare the. We want to understand how we can modulate the stability of ternary complexes. So as I said, I will speak about lifetimes, and it was some distribution of lifetimes even in one experiment. So we watched a lot of long-lived complexes and short-lived complexes in one experiment, but even in one trace here for one molecule, as you can see. And we plotted the distribution of these lifetimes, and also here the fraction of ultra-stable complexes with lifetime more than 10 seconds. And it's possible to see that binding order of RNA didn't play a significant role here, because as you can see, for one pair with different orders, sometimes the fraction of ultra-stable complexes is bigger with pre-bound mRNA, sometimes opposite situation. So we think that on the stability of ternary complexes, it primarily depends on the particular RNA pair, and the binding order did not play a significant role here. And after that, we want to understand, the life end of ternary complexes, so the dissociation of RNA from it. And we, again, observed several possible events, because sometimes complexes were stable in all observation time, and we don't see any dissociation, not a single RNA molecule. Sometimes they dissociate simultaneously, but usually one of the molecule, resident or incoming molecule, dissociate first, and we can watch again on the fraction graph, and the fact is that it's always incoming dissociated first fraction bigger than resident dissociated first, so they think that disassembly of ternary complexes, it's mirroring the assembly process, so the RNA that first to bind with HFQ, it's last to leave it. And last thing, it's about particular structural features that can determine the stability of ternary complexes. To check it, we made a mutant of one of our considered small RNA. It's this area mute, and as you can see, its structure much more linear than wild type in the region of base pairing with mRNA. So it's one of the higher pins presented in the wild type, and complexes formed with participation of these mutants is much more stable. The fraction of ultra-stable complexes is at least twice bigger than complexes with wild type. And previous studies shown that mRNA linearization also increased the stability of ternary complexes. And so this pattern for sRNA is mRNA is repeated here. So as a result, they think that our observations unveiled some interesting features about formation of ternary complexes. And first of it, it's the formation of ternary complexes need more energy than the binding of RNA with a single HFQ molecule. And also we understand that the complexes with pre-bound small RNA is formatted more efficiently. But there's no significant role of binding order to the stability of ternary complexes. And we understand that dissociation of ternary complexes, so the disassembly of ternary complexes mirroring the assembly process. And also we think that our structural features about linearization can help to development of some artificial RNA molecules that can strongly bind HFQ and prevent it from carrying out their functions, and it could be useful for development of some new antibacterial drugs. So I'm very grateful to my laboratory colleagues. It's a small lab now, but we only start our work, and also from people from Johns Hopkins University. I'm very grateful to you once again, and I will be happy to answer to your questions.
Thank you very much, Maxime, for your presentation. And I wanted to open the floor to questions. I have someone there.
Thank you for great talk. I wanted to ask you if you did these experiments at 37 degree or room temperature, like microscopic experiments.
These particular experiments?
Yes.
It's what temperature? Yeah, it's room temperature.
And what do you think about kinetics of this products of this complex formation and degradation of room temperature compared to 37? Because it's a completely different kinetics can be. And how do you translate room temperature results to 37? And why?
I think that it will be interesting to make these additional experiments. So thank you. And if you ask me, what results I expected, yeah, I think that the binding kinetics should be faster for all cases with biggest temperature, but I think that the patterns of all these differences between pre-bound sRNA and mRNA and between binding of single RNA molecule to HFQ information obtaining complexes, it still will be the same. So, comparing between room temperature and 37, but inside the experiments with 37, we will still watch some, same conclusions.
Thank you, but I also want to point that maybe there will be problems that you will have a different ratio between fractions of different states because kinetics of like formation and degradation may be different, and it may affect like fractions and all this composition of fractions.
Maybe we, for example, will have a lowest fraction of not dissociated events, because it will be dissociated often. So yeah, probably.
Thank you very much.
Thank you. We have one more question here.
Thank you for showing this nice piece of work. And you mentioned several times about using your findings to develop antibacterial drugs.
Yeah.
So my question is, how specific is this chaperone this chaperone for bacteria and the overall mechanism? So can you develop really drugs that are specific for bacteria?
Yeah. It's actually only start of work. So we just based on some another articles and other studies, and it's just shown that the deletion of HFQ is critical for some virulence bacteria because the stumps with deletion of HFQ, it's lost the efficiency of virulence, etc. So they think that probably if we study all these possible features to RNA which binds with HFQ strongly, we could create our specific artificial RNA and just so strongly bind HFQ more efficiency than regular sRNA in the cell, in the bacterial cell, virulent cells, yeah. And we could, probably broke up all the mRNA regulation in this bacteria, and I think it will be, deadly crucial for this. Right.
Thank you very much. For the interest of time, I will stop it here. So thank you again, Maxime, for your presentation. And as they always say, last but not least, but this is really indeed the case, we have today also with us Dr. Marta Szabad from the Institute of Bioorganic Chemistry, who will talk to us a little bit about the other very exciting structures of RNAs, which are the G quadruplexes. So, Marta, please.
Good morning, everyone. In the beginning, I would like to thank the organizers for the invitation. And I have a great pleasure to introduce you to my studies concerning RNA G quadruplexes from the influenza A virus. First of all, what is a G quadruplex? G quadruplex is a non-canonical secondary structure which is formed in G-rich sequences and they form G tetrads stabilized by Hooke-Steen hydrogen bonds. What is important, the formation of G quadruplex structures requires the presence of monovalent metal ions, and depending on the sequence, the length of loop, and also strand orientation, we can divide these structures into 3 types. It means parallel, antiparallel, and also hybrid type. Interestingly, here I present a graph on the left which shows a time-dependent increase, increase in the published articles each year on G quadruplex structures, and in the period from 1996 to, in general now, to 2020. And we can observe a dramatic increase in the drug, it's a challenge. A dramatic increase in the discoveries, especially from the drug design and discovery perspective. And on the right, there is a figure illustrating a few key discoveries in the field concerning also G quadruplex structure in viral genomes. And what is important, the first verification of G quadruplex structure in viral genome was made in 2008 for Simon virus 40 genome, until the discovery of G4 structure in also SARS-CoV-2 genome in 2020. So these findings encouraged us to inspect the influenza A virus genome for the occurrence of G-rich motifs which can adopt a G quadruplex structure. So our G quadruplex in influenza A virus genome became our suspect. First of all, we used bioinformatic tools to search the influenza A virus genome, in particular the 8 segments of the influenza A California 04 2009 strain, and we found 12 potential quadruplex forming sequence shortly named PQS motifs, and we named them from 1Q to 12Q, and these sequences differ in the length and also in the G residue content. After that, we performed some bioinformatic analysis, and we determined the conservation level of identified PQS motifs within the H1N1 subtype strains. And to this end, we used web logo application, and we generated logo representation. Here I show only a few examples of logo sequences for some PQS. And in general, based on this analysis, we can conclude that conservation levels for all PQS motifs was different. However, in general, when we took into consideration the G residue within these PQS motifs, we conclude that their conservation level were relatively high. After our bioinformatic analysis, we would like to verify the formation of G quadruplex structures by PQS motifs. And to this end, we used various spectroscopic techniques and also electrophoresis analysis. However, today I present only two of them, the results from only two of them. It means NMR spectroscopy. And here I present the selected results for few PQS motifs. And in these graphs, there are the immunoproton region of NMR spectra. And on the left, the signals from 2 to 12 ppm, which appears in this spectra, indicate the formation of G tetrads stabilized by Hooks-Tintin hydrogen bonds, which suggests the formation of G quadruplex structure. While on the right, there are the examples of immunoproton region spectra for 3 PQS motifs. And we can see that we also see the signals between 10 to 12 ppm indicates G tetrads formation, and also additional we can see the signals in the region from 12 to 14 ppm, which indicate canonical Watson-Crick base pairs. And based on these results, we can confirm that some of PQS motif can fold into stable G quadruplex structure. However, some of them can be in the equilibrium between G4 structure and also, for instance, hairpin structure. In the next stage, we used another spectroscopic methods. It means fluorescence spectroscopy. In fluorescence assay, we used thioflavin T compound, and its fluorescence properties should increase when it interacts with G quadruplex structure. And here it is a general schematic of this assay. And we performed two kinds of fluorescence experiments. And here I present the results obtained from these measurements. And we can observe that the fluorescence intensity, the level of fluorescence intensity were different among the selected PQS motifs, which indicates also that some of them can form stable G quadruplex structure, while the another can fold into not only G quadruplex structure. However, this method also helps us to confirm the formation of G4 structure. And to sum up this part, by using bioinformatic methods, we identified 12 PQS motifs in the influenza A virus genome. And by spectroscopic analysis, we confirmed the formation of RNA G quadruplex structure. However, we observed that 6 PQS motifs can form G4 structure. However, only 3 of them were confirmed by all utilized methods. And the results obtained in this part were published 2 years ago. And now, from the literature, we know that G quadruplex structure can be involved in many biological processes in the cell. And for instance, reverse transcription initiation can be regulated by this unusual structure. And we took into consideration these facts, and we performed reverse transcription stop assay for selected PQS motifs. And we used two G4 specific ligands compounds, chemical compounds. And here I present the results for 1Q variant. and we observed some differences between wild-type variant and mutated variant, which indicate that both ligands which we used can interact with G4 structure with high specificity. What is more, we also observed some differences between these two used compounds, which can result from a different molecularity of the quadruplex structure, different folding topology, and also a mechanism of binding between these ligands and G4 structures. And currently, we are interested in the biological investigation of these structures, and we would like to understand and also maybe describe the potential function this structure during the influenza virus replication. And in the end, I would like to thank you for your attention and thank you my colleagues, especially Mariana Lewaj, who is PhD student and who is a main investigator in these studies, and also Dr. Karolina Zielińska for NMR measurements, and Professor Ryszard Gierzak for RNA synthesis. And these studies is financed by fund from National Science Center. So thank you very much.
Thank you very much, Marta. So I open the floor to questions. Yes.
Thank you for very interesting presentations. For me, this conservation of Gs in the sequences potentially forming G quadruplexes is very interesting or very convincing part of your study. But I have the question regarding the actually the dots in the sequences of potentially forming quadruplexes. Other words, because as I understand, those are, let's say, inter-G sequences, yeah, the dots. So the question is how long or how short those inter-G sequences can be to allow forming the G quadruplex.
In my presentation, I show not true sequence of my PQS motifs, but also some examples of showing that this G quadruplex can differ in the length and G residue content, that's all. But if I answer your question, the formation of G quadruplex requires, of course, the G residue content in sequence, and the length and the space between G residue in this sequence can differ. However, it's not possible too long space between G residue because it is during the formation of G quadruplex, the length of the space between G residue result in the formation of loops in the G quadruplex structures. So if the space will be too long, the G quadruplex
but
don't
looking
form.
for those sequences, did you restrict the length,
Yeah.
maximum length?
Yes. In simulation, yes. In bioinformatic tools which I used, I choose default parameters, and the length of the sequence was 40 nucleotides.
Okay, thank you very much.
Thank you. Do we have any other quick question? Yes, we have a couple. Okay, so.
So can you already predict somehow the functionality of this structures based on the regions in the viral genome where they are placed? And do you expect that these structures can be dynamic during the life cycle of this RNA?
It's difficult to find one function for this structure in virus, in influenza A virus genome, because we found that some of these PQS motifs were located within the segments encoding polymerase units, and it's of course important during the viral replication. However, the another PQS motifs were located besides these segments. But now we are working to answer the question whether these G quadruplex have some potential function, and we assume that they can play some role during the viral replication, but we don't know yet when.
Just curiosity, so you mentioned in the beginning that these G quadruplexes have been identified in the coronavirus. So is it known what they do there? Is it known what the G quadruplexes do in the
it's normal.
In the virus, SARS.
Yes, it was detected in SARS-CoV-2, and the number of maybe not number, but a few papers were published concerning this topic.
Yeah, I understand, but have they understood what the functionality there?
Yeah, they they made some interesting studies, but I don't remember exactly. But in the break, we can talk about, and I give you some paper considering this topic.
Thank you very much. Thank you, Marta, again for the presentation and for responding to questions. And so we are now concluding our first session. I think the bar for the next sessions is very high now, scientifically. So I wanted to ask all of you to join me in thanking all of the speakers of this session now. And I invite you to the break. And please, let's meet here again 11:37 so that we can kick off the next session at 11:40. So please thank me again to all the all speakers, and then thank you for attention.
Yes, welcome everyone after the break. We will be starting our session now, so please come in and join us at the second session of this day of the conference. I think we will start now. I welcome everyone to the second session, Life at the Single Cell Level. And I have a great pleasure to introduce the first speaker of this session, Professor Nikolaus Rajewski. Professor Rajewski created and directs the Berlin Institute for Molecular Systems Biology of Max Delbrück Center. He has pioneered the research of life at the single cell level and has multiple contributions to our understanding of single cells in time and in space. And as a co-coordinator of the Lifetime Initiative, he has also promoted the translation of the technological breakthroughs on the improvement of healthcare. And today he is going to tell us about the latest advancements in single-cell omics in the context of personalized medicine. Professor Rajewski, the stage is yours.
Okay. Okay, let's give it a few seconds for people to come in. While people come in, first my big thank you for having me, for inviting me. It's really a pleasure to be here at the 35th birthday party. And actually, I can tell you that the Berlin Institute for Medical Systems Biology, BIMSB, which is part of the Max Delbrück Center, a big research institute in Berlin, what? Okay. So then I'll give this back. Thank you. We just celebrated our 15th birthday, and there were actually some colleagues, for example, Marek and some colleagues were there. So I think it's a very nice exchange between Berlin and Poznan, but maybe on a broader level between Germany and Poland, and I'm personally, many people are excited about it. So I think many labs contributed to what I call here a revolution, which is a big word, but it stands for the fact that now all the omics methods, most of them that have been pioneered or researched or invented in the past, let's say 20 years, are now brought into single cells. And so what we are able is now to read out what really molecularly inside single cells is happening. And I think many people, colleagues, think this is very important because cells are reading, obviously, the book of life, the genome, and they have to make decisions, and they do this cell type specifically, and they do this also in communication with nearby or not so nearby other cells. So if you really want to understand how, let's say, a tissue is functioning, how the cells are working together to perform function, or how cells become, if diseases start, we think that we should collaborate in Europe to bring these technologies also really in medical applications. And so the obvious thing, if you think about it, is that with these new methods, you have a better sensitivity in detecting pathogenic changes. And so it means if you have the normal medical situation, you go with the symptoms into the hospital or to the doctor, that what the field is trying to do is to push, to use the technologies to see earlier what kind of changes are pushing cells or tissues onto a disease trajectory, and by doing so also to seeing new targets, okay, how to interfere. And I'm going to show you some work we are doing about this. Other labs, there are lots of publications currently going in this direction. So, by the way, I have only 6, if you will, political slides. After that, it's pure science. I'm also happy to say that actually the first birthday of the birth of a new medical systems biology institute has just happened a month ago in Lisbon. We teamed up, or they teamed up with us, and we received, or they received a lot of funding from Brussels for this, and the Portuguese government also gave a lot of money. So a building similar to ours. So the picture that you saw in the beginning, I just showed you because of the beautiful, I don't know where this, of the stars, 3-dimensional, and this is where we want to go. But we have a institute, the BIMSB is a computational, experimental, 50-50 balanced institute. And we are next to the Charité in the center of Berlin, and so obviously collaborating with the clinical partners more and more. And a similar idea was attractive for our partners in Lisbon. Okay, and now this is really dear to my heart for the future. So it is clear that resources in Europe for science are going down. There is no question about it, in my opinion, because the money is used for other things. And so the only way to really go forward is to share and to break the boundaries between institutions and to create new ones where really universities or hospitals or research centers can meet and freely exchange, work together, do projects, share IP, do tech transfer in rapid ways, and not according to the rules of each institution which is participating. And so we have now got funding to build this. There are currently 11 Berlin institutions behind that, but we are open for partners from elsewhere. And so I'm hoping that this will open up Berlin also much more than previously possible. Okay, now to the science. So in the first project, we wanted to see what we can learn by doing a spatial transcriptomics analysis of a patient sample, in this case from a non-small cell lung cancer. So this was a standard routine sample from a patient. And we dissected this and performed a multi-modal, actually not only transcriptomics on different slices to put a 3-dimensional model of the tumor of this patient and see what we can learn from this. So not only RNA, but also some proteins and actually some readout of biomechanical features of the ECM. But we teamed up with Cosmics, with NanoString. They ran 6 of the samples for us, and so they mapped 150 million transcripts for 1,000 cancer important genes to about 350,000 cells. This is done by segmenting the slices by stainings so that you can really see the cells, and then the nanostring works by having multiple rounds of, in essence, in situ hybridizations against target molecules. So the data, I will be very brief on this because this is on bioRxiv, but so we have here the spatial transcriptomics data here just for one slice. I'm not going to show you all the quality controls we did, but actually really the cells very nicely map to what we know about the lung, the healthy lung, and also non-small cell cancer. So we can really focus on the spatial arrangements of, can we learn something how the immune cells talk to the cancer cells and vice versa. That's second harmonic imaging that tells you something about the ECM. I will practically not say anything about this. And then we have some in a collaboration, some spatial proteomics also. So we were interested to have a three-dimensional reconstruction because in the end biology is three-dimensional, and so for a tumor we didn't really know what to expect, but I think in principle it is important to think about how cells communicate in 3 dimensions, and so we are developing methods how to do this in general. Computational methods, but also experimental methods. I'll talk about that a little later. So, but you can see that we could align the spatial transcriptomics slices very nicely with software that we had developed. And now I'll just talk about, I think, an essential part of the project, and this is cell-cell communication. So what we can do is we can use RNA as a proxy for receptor-ligand activity. This has many problems, okay, so we can talk about this endlessly because there are some receptors which are there, but the RNA is not, and so on and so forth. But for many cases, you can actually see that the RNA is active in cells which have the receptor and the ligand. So it's not perfect, but it's a proxy. And what we can do now, and that is really cool, I think, we can restrict this analysis to cells which are actually in a reasonable spatial proximity. And now you can again talk about what should it be, if 50 microns or 100 microns, but we tried out some settings, and you can do this also cell type specifically. These are all things which we can discuss, but it's not really the essential point. The essential point, I think, is that you can score systematically receptor-ligand interactions in space for each cell, okay? And then based on this, you can actually now look at pathways which are active in the immune compartments of the cancer. And you can find the ones which are really active in both, for example, or only in one, or seemingly not active at all. So for example, this is PD-L1. It doesn't score very highly. This is a standard target for immunotherapy, Nobel Prize 2018. But we see that there are other pathways screened from, this is systems biology. You have the ability to computationally screen from a very large number of candidates down to the ones which you predict are relevant. Of course, you have to validate this, but you can do this systematic prediction instead of testing for years, one after the other. So when we did this, so we arrived here at a few candidates, and I'm also showing here what happens when you don't do this in 3D, but in 2 dimensions. If you use only one slice and not the 3-dimensional reconstruction, and you can see that the 3-dimensionality helps you in significance for finding the relevant targets. So what you get from this, and this is not a cartoon, this is actually what we put together from the various data sources. I'll show you a video in a minute where all of this is summarized a little bit, but I just don't have too much time to go into detail, unfortunately, but the paper is on bioRxiv, okay? So you can, if you want, really read about all the nitty-gritty details. But here in T-cell exclusion, if you will, you see how we discovered that myofibroblasts are actually signaling to tumor cells, how macrophages are interacting with the tumor cells, or here dendritic cells, immunosuppressive, regulatory T cells, cytotoxic T cells, how they are silenced. And we also marked in which cases there are actually available drugs that could be used in this case for some kind of treatment. So now, I'm not saying that we are saying this would have saved the patients or something outrageous like this, but what I'm saying is that we are getting into a regime where we can, from a sample, a routine sample, perform an analysis which which allows us to come up with some hypothesis for how to maybe interfere here with the tumor. So I'll show now the video. I think this takes some time to load. Yeah. So this is a collaboration with a number of colleagues also from the Charité. So we identified here all these cell types. We combined this with tissue morphology, some mechanical features. Then we were able to identify in 3 dimensions now the core of the tumor, the surface of the tumor, and the cancer cells which are infiltrating the stroma. We identified an EMT hotspot, and we could show by computational analysis that likely most of the cancer cells which are spreading into the tissue come from this one region. So it's like producing this massive, So you can see how beautifully localized various pathways I showed briefly are in space. And you can identify the immune niches and how the various pathways or key genes are patterning or patterned in space. And so that gives you the picture I showed you in the very end. So I think this general direction is interesting. Now, this is, of course, super not possible to do in standard clinical practice because, I mean, this experiment is a 35,000 euro experiment, which we could only do because a nanostring paid for it. But, of course, you cannot do this in clinical routine. Now, there are 2 things. One, I'll just say, the classical H&E stainings, which tell you about the morphology of tissues, which are routinely done in basically every clinic on this planet. People are working now on machine learning algorithms to map the spatial transcriptomics data like I presented it, or other spatial data to the tissue morphology so that you can predict which subtypes of tumors actually map to which H&E stainings. And then you can hope to actually simplify the problem of subtyping patients or understanding better which drugs to use by basically a routine analysis. Because once you do this with machine learning, you have at your disposal a very large number of H&E stainings which you can use. So I also want to say that for this project, it's a combination of talking to clinicians, the combination of machine learning experts and molecular biology, signaling and cell biology experts, which made it possible to arrive at something that we are at least excited about. Okay, so that's a problem, and I told you about machine learning and H&E stainings, but I'm also really excited to tell you about a method that we have developed over the past years in my lab. And so this is called open spatial transcriptomics. So I don't know if this code is active yet, so don't try it out. But in the paper that we will make public very soon, this is part of a figure, okay, of the first figure. It means that you can point your cell phone at this or whatever, and then you can download the entire experimental and computational protocols, including videos, how to do this, and so on. So we are really trying to excite the labs about it. And so just very briefly how this works, first of all, you do cryosections on fresh frozen tissues. We do staining and imaging for cell segmentation. And then we are actually currently using Illumina flow cells, which we break open to put the tissues on top of the Illumina flow cell, then to capture the RNA in a spatially registered way. We have about 400 capture areas per cell. Then we amplify everything together and we sequence. I should say that the Illumina flow cell is very expensive as a flow cell, but we have developed ways how to cut it into hundreds of pieces which are big enough for standard pathology slides. So the cost becomes almost negligible. The only costs really that remain are for library prep and sequencing. And so we are talking about a cost of, for a reasonable depth of some €100, currently maybe 500 per sample. But it's basically only the sequencing cost. You don't need a machine. You can do this on a bench. So I'll maybe not go into this. So what we have developed further is the not trivial. We have solved the not trivial problem how to marry the H&E stainings, which tell us about segmentation, where the cells are, and the transcriptomic data. So these are now the transcriptomic data that we produce. Okay, if it's very greenish intense, lots of RNA, messenger RNA captured. But how do you actually align these things? So this is not so trivial because the resolution of our method is 0.6 microns, so subcellular, okay? So if you want to align the H&E and the spatial transcriptomics data, you need to work out some computational methods, which we did. And then we also worked out a method how to take these slices, and if they're adjacently sliced, how to then align them in 3 dimensions. And so what we get are these objects which are 3-dimensional H&E stainings for sure, but also 3-dimensional spatial transcriptomic maps of your tissue of interest at a subcellular capture. So and now you want to see some data. So here's a mouse head. Here you see the eye. Here are some of the cell types. You can also subcluster this. And we have now enormous numbers of these validations. These are just 3 to demonstrate this. So this is not really an image. We asked the computer to stain or to make visible the RNA for some marker genes. And then we compared to the Allen Atlas, which is really classic biochemical in situ hybridization, and we get a really very nice agreement for now a very large number of genes we tested. Now, is it really subcellular capture? So when looking at all cells, you can mathematically put them into a model cell, and then you can ask, is MALAT1, which is a nuclear transcript, actually really nuclear or not? You can see it's highly enriched in the nucleus, while mitochondria encoded RNA is typically found in the cytoplasm, so you see that really working out. Now, as a kind of proof of principle, we took a human metastatic lymph node. We also have the healthy lymph node as a control and the primary tumor, which is a head and neck cancer, from the same patient. And we sliced this into, I think, 27 slices and ran the entire procedure, and you get this beautiful object now where you get very large number of transcripts in 1 million cells. So actually this one experiment is kind of much bigger than the usual experiments that are done on dissociated tissues with single cell omics. We get UMIs per cell at this depth of sequencing. You can go deeper if you want, but we also stopped here. The median is about 800, but you get many cells where you have thousands of UMIs, and that allows a very nice analysis. I will be brief on this. Much more will be in the preprint, but this is a primary tumor. This is the healthy human lymph node. This is a metastatic human lymph node, now projected into 2D just to visualize it. And you can now study the gene programs and cell-cell communications, which are specific for the primary tumor, for the metastatic tumor, and for the healthy lymph node. And this is just one example we found, for example, in the metastatic case, a marker which is involved in calcification probably of the tumor, which is highly expressed at the leading edge. So this is the growing metastasis. This is the surrounding lymph node. So you see this AMTN, and we think it's driven by wind. So you can also get at some mechanistic, like in the first project, ideas how the biological processes are driven. And now to show you what you learn, what you get from the three-dimensionality, things which appear as spots here in a two-dimensional cut actually turn out in 3 dimensions to be structures. And this is really very useful to understand some of the immune compartments properly, because otherwise you just don't know why the molecular signals in these cells are as they are. So you need this for comprehensive understanding. So you see here how these structures, you can see how they form in 3D. And you can also see, for example, for the leading edge, okay, of this metastatic lymph node, this is a macrophage marker, and it marks actually the leading edge, as we found. And so this is something that you learn now about how macrophages are maybe specifically reprogrammed or acting at the leading edge. It's just one example. There's so many more you could get by analyzing these data. So that's just basically a teaser. You may have heard that there are people discussing that there's maybe more pathogens like bacteria, virus, fungi in solid tumors than previously thought. Some bacterial RNA is also polyadenylated, so we checked if we can capture this with our assay. And so for the metastatic lymph node, we actually find bacterial transcripts marked in red here in our sample. I have no idea if this is contamination or if this is important or unimportant, but just to say that you can now use this method also to locate things like bacteria or other things where they are really in the tumor. So, just for the single cell aficionados, we can also very nicely quantify adipocytes. That's typically not possible. They're gigantic, 100 microns or so. And if you do a single cell classic experiment, you cannot really get them. But this works in our case. So that's our open method. The resolution is 0.5 microns. We are flexible in how big the samples are. But then I wanted to close with this. Please allow me for this. I think we really, in view of the situation, how things are developing, we really need to collaborate much more than previously. So I told you how we are trying to do that in Berlin, how to form a structure which allows people to collaborate freely. But this, I think, should happening across Europe. And then I think also regarding the topic machine learning, which I think is really changing many things how we do, and also some of the talks we saw previously made clear how strong mechanisms are. For example, the talk by Magda Konarska about the splicing is super mechanistic insight into how things really work. Okay. Now the machine learners come and they take all the data and they suddenly predict things which actually make sense, but you don't know why really, okay? So you don't really know what's behind that. And so on the one hand, this is super exciting. You can predict protein structures, you can predict DNA that expresses a gene in cell X. Okay, I mean, can you imagine that? I mean, to me, that's like a dream come true. But I think the next maybe 35 years or whatever years it's important to think about marrying both approaches. I think one without the other is less powerful than both together. So this will require lots of collaborations with the machine learners and big data and computing, and I think these are exciting possibilities. I'm excited, many people are excited about spatial multi-omics. That's my lab at a retreat with some guests. These are the fantastic people who never gave up on the impossible spatial transcriptomics method. Thank you very much for your attention.
Thank you very much. Thank you very much. Now the floor is open for discussion. Do you have any questions? Are there any questions or comments? Yes, there is one question. Can we have a microphone for... Oh.
Thank you very much for a lovely lecture. I have one question. I spotted that the thickness of the slice was 1 millimeter, if that's correct. And I wanted to ask how good the H&E stains correlates to the slice that you analyzed with the in situ, since the segmentation is the most tricky part, and that's adjacent slide. So how good you can really overlay it with this thickness?
Our slices for the metastatic lymph node are 10 microns in thickness. So it's a single cell resolution, but occasionally you will cut through two cells which are kind of meeting. And so there is a cell typing problem in the Z direction, which is coming from that. But mapping the H&E on the transcriptomic data, this problem we have solved. So that works at a resolution of 1 micron for sure. Okay.
Thank you. Another question?
Question regarding the identification of cell subpopulation. Do you have the single cell data for the convolution of cell transcriptomic, or you are using just cell population scores to identify the cells of interest?
Yeah, for the lung cancer project, we have a huge number of single cell data that we use basically to check the quality of the spatial data. There was no need to use that for deconvolution. The segmentation the nanostring machine does is in this case at least really working very nicely. It has some problems, but it's altogether very nice, and you get sufficient depth to type the cells. But you can add these data and then you get even deeper. But it's a cancer, so you never really know. So I'm a bit hesitant in this case to map cancer from other samples. But for healthy samples, we show and you can do that. For our metastatic lymph node project, we did not use single cell data to do that, but you could take an adjacent slice or an intermittent slice that we saved for validation, do single cell sequencing, map it and see that it matches. Yes, absolutely. You know, for Visium, these methods where you have, I don't know, 50 cells per RNA quantification, which is a problem, you need to deconvolute. Here, in principle, you don't need to do this anymore.
One more question, Monica.
Hello, thank you for a very nice talk, and I'm looking forward to read manuscript. I have also a question about the segmentation, and since also there was somewhere information that it's based on machine learning, and I know also that you run OpenST on different tissues. So does it require that for different kind of tissues, different kind of cell types within different tissues, this has to be somehow, I don't know, repeated, or it works the way that you can use, I mean, because I can imagine that, you know, it looks different for the tumor, it looks different for healthy tissue, the cell volumes and so on and so on. So how does it work really?
So, all right. I really don't say that we have a universal answer to this question because it's super complicated, right? But we provide the code that we develop with instructions how to run it and how to change parameters if you look at when it doesn't work, let's say. We also tested it on situations where you have very large cells together with very small cells. So we did lots of controls to make sure that it should run stably, but this is again a very complicated problem. On top of that, currently we use only H&E for segmentation, which is suboptimal. You should use also stainings for cell membranes and so on specifically, so there are lots of technology out there. We have not yet integrated that, but I think that's possible.
Okay. And also maybe
one short.
Okay. So since you have really subcellular resolution right now, or you are at the subcellular resolution, maybe it's there would be some RNA, subcellularly localizing close to membranes that you can use also as a even proxy for.
Yeah. So we have also tried that, and that probably can also work. It would require just really a project.
Okay.
Okay. 2 short questions.
Okay. Very short question. Yes. Okay.
Yes.
So you show us some spatial transcriptomics, which is based on whole transcriptome analysis, yes? But from single cell analysis, we learn that not all transcripts are necessary to analyze if you would like to learn about cells, differences between cells. So do you think that it is possible to have targeted spatial transcriptomics? And in this case, it can be really simplified and more cheaper and easier to analyze.
Yeah. So if you have a targeted approach, that is basically what the nanostring and also the next generation of Visium does. But then, of course, then you're targeted, great. But you run into other problems. For example, then if you switch from human to mouse, or something else, then things become difficult, or if you have mutations in there, in particular in cancer, it becomes difficult. So, but these methods have their value, they're just super expensive and they have limitations as well. We do sequencing, so we are agnostic to the sample, it could be anything, from fish to anything. But you can, what I wanted to say is if you want to insert primers that fish for specific genes, you can do that. We are doing this currently for bacterial transcripts because we find that very interesting. But there are, I think, no limits in the sense of that you can put in oligos primers onto the flow cell for whatever you want. You just need to barcode them with a random barcode and register them in space, but that's doable.
We have last question now in this discussion.
Since you have shown this cartoon, which is actually not a cartoon, and there was a pill on it, is there any drug candidate already?
Yes. Yes.
On this interface and the non-small cell lung cancer and the PDL, for example, is there any?
Right. Yeah. So the pills marked the ones which are in principle druggable.
Okay.
Just don't know if that works in that patient and for non-small cell lung cancer. Okay. Yeah. But that's it's a prediction that we make.
Okay. So there are some, but cannot be revealed.
Okay, thank you very much once again. Thank you for this very exciting talk. And the second speaker in this session is Professor Bożena Kamińska-Kaczmarek, head of the Laboratory of Molecular Neurobiology, Nensky Institute of Experimental Biology. And she is particularly well known for her discoveries of the effect of tumors on the local immune system. And today she's going to tell us about the, let's say, single cell landscape of tumor microenvironment.
Thank you very much for invitation. It's really a pleasure to share with you our recent data showing that to some extent we really dissect the composition of the tumor microenvironment. We were interested for years how these cells, which in case of brain tumor are composed of both resident myeloid cells called microglia, but also the peripheral immune cells which infiltrate the tumor. We were trying to understand what each type of cells is doing, but if you think about the tumor right now, the tumor is composed not only neoplastic cells, but many other cells which create so-called tumor microenvironment. And this is composed also of many cells which are coming from the periphery immune cells. And you can also imagine that the same cells which is located in the tumor center or tumor periphery, where there is different state of hypoxia, there is different pH, there is different gradient of molecules, can adapt different functional phenotypes. So when you take the piece of the tumor, you actually lose all the information about the specific pattern of cells, and you lose information about the interaction the cells can possibly have in the space. This issue of tumor microenvironment became immune tumor microenvironment became very important, because right now when you look at the therapies, you can see that immunotherapies, the CTLA-4 antibody is the most important one, the immune checkpoint antibody. All these therapies which promise to be very specific actually do not work in many malignant tumors, and the percentage of patients benefiting from the immunotherapy is about 30–40%. And there is growing understanding right now that many tumors have so-called cold immune environment, and there is no really much immune cells which could be supported by checkpoint antibodies, and we need to understand why there is so little lymphocytes, why there is so many myeloid cells programmed to support tumor growth. So this issue became really important to understand. And when the technology became available on the market, because previously we were trying to sort cells, but when we focus on myeloid cells, there is no really markers to distinguish between different populations. So we decided to employ, together with my two PhD students, Natalia Ochocka and Paweł Segit, to start with the isolation of the immune cells from the tumor-bearing hemisphere. We did sort CD11B plus cells, which mean myeloid cells, and then we used single-cell sequencing. And we got really lucky because we could sequence like 4,000 or to 5,000 cells per conditions. Altogether, this was a very high-quality experiment with more than 40,000 cells sequenced, and we could see the really big heterogeneity. First of all, we found that even in normal brain, there is quite huge functional heterogeneity, and we can see cells with different transcriptomic pattern. But you can see that in hemisphere with tumor, we can distinguish many subpopulations, and we discovered microglia, macrophages, monocyte and macrophages, vessel and meninges-associated macrophages. So we could see the very large heterogeneity. But at that moment, even if we found quite interesting markers, so for example, we were looking for markers to distinguish microglia and macrophages, and we found some, including the new one, a gene encoding beta-galactin-3, which was expressed only in those infiltrating immune cells from the periphery. And using this marker for immunofluorescence, we found that actually these cells are exactly where the tumor is. So these cells do not infiltrate in any space around the tumor. They infiltrate where the tumor is, while actually microglia are very much around the invasive age. And from other studies, we knew that the microglia supports the invasion of tumor cells. So we found the marker, but we were not happy with distinction between monocyte and macrophages. So we decided to implement the site-seq, which combines the single-cell RNA-seq with the antibodies. In this case, we use 10 antibodies, which are also barcoded for sequencing. And you can see, for example, that in some cases, like gene encoding protein CD45, which is a marker of myeloid cells, at the protein level, and also this one, and PDL1, at the protein level, you increase the dissection of subpopulation very highly, and thanks to this, for the first time, we could distinguish different subpopulations. So, for example, we could distinguish the population of freshly arriving monocytes, which actually we found have mostly interferon signature, which means that they are coming to the brain as inflammatory cells ready to fight the tumor. But during the exposure to the tumor microenvironment, they became reprogrammed into two subpopulation of tumor-supportive macrophages, which do to some extent antigen presentation, but they are highly immunosuppressive as they produce a lot of PD-L1 protein. They are phagocytic, they are tumor supportive, while, for example, microglia has many cells expressing cytokines, chemokines, and also proliferate. So this was one of the best discrimination of function, and what was also very, sorry for that. What was also very important, in my opinion, that we found that this reprogramming of infiltrating monocyte to pro-tumor macrophages take place in the tumor, because when we isolate cells, those cells from the periphery, they do not express markers like PD-L1 or arginase 1. They do express these markers only when they arrive to the tumor and through the stage of intermediate monocyte macrophages are transformed to mature macrophages. So we conclude from this that using the site-seq, we can really establish the functional phenotype of myeloid cells. We can see the mechanism of the reprogramming. We use pseudotime to show that this is really macrophages giving, monocyte giving rise to macrophages. We found also some sex-related differences in this immunity. But we were kind of attracted by this idea that there is a spatial localization of cells. So for the new experiment, we decided to look for the entire immune component, and we expanded the site-seq part because we use right now 45 antibodies to detect different myeloid and lymphoid subpopulation, and we do single-cell RNA sequencing. And in parallel, we also take the cryosections, and we use Visium slides. So Visium slides is a slide where you have 5,000 libraries spotted in specific places and barcoded. There are coordinates on the slide, so you can, after taking all RNAs and sequencing, you can say more or less where are the cells expressing specific genes on the slide. And just simple analysis shows a very complex situation. So thanks to including so many markers, we can right now distinguish all the myeloid and lymphoid subpopulation, and we also can find not only cell lineage subpopulation, but we can see functional subpopulation. So we can see different stages of NK killer, we can see T-cell progenitors, T-cell exhausted. We really can have a very complex view at the composition of the tumor microenvironment. And single-cell transcriptomics, here is just the histological staining with tumors is very simple because the tumors growing in the mouse brain are very localized. But you can see by simply using the genes which are stemness gene, you can very nicely visualize the tumor, and then we confirm that infiltrating cells indeed localize in the tumor core, while, for example, astrocyte creates a beautiful ring around the tumor, which is consistent with the data from humans. And we play with this data. So we could, for example, see how the monocyte convert into macrophages using different type of scores. We could also look at the communication, at the gene expression patterns. Here we have different subpopulation, and you can see that indeed there is a switch between monocyte and intermediate, where they have very strong expression of toll-like receptors, cytokines, and things related to inflammation, to the very low expression of those genes in the mature macrophages, while, for example, oxidative phosphorylation, fatty acid metabolism is activated in those cells, which is exactly what people see in the human tumors. And when we do analysis, in this case, we did the convolution using single-cell RNA-seq data. We did the convolution of the gene expression in tumors, and you can see that indeed the infiltration of cells expressing markers of monocytes is very localized, and we found, for example, that this subpopulation of immunosuppressive or phagocytic mature macrophages are not the same. I mean, they have different localization within the tumor, which shows that there are special microconditions which could be also important in this. We did very interesting observation about the cytotoxicity and exhaustion using some gene scores specific for the phenotype, so we know which cells represent cytotoxic cells or exhausted cells. We found very interesting story about the development of natural killers, because, for example, this is the pseudotype showing that the NK cells, mature NK cells, and so-called glioma-associated NK cells comes from the premature NK cells because they have different maturity score, anti-tumor score, and you can see that these glioma-associated NK cells do not show either maturity or anti-tumor response score. We did analysis what are the genes and transcription factors associated with specific state, and you can see that premature and glioma-associated have really different transcription factors. And when we look at the spatial transcriptomic data for the proportion of these three different NK subpopulation, we could see that mature NK cells are more abundant at the tumor periphery than in tumor core, which means that there is a direct impact of the tumor on the reprogramming. And as has been shown before, we did also analysis of communication between cells looking for ligand-receptor interaction. In this case, it was too difficult to work with 34 types of cells, so we pull all together lymphoid and myeloid cells, and we focus on the interaction between myeloid cells as senders and lymphoid cells as receivers. And you can see by this red color that probability of the interaction from cell chat was very increased for some specific interaction, which from the literature it is known that they are important. Another type of analysis goes further. This is called cell call. And in addition to looking for the expression of ligand or receptors in receiver cells, it looks also for the expression of genes which are downstream in the signaling. So even if this is the static situation, it shows that the signaling pathway leading from the myeloid cell is really working on lymphoid cells, and there are specific signaling effectors which could be detected in this way. So we had this very important atlas and methodology to detect different cells. But when you go to the human tumor, they are very different. And even in glioblastoma, GBM, there are different oncogenic drivers which actually are responsible for some subtypes, proneural, classical, or mesenchymal. And there is PDGF amplification, EGF amplification or mutation, and the mesenchymal has also some set of genetic alteration. So we took the advantage of collaboration with Maria Castro from University of Illinois, and we used 4 different cell lines differing either in NRAS or PDGF as oncogenic drivers, and they have also mutation in IDH1, which is very important for epigenetic regulation. And then we did site-seq with 45 antibodies. Here you can see that this cell line really expressed the mutated human IDH1, and there is a difference in survival. All those which have mutation have lower survival. And here there is information about the subpopulation. So first of all, if you look at this too, there is a difference in the composition of the immune compartment depending on the tumor. But when you also compare this way, you can see that there are differences, and some subpopulations are more abundant in mutants, irrespectively of the oncogenic driver. And here there is a kind of visualization of the contribution of specific population, and you can see that, for example, PDGF, IDH mutant have very strong accumulation of specific myeloid compartment. And we did also a single cell spatial analysis, and seeing is believing. So here you can see, when you look at the macrophage score, you see the difference. Indeed, the wild type have higher macrophage score. However, the mutant have also accumulation, and this is flow cytometry confirmation that in the mutant you have really. And here there is a difference in microglia between the wild type and mutant for each oncogenic subtype. But the biggest difference we found was when we look at the cell score for T effector. So we take several genes which in single cell studies were found as a gene highly expressed in T effector. And here you can see that there is reduced accumulation of T cells in the mutant, IDH mutant. And for example, this type of oncogenic alteration is associated with almost absolute lack. And this is really something we can confirm also by flow cytometry when we look for the number of T effector cells or number of exhausted T effector cells. They are really reduced in the tumor. So concluding this type of data, we would say that if we can relate this to the human situation, generally the immunotherapy should not be considered in the patients with mutations, because they have super cold immunological environment, and they just are not able to launch any anti-tumor response, even if you put a lot of immunotherapies into the system. I'm just going to conclude my presentation with just a very small piece of information, because of course it's important from the point of basic science to understand and to visualize and identify all this subpopulation, to know what the cells are doing. But we are doing this to understand and to find a new target. And we really found several targets. One of them is SPP1, but the SPP1 aberrantly produced by the tumor. And we have shown previously that this is really important for interaction with myeloid cells in the microenvironment. When we knock down the SPP1 in human tumor, there is no tumor growth at all. So this is really important for the interaction with microenvironment. So we decided to look for the peptide which will block the interaction between tumor-released SPP1 and myeloid cells. And we started with the series of peptides from the integrin. We went through the spotting the peptide, binding the recombinant protein to the peptide, selecting the peptide which on one hand bind SPP1, on other hand are not toxic to cells. We have shown that one of the peptides is very strongly blocking the interaction between tumor and blocking the invasion of tumor cells, which is dependent on the microglia. When the peptide is infused by the osmotic pump to the tumor, and then we do imaging, tumor imaging by MRI, you can see the tumor is much smaller. So this is one of the examples. We have 4 different targets based on the microenvironment. And of course, work would not be possible without engagement of my fantastic group, mostly PhD students. I mentioned some of them. And we had also tumors of different type from Maria Castro, and we collaborate with some colleagues from our institute helping us in specific projects. Thank you.
Much for this exciting talk. Thank you. Do we have any questions? Yes, so maybe it was...
There is a question.
Yes, there was a question? Yes.
Yeah, I was just wondering in the very last part when you talk about the peptide kind of immunization, or let's say using them to target tumors, do you think that this really goes into the interior of the tumor, or that it's just there better to work on the,
So in this case, the osmotic pump has a needle which directs the peptide directly to the tumor. And for 4 weeks, there is a constant delivery to the tumor core. So this is well known. And we did studies with biodistribution of the peptide, which is not great. But we work also with the nanocarriers. We have a project from Immunomed on biocarriers, in which we put the peptide into the nanocarriers decorated with mannose, which has preference to myeloid cells, and then we apply this systematically.
If you allow me the follow-up, I'm asking because for the glioblastoma, in many cases, it's just this horrible amorphous structure in many different parts, and so I'm just wondering how you would...
So our idea is not to target the primary tumor. With glioblastoma, the issue is recurrent tumor. So we believe that after surgery, we can put the peptide into the tumor niche, like a gliadal buffers, for example, and in this way we will prevent the recurrence of the tumor. So this is the idea. By changing also microenvironment and reviving anti-tumor immunity.
Thank you once again. Yes, one more question. We have time for one question.
Sorry.
Yeah, so you mentioned about some sex-specific differences in the immune response. So, well, there's like really plenty of data. So, do you already have some deeper insight? Is it just only some hormone-based or
In the Cell Reports paper, we shown that the female mice, but also human female, have more interferon-activated monocytes. There is much more inflammatory monocytes in female, so the reprogramming is slower or more difficult. We detect more pro-tumor phagocytic macrophages in males. So that's why we believe that this supports more development of gliomas in males, which is also observed in human pathology. There is almost twice more glioblastoma than in males than in females.
Okay, so it's not known what is the basis of this. It's like observation at the moment.
Excuse me?
Yeah. Okay. So thank you.
Thank you very much once again. Thank you.
Can I?
And I have the pleasure to introduce our third speaker, Professor Agnieszka Hacińska, Director of the International Institute of Molecular Mechanisms and Machines. And she has made substantial contributions to our understanding of biogenesis, transport, and degradation of mitochondrial proteins. And today her talk will focus on protein homeostasis at the crossroads to mitochondria. Professor Hacińska, please.
Thank you so much. So I would like to thank the organizers, Marek, and for this invitation. And I'm really happy to be here and contribute to this anniversary. And I'd like to switch the gears in this session a little bit and go into some basic concepts in the cell. Which concern cellular organelles, such as mitochondria. So we probably all know that mitochondria are called powerhouses of the cell because of the important role in energy conversion. Of course, that's not everything about these organelles. They also actually constitute a very important hubs for biochemical reactions. And there is no surprise that the cell really needs to know about the status of these organelles. Dysfunction of organelles of mitochondria lead to several types of diseases, very broad, spanning from actually cancer through metabolic diseases and neurodegeneration diseases. And the association of mitochondria with these diseases is clear. Sometimes what is less clear is actually a causative effect. How much actually mitochondria contribute to the cause for some of those diseases. This is still an open question. Of course, we also have some rare mitochondrial diseases which are caused by genetic diseases, but what I'd like to show with this slide to you is that these organelles are very important and their dysfunction will lead to very many diseases in all in all kinds of aspects. And I think this is basically everything what I will say today to you about diseases. I would rather say about a little bit more about some basic concepts of challenges that cell actually faces with biogenesis of mitochondria and the consequences of that stress responses. But we believe all these things have actually big impact, not yet completely clear, on the diseases that I just mentioned. So stress responses are known, several stress responses are known that operate to actually respond to mitochondrial dysfunction. So one group of those stress responses is called unfolded, so it's a derivative of unfolded protein response, the mitochondrial branch of this response, and this response is actually aims to increase the fitness, the ability of mitochondria to deal with the problems through increasing chaperones and proteases inside mitochondria. Another very important stress response that also cross-talks to other stress responses in the cell is that mitochondrial defects are actually transferred to the cytosolic translation, and they, like many other stresses that cell faces, they inhibit cytosolic translation, so the production of proteins. And this stress response is commonly known as integrated stress response, has also very many arms and several aspects and several mechanisms, but I think the common feature is actually translation inhibition here. And we have been interested for now more than actually 2 decades, starting from my postdoc, in biogenesis of mitochondria from the perspective of proteins. So mitochondrial proteome is actually quite big. In yeast, it's about 1,000 proteins. We know it very well from proteomic studies. In human, this is 1,000, so 1,500, maybe even up to 2,000 proteins. And the large majority of them, 99%, with a few really very important, but with a few exceptions, are actually synthesized on the cytosolic ribosomes because they are nuclearly encoded. So now what's happening in the cytosol, it's a very important question for us. It's very slow. Yes, okay, I have to point it here. So once the proteins are synthesized, they will go into mitochondria and specific submitochondrial compartment via unique pathways, and we have a whole selection of these pathways, but I still I would like to focus, and also the research in my lab has been focusing very much on these early stages of protein import into mitochondria, which is maybe the best depicted on this slide. So we have been interested here on this, and this is the stage, the cytosolic stage with big traffic, and that was something that we more than 10 years ago initiated when I set up my lab in Poland. It's more than basically 13 years ago, actually. So now we ask the question, what are the consequences of protein import deficiency? And we didn't look, and we posed this question not from the perspective of mitochondria, but actually what's happening in the cytosol. And maybe I go. So first of all, we even very recently we actually discovered that defective mitochondrial protein import results in mitochondrial precursor protein aggregation. So those mitochondrial precursor proteins, so the proteins that did not reach productively mitochondria, are aggregated in the cytosol. This is maybe not so surprising when you think about these proteins, because, you know, in the cytosol they are alone, they cannot assemble with the complexes, they cannot take cofactors, bind cofactors, they have no membranes. And of course, it's not really surprising that they will aggregate or form other forms of deposits. But what actually is surprising is that these aggregations, this mitochondrial deficiency, which starts with this relatively small and limited aggregation of some proteins, will accelerate, and many other precursor proteins will start to actually aggregate. And then we also found that proteins that are completely unrelated to mitochondria, even those related to models or to neurodegeneration processes, like in this case alpha-synuclein, they aggregate much more in the cell which has mitochondrial protein import defect, or in generally deficiency of mitochondria, because deficiency of mitochondria will also lead sooner or later to delays in protein import. So that was the case in yeast, where we see this alpha-synuclein aggregating in the mitochondrial import mutant. We also see the same thing in Caenorhabditis elegans when we look at the model of which has the Alzheimer's disease with beta amyloid. We see much bigger aggregation of beta amyloid. This is kind of very interesting, because this is for the first time that we call this phenomenon snowball effect. And summarizing this part of my presentation, what I showed you is that if the protein import is not efficient, mitochondrial precursor proteins stay in the cytosol, but then they start to aggregate or form create other forms of deposits, and this will actually affect cellular protein homeostasis as whole. So we have metastable mitochondrial precursor proteins aggregating, and we have compromised cellular proteome through aggregation. And this actually explains some unclear aspects of many neurodegenerative diseases, including Alzheimer and Parkinson. For all these and dementia, for all these type of diseases, actually protein homeostasis defect, so the regulation of protein homeostasis and mitochondrial dysfunction are very characteristic features. For the first time, we provide here a causative link between these two processes. And if you imagine how this can happen, starting from the small deficiency here growing more and more, we call it a snowball effect, and I think this effect can be very valid in thinking about therapies for neurodegeneration, actually preventions for neurodegeneration. So going a little bit further with these topics, we focus on the cell responses, which I introduced to you a little bit from the general perspective of those cell responses connected with mitochondrial dysfunction. So many years ago, we actually found that the ubiquitin, the major degradation machinery for proteins in the cell, which actually at that stage has not been known to operate to have mitochondrial protein as substrates, we found that this ubiquitin proteasome system very much regulates mitochondrial proteins. And to the extent that basically proteasome competes with mitochondrial import machineries for these newly born, newly synthesized mitochondrial precursor proteins that are present in the cytosol. Okay, so then that has been clear from this type of experiments, where when we inhibit the proteasome via typical inhibitors such as MG132, we get much more. Some of the proteins much more, some a bit more mitochondrial proteins, and actually inside mitochondria. So that was an important finding. And this finding has laid the ground for the entire really very intensive research field right now, which dissects, and this is now taken, many laboratories are involved in it, which now focus on the many responses that are happening within the cytosol to mitochondrial dysfunction and mitochondrial precursor proteins presence in the cytosol, and also clogging on the entrance of mitochondria at the TOM complex, which is the main entry gate for proteins. And we are also involved in this research. And then specifically, I'd like to show you, hello? Oh. Yes, so this summarizes this part that I showed you, that if you, as a mitochondria precursor proteins, if you don't get to mitochondria, you will be actually degraded by the proteasome. It has quite some consequences. And what is the stress response here? So the stress response here is that actually this proteasome is upregulated. And this our finding was originally done in 2015, and later on we asked the question, was in 2015 and concerned in yeast as a model, Saccharomyces cerevisiae. But we are also concerned and interested to see whether this kind of regulation is actually typical for example, human cells. Also C. elegans, but I will not show you the data from C. elegans. And to look at human cells, we took 2 models of mitochondrial deficiency, and they have nothing to do directly with mitochondrial protein import, because these are knockouts of complex 1, of the respiratory complex 1. But we assume that these knockouts, because of bioenergetic problems, they also cannot import efficiently proteins. And in this case, in these models, we indeed observe the increase in proteasome activity and the increase in proteasome per se, which you can see here, with the 20S as a core subunit of the core particle of the proteasome, and the regulatory particles as well. And we looked at actually we wanted to understand, both in yeast and in human, but in human it worked much better. We wanted to understand what's the reason for this increased activity, and we compared, then we purified both proteasomes from wild type and also from these mitochondrial deficient models, knockouts of complex 1, and we found one protein that was we found a protein that was actually much more present in the present compared to 0 in the proteasomes of the mitochondrial deficiency models. And this is PSMB9, and this is a very interesting subunit because this is a subunit connected with so-called immunoproteasome, which in the immunoproteasome, in the cells that produce anti-immunogenic cells that produce immune cells that produce antibodies, actually changes for normal PSMB6 subunit. And here we have a very brief look on the proteasomes. We have this core particle of the proteasome, catalytic particle, and it consists of rings, which consists of several subunits. One of these subunits is PSMB6, and this one is changed to PSMB9. immunoproteasome has 2 more subunits changed if you compare to normal proteasome. We observe usually PSMB9. Sometimes we also observe one of the other ones increased in some other mitochondrial deficiency models. But nevertheless, we have the situation that normal cells that you do not detect any immunoproteasome, when they are exposed to the actually genetically, they are genetically modified and they have mitochondrial stress related to protein import, they start to express immunoproteasome-specific subunit PSMB9. So this protein is actually expressed on the mRNA level, which was not so obvious, and you will see in a second that this is why it is not obvious at all. And on the protein level, you really very nicely see where the protein is not this immunoproteasome-specific subunit, is not present in the wild type, but when there is a mitochondrial deficiency caused by mutations, in this case, you see very clearly this protein increases. And for control, you also see that other subunits do not change, and neither on the level of mRNA nor on the level of proteins. So how this PSMB9 is regulated, and this is maybe perhaps the biggest mystery here, which we are still investigating. So first of all, we noticed one more difference in our studies, that our proteomic data and also literature proteomic data show that there is a very interesting and mysterious elongation factor, cytosolic translation. We are talking about cytosolic translation that is much more produced in the cells that have various mitochondrial dysfunction. So normally we have this version 1A, and this is a main translation elongation factor, yes, and exactly. So, and then we have 1A2 is predominant. So we have only data on this other version of elongation factor from high throughput studies, and really no function has been was clearly assigned to this elongation factor. So we were puzzled by the fact that we actually in our models very clearly see the increase of the A2 version. You see this on the level of mRNA, whereas A1, normal elongation factor, goes down. And when you look at the protein levels, this is really, this doesn't leave any doubt that basically A2 starts to exchange for A1. And now whether this A2 regulates PSMB9? Yes, it does, and interestingly, it does it on the level of mRNA, which we do not completely understand, and we think that we are missing one more player here. Nevertheless, this A2 is absolutely necessary for PSMB9, so for the immunospecific subunit of the proteasome to be expressed in these mitochondrial deficiency models. Interestingly, also those now I will come back. So we have this very big mystery with A2, elongation factor, a different form of elongation factor, and we are now currently studying this a little bit broader, trying to identify the role of this elongation factor. Nevertheless, coming back to the immunoproteasomes, or those proteasomes that are created open mitochondrial dysfunction that contain PSMB9, we also see they are enriched a little bit more than normal proteasomes on the surface of mitochondria. So that's also very interesting. So there is also interesting space regulation. Again, another topic to be investigated in the future. And now I come back to aggregates, which I started my talk, that both PSMB9 and the factor that is needed for expression of PSMB9, this untypical elongation factor A2, are both required to maintain proteostasis in the cytosol. So once we lack any of those factors, we basically we detect many more aggregates, and now we are talking about unspecific aggregates detected by one of the assays that basically does not differentiate between different proteins. It's called proteostat. So we see much more aggregation going on, and this I'm coming back to this concept of the snowball effect. And the snowball effect will happen if you don't have proper responses, so proper activation of the proteasome that comes together with, in human cells, together with the expression of PSMB9 and mysterious elongation factor, a variant of normal elongation factor that actually is required for PSMB9 expression. So just very quickly summarizing, immunoproteasome subunit is induced in the mitochondrial deficiency, and this elongation factor A2, untypical one, regulates this expression, and the proteasomes, which usually are not bound to the surface of mitochondria, those that contain PSMB9 are more enriched on the surface of mitochondria. And the final take-home message is that mistargeted mitochondrial precursor proteins that are that are delayed in the import into mitochondria, they impact cellular protein homeostasis through doing protein deposits and aggregates, and actually causing other proteins also to aggregate, and through regulation of the proteasome and the entire ubiquitin proteasome system, which I didn't speak, and through the regulation of chaperones that I also didn't speak, but you can find this in our recent papers. Thank you so much, and I'd like to thank those who were involved in the study, and I should mention Minji Kim for the PSMB9 work and Urszula Nowicka for aggregation work, and we collaborated here with Bettina Warscheid and Michele Wondruskolo for aggregates. Thank you very much, and I'm happy.
Thank you very much. Great talk, thank you. Are there any questions? Yes, there's one question. Mm-hmm.
Hey, very nice talk. I have a question concerning
Can
here.
you,
Alexandra, I'm here.
Yes,
it's just the light I see. So I have a question concerning the ATP level. So it has been recently published that ATP can dissolve aggregates. And in your mitochondrial disorders, you obviously might be producing much less of ATP. Would that be?
Yes, I didn't know that ATP can dissolve aggregates. I will come back to. I have to to get the eye contact. It's better. No. So I didn't know about that, but I'd like to comment on the levels of ATP in our system. So actually, in this kind of situation, we basically have no changes in ATP levels whatsoever. The cell actually metabolically changes. These are long-term genetic modifications, and the cells change so much metabolically that they start to produce ATP through glycolysis, and we still deal with the
Makes sense. Thank you.
Cultured cells. So it's easy for them. Thank you.
Yes, one more question. In the back again.
So can this be triggered the other way around? So first aggregates and then mitochondria?
Yes, I think I would say hypothetically that this can be triggered that way, but what I'm telling, it's not only hypothesis. So there are pieces in the literature that show that, first of all, aggregates of specific neurodegenerative related proteins, such as huntingtin, for example, they make some kind of mess on the level of outer mitochondrial membrane. On the other hand, we also know from other set of research that cytosolic aggregates some of them tend to actually accumulate on the surface of mitochondria outside. But this will, of course, lead also to protein import deficiency. So I would say, hypothetically, the answer is yes. Nobody has shown that directly, but there are several evidence in the literature which can actually support that.
Yeah, we did not show it directly, but we have aggregates and mitochondrial defects like migration, size, and stuff like this.
And aggregates of a protein that is not a mitochondrial protein, right?
No.
Okay.
No.
Interesting.
Yeah.
Okay, thank you once again for the very exciting talk. Thank you. And now I would like to introduce our next speaker, Dr. Agnieszka Ciesielska. She is Science and Technology Advisor at 10x Genomics, so a company that provides the single-cell tools. And today she is going to tell us about the future of this technology, especially for the spatial solutions. So Agnieszka, the floor is yours.
So welcome, ladies and gentlemen. It's really a pleasure to be here today with you and celebrate the anniversary of the Institute, especially that 10 years ago I had the pleasure to graduate the Institute, but I can tell you that I'm double stressed today than versus on the PhD defense, right? So eventually I would like to make a loop and kind of correlate more the talk for what have been shown already by Professor Rajewski and actually show you where the single-cell journey goes to. But I don't think I have to convince you today that the biology is very complex and that every type of biological material is built of hundreds of thousands of the different cell types and stages. And actually that heterogeneity is kind of limiting us to get the deeper insights, right, from the biological samples. So what Professor George Church mentioned is that almost everything in biology is structured, and even the function is the product of the structure. And I think in the, let's say, society of the Institute, that statement should resonate very clearly, that eventually the structure corresponds to the functionality, and the same we have dealing with in case of the tissues. So the scientists look at the tissues with the microscopy tools since actually many decades, and depends on which level of the architecture of the tissue you look at, you can answer to the different questions. Starting from the simplest one, like a tissue classification, then we move towards more deeper, let's say, unrevealing of the tissue heterogeneity when we talk about the microenvironment. That can be tumor microenvironment, that can be immune microenvironment, and then we move at the top edge, which are the mentioned before cell-to-cell interactions. So actually already Sidney Brenner 2 decades ago mentioned that the science in the next years will be kind of occupied with creating a cell maps, cell atlas, and actually trying to understand how the cells talk to each other. So I tend to say that he was kind of the visionary of two driven technologies right now. The single cell technology, but also the special transcriptomic one when we think about cell-to-cell interactions. So no surprise over here that 10x Genomics within its universe actually have all of these leading techniques in-house. And you have already heard the amazing lecture from Professor Kaminska and utilization of the single cell techniques in the studies, which also the system is located in the institute over here. But I would like to move your attention a little bit in the field of the special transcriptomics. So one part of that field is either profiling of the whole transcription or transcriptome on the tissue sections, and we have in the audience also the representative from the Institute of Human Genetics, who is a certified service provider for that type of techniques in the field of Poland. But now we are digging deeper, so we are going with subcellular resolution. And for that, we have just launched 1 year ago in situ, so the Xenium platform for actually single-cell special transcriptomics on the tissue sections.
What if you had the capability to image and analyze the expression of hundreds of genes alongside multiplexed protein in every cell of a tissue sample, and their location in situ, at subcellular resolution? And what if you could directly explore your data almost instantly after your experiment with minimal processing. Imagine the discoveries, the deeper insights into cellular interactions, into pathology that will transform how we advance therapeutic and diagnostic development. That's Xenium.
So Xenium is our newest baby. It was just launched last November. Actually, that's the platform for special profiling on the single cell level, both fresh frozen material, FFPE blocks, but also tissue microarrays. And recently, on the demand from some scientific community from Neapoli, we adjusted that platform to work on the cell lines, which can be grown on the slides as well. So just to give you a hint how the workflow in general looks, you start with the slicing of your material on the Xenium slides. That can be a pretty big pieces of the tissues. And then we follow with the targeted approach, something that was discussed on the during the first lecture. So actually we have a basket of paddle lock probes, which are then applied on the tissue. So each probe is barcoded, and we eventually follow with the detection of the barcodes via the fluorescently labeled nucleotides in the Xenium platform. So Xenium itself is like a linker of the microscope, fluorescent light microscope, and the transcriptome profiling, let's say, on the tissue section. Eventually, we do not marry H&E stained images with the transcriptomic data, because this is a unique platform, the only on the market, where the workflow is not destructive. So at the end of the story, you can take your tissue out of the machine, you can do the H&E stained or IF staining on the same tissue sections. So eventually, all of the misalignment, which potentially can happen when you take the adjacent slide, are out of your scope. So we surely give you the software for the data visualization. Why I'm saying visualization? Because that machine makes the hardest work by itself. So instead, not only it takes the images, it also do the eventually analysis of the transcript, locating them within the nuclei or cytoplasm, but it also does the segmentation, so something that most people in the lab really don't want to do by itself. It's the hardest piece of the cake, and the machine is doing that based on the DAPI-stained and implemented AI. But it also does the cell alignment and the clustering. So actually what the user is doing at the end of the story is visualizing of the genes and the cell types which were detected during the workflow itself. Every panel that we apply contains the genes which are suitable for the cell, let's say, atlasing or cell eventually assignment within. So you can visualize the data looking at the density plots, you can count the transcript, localize them. You can see each cell individually with the subcellular localization of the transcripts. So eventually, what actually you can use as a material for that platform analysis. As every new technology, it's intended first to be used in the medical field, right? So no surprise that we have already panels for the human and mouse, and actually covering pretty big baskets of the different tissue types, starting with the breast, lung, brain, multi-tissue panel. But actually that platform is intended to stay very open. So if you don't like the panel that we have in the sizes of the hundreds of genes, which you surely can customize and add up to hundreds, let's say, on top of that, which are in your scope, we can create completely custom panel in the flavors as you wish. So either starting from the very small, like 50 genes, ended up with the higher, around 500 genes. But I think it's important to mention in the Society of the Institute that it's not just a human, not just a mouse, but we can create the panel for any type of the species which you are working on. We have just launched a few weeks ago the panel designer, where you just write in an Excel the list of genes that you are interested in, and all of the bioinformatics is done somehow behind, and it's for you. That's also the platform which can colocalize isoforms, fusion genes, splice variants, so we have such data already presented.
And we are really going into covering full applications in terms of the personalized medicine field. So in our scope for the next year is identification of the expressed mutations, so on the mRNA level. And that's what we have already proven with tracking KRAS and RAS mutations on the different materials. Also working with the xenografts or mentioned before colocalization of bacteria or viruses genes within the tissue section is already in our scope that you can do. In the next year, we also plan to extend the immune profiling application. So the TCR profiling, BCR profiling, something that you can do now with the single cell applications, you will be able to allocate on the tissue section. So eventually, we also grow with the size of the panels. You might use very small ones like 50, moving through the hundreds, and next quarter, so beginning of the 2024, we will start with a big jump into the 5,000 plex. So you can see that we will cover really plan almost all of the signaling pathways, many druggable targets, and actually very many genes that can be suitable for you to even do the discovery type of analysis with the subcellular resolution on the tissue sections. And something that have been touched already by the few of us in the room is the segmentation. That's the biggest or more tough piece of the cake within the in situ analysis in general. So what I can tell you now, there is no perfect segmentation method. Surely H&E is one starting point, but it's way not enough. So what we have now, you can implement H&E, you can implement DAPI stain, you can use AI, which we implemented, but it's still not enough. So in the next quarter, we implement also the segmentation based on the membrane proteins. But sometimes your tissue, when it's sliced with the laser, it's also not in the perfect shape and the membrane might be disrupted. So in that case, also that type of the segmentation may fail. So we really extend that approaches and we would like to implement, and that's what's happening in the next quarter, actually the staining interior proteins, but also interior rRNA. In that case, any type of the tissue you are dealing with will be able to be getting the proper segmentation with actually jongling in between the methods that are already kind of in the basket of choice. So we have already over 100 machines around the globe, over 30 within the Europe in many really leading institutions. So I'm pretty sure if you wish to see the machine is running, if you wish to test it, we are able to organize it and make some testing runs on your samples. Also, first peer review papers shows up from many groups, surely oncology as a leading starting point, the paper on the breast cancer FFPE blocks, where all of the three technologies were used, single cell, special transcriptomics with the full transcriptome profiling, and in situ for confirming, for validation of all of the discoveries. Then we move towards the PDX model of the glioblastoma, where the Professor Gregory Simon was actually colocalizing also the isoforms of the EGFR gene, and none of the other, let's say, commercial approaches for the in situ is able to do this. Then we go over the immune microenvironment profiling and ending up with the neurodegenerative diseases. So that's the starting point. I haven't mentioned, but in our scope are also the plant analysis, plant material analysis. So eventually that platform actually will be utilized in all of the different materials that in the institute is actually used. So I would like to encourage you at the end to maybe listen a webinar which will be held by the Professor Luciano Martellotto from the Adelaide University. He used to be the head of the single-cell core facility at Harvard as well. He did many analyses with many different platforms. He has a lot of experience in his hands, and he will be sharing that experience on the 15th of November. So if you are interested how easy it is to use, what are important tricky points in the in situ, what could be the drawbacks and advantages, I would highly recommend to register. So eventually, if the pointer will work, yep, I started with the Sidney-Brenner, and I would like to end with the Sidney-Brenner sentence. So, as he said, the progress in the science depends on the new techniques, new discoveries, and new ideas. I don't agree with that order. I think it's the new techniques, new ideas, and then you do the new discoveries. So if you have in mind any type of discovery, you would be needing that technology, please get in touch with us. We will be there and happy to chat to you. Thank you.
Thank you so much. Thank you very much. Yes, can you hear me? Not really. Yes, okay. Yes, so thank you very much for this great talk. Are there any questions? I am curious about the software solutions for that. So how does it work, and what are the major advantages of because this is image analysis, right?
So actually the biggest advantage of that solution is that you don't get any FASTQ data, FASTQ files like from the sequencing, where many of the users actually are a little bit reluctant to start the analysis if they are not familiar with the sequencing. Over here, you actually get the transcripts located and within the tissue, and you don't do any bioinformatics behind because it's done already by the machine. So what is to be done by the users is the visualization. So you simply plug and play within the software, and you show the genes you are interested, you show the cells which you are looking for, and it's actually the software which works on any type of the classical computer, no special computing power, right?
Is it opened also for other, custom-made AI approaches or something like that? Is it like, can it be used for custom pipelines as well?
Yeah, so eventually the machine generates data in a pretty open format. So if you would not like our software, where it's actually pretty good one because you incorporate H&E, you incorporate IF, none of the other solutions does it, right? But if you don't like it and you have a lot of experience with the bioinformatics, you can use the data from the machine and go to setup package, Python, Giotto. It's a really pretty open scope. With the nanostring, there are lots of other issues, let's say. First of all is that we analyze regions of interest and not the full tissue section. So you have a lot of problems with linking. Anyhow, if you would like to discuss in the more deep, we will be there because I guess the time is running.
Thank you once again. Thank you very much, Agnieszka, for this great talk. And now I would like to introduce Dr. Paweł Świtoński, our next speaker. He is the head of the Department of Neuronal Cell Biology at the Institute of Bioorganic Chemistry. And his research has focused on neurodegeneration, and today he will share some latest results with us. Paweł, the floor is yours.
Good afternoon, everyone. Let me first start with saying big thank you to the organizers for inviting me to such a great and wonderful event. So what I would like to tell you today is our latest approach in studying and understanding rare neuronal populations in the context of neurodegeneration, particularly in disease that is called spinocerebellar ataxia type 7. So what is spinocerebellar ataxia type 7, or SCA7 for short? It's a genetic neurodegenerative disease that is caused by a mutation in a single gene, the gene called ataxin-7. And this is interesting mutation. It's this dynamic elongation of CAG triplets in the coding region of ataxin-7 gene. As a result, we see formation of this elongated polyglutamine domain in the protein product. And we know that this domain is toxic, and it induces this cascade of events that eventually lead to degeneration, dysfunction, and death of Purkinje cells. And Purkinje cells, you probably can see them here in this yellow part. in these pictures are big GABAergic, so inhibitory neurons that live in the cortex of the cerebellum. And they are very important for the proper function of the cerebellum, as they are sole output of cerebellar cortex. These cells, we see them degenerating in spinocerebellar ataxia type 7 and other spinocerebellar ataxias. And since they are so important for the proper function of the cerebellum in patients, when we see this degeneration, we see a cerebellar degeneration, as you can see here on these CT scans in patients. Patients suffer from symptoms related to this brain structure. So they have problems with coordination, with the movement, synchronization, problems with walking and talking, grabbing things. This is really, really a bad disease. It's not curable at this point, and it always leads to death. So when we take a step back and we look at the neurodegeneration from some perspective, we will notice that there is this pattern of neurodegeneration that we observe in different neurodegenerative diseases. Some specific populations, subpopulations of neurons are more affected, more sensitive than the others. And I just mentioned Purkinje cells in spinocerebellar ataxias. Other examples are dopaminergic neurons in substantia nigra and Parkinson's disease, or motor neurons degenerating in ALS. And there are many other examples of this phenomenon, and this phenomenon is known as selective neuronal vulnerability. And the question why these cells, some specific population of cells, are more sensitive than the others, are more prone to degeneration and death in different neurodegenerative diseases, has been being asked for like many decades now, since we started looking at neurodegeneration, neurological diseases using molecular tools, and we're still waiting for a good answer for that. It's still not clear entirely from mechanistic point of view why it happens, and this is also the question that we are asking in my lab in the context of spinocerebellar ataxia type 7 and Purkinje cells. And there are many established methods to study rare neuronal populations, including very laborious and sophisticated methods like laser capture microdissection, trap assay that you can profile mRNAs being translated in a selected population of cells. And we live now in the era of single-cell multi-omics, like tools that are designed to ask questions like these about selective vulnerability, tools that we are also using in our research, but they all suffer from this one fundamental problem. There are not that many of these cells in the brain to start with. So we can, using sequencing, single omics, we can probably brute force it to sequence hundreds of thousands, if not millions of cells to get enough information to talk about these rare neurons, but usually is not like a valid approach. We cannot always do that. So when we first thought about tackling this problem, we had the idea that we might optimize several things here. Our idea was to maybe we can isolate these Purkinje cells from the tissue and then enrich our samples to use it later in multi-omics. And first thing that we did was to reduce the complexity of the system, of the problem. Because when you work with cells, when you try to isolate cells from tissue, it's not an easy task, especially when we're talking about these neurons, oddly shaped, fragile neurons, like with intertwined neurites. So it's not really easy to do it. But we decided maybe we can focus on just nuclei of these cells, because nuclei are still pretty good to work with. They still have a lot of information in them, all genetic and epigenetic information, partially transcriptomic information. and they are way easier to work with. They're way easier to handle, to isolate. So this like reduction in this complexity of the system was the first thing that we did. So we decided to focus on nuclei and to try to isolate these nuclei from the sample of just Purkinje cells. And the way we decided to do it was to take a mouse line, reporter mouse line that was already available. These mice have the expression of the protein, which is integral part of nuclear envelope. It's called SUN1 with additional GFP tag. If we cross this mouse line with another mouse line that expresses Cre recombinase under the control of PCP2, so Purkinje-specific promoter. I'm not going to go into more details here about like mechanism of this induction, but what we can basically do, simplifying things right now, what we can do is to genetically tag with fluorescence nuclei of Purkinje cells, only of Purkinje cells. And this is the approach that we took, and the results that we got were not very fantastic. This is like we were kind of disappointed. It was not what we were looking for, because like ideally we would have these Purkinje cells, these big Purkinje cells. You can see them here, like denoted with arrows. Only them being, positive for GFP. The problem was it wasn't the case. There was like virtually all the cells in our cerebellum were positive for this GFP, and that was disappointing. But eventually it led us to discovering of even better technique to isolate these cells. But for now, what was kind of disappointing. The reason, like you can think about many reasons. One of it is germline recombination or promoter leakage, stuff like that. But either way, we got all the cells positive. But we decided to do what we planned on doing, so we just repeated that. We isolated these nuclei, and we used flow cytometry to look at them. And by looking at specific features of these nuclei, like the ability of them to scatter light and intensity of this GFP signal, we saw that on these flow cytometer plots, these nuclei form different clusters. And one of them was particularly interesting, high on side scattering and high on GFP. And we thought, we hypothesized, these are our Purkinje cell nuclei, and we can try to sort them out and test it. So we did a bunch of, the battery of tests. We looked at the expression of markers specific for Purkinje cells. We looked at their size, morphology. We looked at, finally, at the number of the nuclei. This is a distinctive feature of Purkinje cells. They have one centrally located big nucleolus. And basically, we were able to confirm that this population of nuclei in these plots, cytometric plots, are Purkinje cell nuclei. So we did it, and we were like really happy about it. But then realized we had another idea. Since we were able to do it just using, we were able to do it even though we had all these cells, all these nuclei positive in the sample. Could we possibly replace this genetic tagging, this long genetic breeding process, and just reduce it to just immunostaining? So we thought about maybe we can come up with the idea of using like protein that is present in the envelope, on the nuclear envelope, is highly expressed in Purkinje cells, and if we stain it, can we get the same similar results? Can we get these this cluster that might be the cluster of our Purkinje cell nuclei? So we actually did it. We selected the protein that is called RAMBP2, which is a part of nuclear pore complex. And surely enough, by just staining these nuclei with RAMBP2 antibody, we were able to see this cluster that we also confirmed that this is the cluster of Purkinje cell nuclei. So that was fantastic. We were able to like just contract weeks and weeks, if not months, of protocol time of breeding these mice to just like simple 4-hour experiment that you can do it, Monday morning on the bench. So this worked, and we now have this protocol to, in very efficient and very cheap way to get these Purkinje cell nuclei out, and now we can increase the concentration of our cells for our single cell omics. We can go from less than 1% of these cells to like 50% if we want, in very controllable and repeatable manner. But this is like these single cell omics is not the only thing that we can use, to study our cells. And I would like to very briefly tell you about our new exciting approach that we doing using this protocol. And it involves using artificial intelligence. It's a very hot topic these days. Artificial intelligence, basically what we're trying to do is use the potential of machine learning classifiers to visually inspect our nuclei for potential phenotypes that they have in nuclei of Purkinje cells, SCA7 Purkinje cells. And how do we do it? Having access to high throughput, image flow cytometry, the machine that can take pictures, thousands and thousands of pictures of the nuclei or cells that you're using. We can now create, these thousands and thousands of wild type and SCA7 Purkinje cell nuclei and use it as a training set for neural network classifier. And you can think about neural network classifier as big and complicated, but still mathematical function with many terms. And what you do, you give this function, this training set, and you ask it to tweak all these parameters in this, iteratively until it comes to like final form, where it can, this function can now classify, can assign right class of the image, either SCA7 or wild type in this example. And if you look at our results, our efficiency test at the bottom, just using hex alone, so staining these nuclei with DNA, to visualize DNA, and training this neural network model, we were able to achieve more than 90% of efficiency. So in 90% of the time, the model assigned right class to the image. So it was incredible, because now we know that this model has information. It knows what is there to differentiate these two cells, two different set of cells. We don't know that yet, but we can ask it, and we did. And we used like this grad-cam method, which is like class activation mapping. Simple words, you can put like a heat map on your cells to ask, which parts of the image the model focused on the most during this classification process. And what we saw was like these beautiful image of these like hot spots around central nucleolus. And when we look at the image it came from, these are these like strong hex stainings, these heterochromatin stainings around nucleolus. So it is very exciting. It gave us a new way, like this approach gave us a new way to study degeneration in these cells, to look at heterochromatinization and epigenetics, and we are very exciting in developing this project alone. So we want to share it with the community. We're still working on it. So with this, I would like to thank all the people that were involved in this project, especially people in my lab, Grażyna Adamek and Mo Fakhri. Without them, like this wouldn't be even possible. I like to give like huge thanks to Laboratory of Single Cell Analysis, people like very skilled and very great to work with. This project would not be possible without them. I thank also other people, external collaborators, and thank you for your attention. I'll be more than happy to take any of your questions. Thanks.
Thank you very much for your lecture and for the nice words as well. Yes, are there any questions? Do we have any questions? Maybe I will ask one question about this machine learning approach for that data analysis. Was that completely unsupervised, or how did
this was supervised learning? So what we did, you know, in this classification model, you basically, you know, give this big training set, you know, with labels. So each image in the training set is labeled with the right class, and then you start with with random numbers in this big you know, function, which is neural network, and it's just do this, you know, tweaking all these little knobs there, and test itself if it did the right choice, and now looks at these labels and see, okay, I did it right here, but wrong here. So basically, it improving itself step by step until it goes to you know, until you train your model enough, so it's very you know, trained and can do predictions. So here we doing kind of I don't want to say opposite idea, but we are training our models with these stainings, different stainings, because we can use other stainings, we can use other antibodies to stain it, and then, you know, how much it trains tells us about how much information is in these stainings that can differentiate these cells, and then we can try to reverse engineer the process to look what actually it focused on, and this is the only way that we can get meaningful biological information back.
Okay, thank you very much. One more question.
So what will you do next with these spots? You have spots in the nuclei. How will you use it then for?
Yes. So as you mentioned, this is just the first step. So this is sort of unbiased way to find features, find hypotheses to work with. What obviously this is not the only way that we can inspect these cells, and we do other analysis. So we have this hypothesis right now that these Purkinje cells are, you know, they're very open chromatin cells that are, you know, normally when you look at all these proteins that can modify chromatin, histone acetyltransferases, they're highly expressed in Purkinje cells, and we see it. And what we think, these cells are normally open, and when we look at our Purkinje cells in our single cell analysis, we saw that these enzymes, these proteins that open chromatin even more, they're even more highly expressed in our SCA7 cells. So the hypothesis right now is that we have these open cells with open chromatin that is getting even more open and more prone to, let's say, DNA damage, for example, problems with later transcription and so on. And this analysis that we did with artificial intelligence approach kind of support this idea, because, and I didn't tell you that, but now what we are trying to do is to look at these spots, these heterochromatization spots, and see if these are bigger, smaller, where they are located in the nucleus, and it kind of agrees with what we saw.
Kind
before.
of an assay, yeah.
An assay.
An assay, like a big spots, small spots.
Yeah, you can use several things to do it. You can use ImageJ or Cell Profiler. There are tools that you can actually measure it and then compare it to between SCA7 wild type, between Purkinje cells during the disease course in young animals, older animals, and symptomatic animals. So there are many ways you can do it.
So if the cells are degenerated, for example, in the mice, but you will still isolate them, but are they dead and the nuclei are there, or are they just degenerated?
Yeah.
You know what I mean?
Yeah. This is like the key point here, and I think that like I would normally don't suspect that nuclei will still be there if cell is dead, but yeah, this is important thing, and we probably might have a situation when we select, for cells that are like more healthy than the others, because like other died, and this is like really important thing to have like when you analyze the data.
Okay, thank you very much.
Thank you.
With this, I would like to thank all the speakers of this session. And now it's time for lunch, and please make sure to be here on time on 2:50 for the next very exciting session of this conference. Thank you very much.
like to avoid this more noise once you are. The last session was horrible, you know. Okay, good afternoon. I would like to welcome you in this afternoon's session, in which we are going to learn a little bit more, but also at the same time underline the role of different model organisms in various types of the research. We have a very heterogeneous, but at the same time very interesting lineup of speakers, and without any further delay, I would like to invite for a stage Professor François Chaumont from the Institute of Biomolecular Science and Technology of Louvain-la-Neuve University in Belgium. And his major interest is in the membrane proteins, which are molecular determinants behind the water status in plant. And today he's going to tell us a little bit more about aquaporins.
Yes, so thank you very much to the organizer for inviting me and for giving me the opportunity to share with you the recent data we obtained in my laboratory. So I'm very honored and very pleased to be here and to celebrate with you the 35th anniversary of your institute. So as Michael told you, I'm very interested by the function and regulation of aquaporins. And because we are in the live at the organismal level session, so I will talk about the role of aquaporins in relation to the water relations and to the plant physiology. So my talk will be divided in three parts. So first I will introduce you water movement in plants and aquaporins. Then I will talk about plasma membrane and aquaporins. And I will talk about the expression and the activity in maize, roots, and leaves. Finally, I will focus on specific cell types, cells that compose the stomatal complexes, and I will show you that aquaporins are important for the dynamics. Okay, let's start with water movement in plants and aquaporins. So you all know that water always moves from a place where water potential is higher to a place where water potential is lower. And in plants, it is the case during transpiration, where water evaporates at the leaf surface due to the dry atmosphere, and this creates a tension in the leaf, the stem, and the roots to trigger water uptake. Long-distance water transport is also observed during the distribution of sugars synthesized during photosynthesis. And this involves also long-distance water movement. But of course, water movement is also important at the cell level. For instance, for the stomata aperture and closure, I will talk about that later on. And also to regulate the cell turgor, the cell volume, and the cell elongation. So water and solute movement across tissue, across plant tissue, is possible through three different pathways. First, we have what we call the apoplast pathway, where water and solutes move around the protoplast through the cell wall. Then we have the movement from cell to cell through small connection called plasmodesmata. And this movement is called the symplastic pathway. And finally, we have the movement through the membrane, the plasma membrane and the tonoplast membrane, the vacuolar membrane. And of course, if we are talking about this pathway, named transcellular pathway, we are thinking about aquaporins. Aquaporins are small channels, about 25 to 30 kilodalton, and they are composed of 6 transmembrane domains connected by 5 loops, A, B, C, D, and E. And you can see here that the loop B and E form short helices that dip halfway in the membrane and contribute to the pore specificity. Here you have a 3D representation of the first aquaporin that have been identified by Peter Agrey in US, and he got the Nobel Prize for this discovery in 2003. And here is the 3D structure of this aquaporin, which is present in the erythrocyte. And you can see here the 6 transmembrane domains that are a little bit tilted in the membrane, and the 2 short helices, the B and E, that are also found in the membrane. And this constitute like a funnel in which water molecules can move as a single layer. Another feature, is that aquaporins assemble as tetramers. It means that in the membrane you have 4 monomers acting together. Each monomer contains a pore, so it means that in the tetramer we have 4 pores. And this is important to regulate several aspects of the trafficking and the activity of the channel. So aquaporins constitute a large and divergent protein family in plants. In humans, we have 13 genes encoding aquaporins. In plants, we have much more aquaporins. For instance, in maize, the model plants I'm studying, maize contains 43 genes encoding aquaporins. And we have some species, like cotton, that have more than 100 aquaporin genes. So it's quite difficult to understand why, but anyway, we will try to figure out why plants have so many aquaporins. So here you have a phylogenetic tree coming from comparison of the different proteins from maize, and we can see that we have 4 subfamilies. So we have here the families that contain the PIPs, the plasma membrane intrinsic proteins that are located mostly in the plasma membrane. Then the proteins that are located mostly in the vacuolar membrane, the tonoplast, and called TIPS for tonoplast intrinsic protein. Then we have a subfamily called not-26-like intrinsic proteins, and most of the proteins are located in the plasma membrane. And here we have 3 guys that are located in the ER membrane. The function is still not known exactly, and they are called CIPs. So the numbers is also related to the localization, to the expression, I will talk about that later on, but also to the specificity of this channel. Indeed, aquaporins can transport other solutes than water. So for instance, the PIPs can transport hydrogen peroxide, carbon dioxide, urea glycerol. We have ammonia, which is also a substrate for the TIPs. And for the NIPs, I mean, they are the more divergent protein according to the substrate. They are able to transport metalloids that are like boron, silicon, arsenide, and boron is an essential element for the formation of cell wall in plant. So today, I'm going to focus on the PIPs, the plasma membrane intrinsic proteins. And research done in my laboratory, but also in many different laboratories around the world, show that these channels are highly regulated at different steps. Of course, we have regulation at the transcriptional level and the translational level, and then the protein uses a secretory pathway to reach the plasma membrane. And this journey through the secretory pathway involves many different protein-protein interactions. First, we demonstrated that different aquaporins can form heterotetramers. And this is important to address specific proteins, specific aquaporins to the plasma membrane. But there are also many other proteins interacting to the aquaporins in the secretory pathway. Once in the plasma membrane, the aquaporins can be internalized for resecularization, but also for degradation through the proteasome, but also through autophagy. Once in the membrane, the protein can be active or inactive. There is a gating mechanism opening and closing these channels, and this is due to post-translational modification like phosphorylation or protonation. All these steps are regulated by phytohormones, the circadian clock, and signaling molecules, and are, of course, dependent on environmental cues and the development. So in my lab, we developed several tools in collaboration with different laboratories. Right now, we are focusing on the transcription of the genes, and we are working a lot with the mechanism regulating this and the identification of transcription factors. And to do that, we perform genome-wide association studies and EQTLs that allow us to identify factors, transcription factors that are regulating aquaporins. We are doing also cell-specific RNA-seq, a lot of qPCR, and we are also doing a lot of macroscopy, confocal macroscopy, to follow the interaction between different proteins in the secretory pathway. And then we are able also to measure the activity of the aquaporins once in the membrane by doing expression in different cells, for instance, in xenopus oocytes, or in yeast, or also in plant cells. We are also very much interested to measure the role of aquaporins in plant, and for that we use different biophysical methods. We are measuring transpiration, stomatal conductance, and so on. I will show you some examples in a few minutes. Okay, so now let's talk about expression and activity in maize root and leaves. So here I'll show you a representation of a main root of maize, and you can see that there are different zones: the zone of cell division, zone of cell elongation, the maturation zone, and the mature zone. And what is interesting to see, to observe, that in the maturation zone and mature zone, we have a suberization of specific cells that prevent the apoplastic pathway I showed you before to occur. It means that the water, to continue its movement through the plant's body, need to enter the cells. And these are called the endodermis and the exodermis. So to make a long story short, we show that all pips, except one, 12 different pips in maize, are expressed in roots. And the mRNA and protein amount increase with the root developmental stages, with a low expression at the very tip, as you can see here for one example, PIP25, which is the most expressed aquaporins in maize root, and then its expression increase in the elongation zone and is maximal here, where the water uptake is also maximum. And interestingly, we also show that PIP25 is correlated with the presence of apoplastic barrier. When we grow the plant in an aeroponic condition, it means that the roots are in a humid air, and we pulverize the nutritive solution every 30 seconds. In this case, we observe the development of a suberized endodermis and exodermis. And when we look at the protein level by immunocytochemistry, we can see that PIP25 is highly expressed in these cells. So here we have expression in the exodermis, in the cortex cells here, and also in the endodermis. On the other hand, when we grow the plant in hydroponic condition, it means that the roots are in the liquid medium with aeration, of course. In this case, we do not observe any suberization of the exodermis. And actually, we cannot see expression of PIP25 in these cell layers. So we have a very good correlation between expression of PIP25 and the presence of apoplastic barrier. We also show that the expression of the aquaporins are more abundant during the day than during the night. During the day, what we have a transpiration, the stomata are open, a lot of water moving through the plants, and in this condition, we have a lot of aquaporins expressed. And actually, we were able, using this cell pressure probe, to correlate the expression of aquaporins with the permeability of the cortex cell membrane. To do that, we use a thin capillary, and we are able to introduce this capillary in cortex cells in intact roots, and this device gives us information about the trigger pressure of the cells. But we are also able to increase or decrease the trigger pressure, and we are recording the time required to get back the initial trigger pressure. If you have many active aquaporins in the membrane, this time will be shorter. And analyzing the data, we were able to demonstrate that believe me? Yes. So you can see that the permeability of the cortex cell membrane is higher during the day than during the night when we have more aquaporins expressed. So we did also experiment with the leaf, and the story is similar. So we also analyze the expression of the different PIP aquaporins in the different part of the leaf. We have also something which is regulated by the developmental stage, low expression at the very tip, and then expression that increase in the elongation zone is maximal here when the leaf, the maize leaf, exit the sheath of the older leaf, and then in general the expression decrease in the mature zone. You can see here, for instance, this kind of pattern for PIP11, low expression, increase and decrease. I'll show you also PIP25 because I'm going to show you data about this guy later on. So PIP25 is very lowly expressed in plants, but actually it's expressed, and its expression is maximal in the mature zone. And I will show you that this expression is specific to several cell types. Okay, so now we wanted to know the physiological roles of PIP25, and then we obtain maize plants deregulated in its expression. So we have plants that are overexpressing PIP25, and you can see here the Western blot showing that in low wood we have an increase of expression in the PIP25 overexpressing line. And we can see this better in the leaf, because in the leaf there is very few proteins present in the wild-type plants, but then when we overexpress PIP25, we can see a very strong signal. We also obtain a line which contains a transposon in the gene, so we have a knockout line. And in this case, you can see that in the root, the PIP25 signal dramatically decreases. In the leaf, we cannot see any difference because PIP25 is not very well expressed. Okay, so now we analyze these plants, and the first things we did, we use a cell pressure probe I just explained, and we look at the hydraulic conductivity of the cortex cells. And when PIP25 is overexpressed, we increase the membrane permeability. It was expected. And when we knock out the PIP25, we have a decrease of the osmotic water permeability. Now we measure the whole wood water conductivity. So we were analyzing the amount of water going through the whole wood. And interestingly, we did not see any difference in plants that are overexpressing PIP25. We were a little bit puzzled by that. While when the PIP25 is knocked out, we have a decrease of the wood hydraulic conductivity. How can we explain this data? So we hypothesize that in the cells, we have specific gatekeeper cells that are controlling the water movement. And of course, we were thinking about endodermis, this cell layer that can be suberized. And actually, to address this question, we use a mathematical model developed by a colleague in Louvain-la-Neuve in Belgium, Valentin Couvreur and Xavier Drey. And this model actually computes the radial flow of water across the root, taking into account the plasmodesmata, the membrane, and the apoplast. So using this model, we were able to test different hypotheses. And to make a long story short, we show that the experimental data we obtain is due to the fact that in wild-type plant, we already have a saturation of the aquaporins in the endodermis. It means that there is no need to overexpress more aquaporins to have more water flow. We have already maximal permeability of the endodermis. Okay. Okay, so we also analyze what's happening at the leaf level. So we measure the permeability of mesophyll cells. To do that, we prepare protoplast, and we are performing swelling assay. The swelling rate will be faster if you have active aquaporins. And we observe for the overexpressing plant an increase of the mesophyll cells permeability, and no difference for the knockout plant because PIP25 is not expressed in these cells. And also, when we measure the flux through the whole leaf, we also observe an increase of the leaf hydraulic conductivity in the overexpressing plant. Okay, so let me briefly conclude this part. So most PIP aquaporins are expressed in maize, roots, and leaves. The mRNA and protein expression changes according to the developmental stages. Overexpressing of PIP25 genes results in higher mesophyll cells and leaf water conductivity. And the absence of PIP25 gene expression decreases the cortex cells and the whole root water conductivity. And I show you also that expression of PIP25 in the exodermis and endodermis regulates the radial flow of water. So the last part of my talk will address what's happening in the stomatal complexes. So here you have a representation of a grass stomata, like in maize. And actually these stomata are highly responsive to rapid change of the environment. It means that they can open and close more rapidly than other species. And this is mostly due to the cell form. I mean, the grass stomata are formed by two dumbbell-shaped guard cells here, surrounded by two subsidiary cells. And actually, what we observe in these two cell types is a reciprocal regulation of cell turgor during the aperture and the closure of the stomata. Here, for instance, to trigger the aperture of the stomata, you have accumulation of ions and osmolyte coming from the subsidiary cell that accumulate in the guard cells, and then we have a movement of water. And for the closure, it's a reverse process where we have the movement of ions and metabolites inside the subsidiary cells and movement of water that decrease the turgor pressure in the guard cells and increase it in the subsidiary cells. And we were very much interested to what's happening in the plasma membrane of these cells. There are plenty of ion transporters that were characterized in guard cells, but much less is known in the subsidiary cells. And in my lab, we are very much interested by knowing what's happening for the aquaporins, for the plasma membrane aquaporins. I mean, there are studies performed in Arabidopsis thaliana that show that aquaporins are indeed expressed in guard cells, and this activity of these guys are regulated by abscisic acid, which is an hormone which activates the closure of stomata. And there is a signaling pathway that activates a kinase, OST1, that activates different transporter, anion transporter, but also aquaporins. And it was shown that aquaporins is able to facilitate the diffusion of hydrogen peroxide, which is also a signaling molecule important for the closure. And at the same time, the aquaporin can facilitate the diffusion of water out of the cells. And this process is decreasing the turgor pressure and inducing the closure of stomata. There are also reports from Julian Schroeder in the US that show that PIP21 is able to transport carbon dioxide, which is also a signaling molecule to trigger the closure of stomata. And this is another aspect of aquaporins which could also be involved in the transfer of these solutes. So we were very much interested to know what are the aquaporins expressed specifically in the guard cell or in the subsidiary cells. And actually, we micro-dissect both cell types from maize epidermal cells. We did that during the day and during the night, and we perform RNA-seq. And I will show you only here the data regarding the PIP aquaporins. And once again, we are very surprised to see that there are many different PIP aquaporins expressed in these cells, in the guard cells here. And the expression is higher during the day than during the night. It's the same in the subsidiary cells. So we have also expression of several aquaporins. And in general, expression is higher during the day than during the night. And when we compare what's happening in the subsidiary cells and guard cells, we can see that we have differences for some isoform. Some of them are more expressed in the subsidiary cells, like PIP21, and some are more expressed in the guard cells, like PIP25. So showing that, we were interested to know, okay, so let's have a look to the plants I explained to you before. I mean, deregulated in the PIP25 expression. What's happening at the guard cells level? So to do that, we first demonstrated that PIP25 is indeed expressed in the stomatal complexes and the guard cells. So we prepare maize lines expressing PIP25 fused to YFP under the control of PIP25 promoter. We can see expression in the epidermis and the stomatal complexes. And in some plants, we have a restriction of the expression in the guard cells. So in the transgenic plants we characterized before, we can see also in the overexpressing plant an increase of the protein in the stomatal complexes. And in knockout plant, we can see a decrease of the expression. So it's a good genetic tool to analyze what's happening at this level. So the first thing we analyze is a transpiration in different conditions. So here we have an image of the transpiration of plants in the dark, in the day. In well water conditions, there are plenty of water in the medium. And what we can see is the overexpressing plant transpire more water, the pink trace here, than the wild type plants. And for the knockout plants, we have a little decrease of the transpiration. But when we grow the plant in a mild osmotic stress, the pattern is reversed. Actually, we observe that the PIP25 overexpressing plant transpire less. You can see here, compared to the wild type, and the knockout plants transpire more. How can we explain that? So we are talking about transpiration, and transpiration is, of course, dependent of the aperture and closure of stomata. So then we analyze the stomatal conductance of these plants, and we use also ABA to trigger the closure of the stomata. Here we analyze the stomatal conductance using this machine, the LICOR, and we follow the stomatal conductance upon addition of abscisic acid. And you can see that when we do that, when we apply ABA, we have a faster closure of the stomata in the overexpressing plant. And this is reverse for the knockout plants. The knockout plants close the stomata much slower. And we also measure the aperture of the stomata upon ABA treatment. In this case, we use epidermis, peeled epidermis. We have the stomata here, and we measure the aperture of the stomata. And upon addition of ABA, we can see that we have a faster closure of the stomata compared to the wild type. And for the knockout plant, the stomata are not closing. They are insensitive to ABA. I showed you before that aquaporins could be also a channel for hydrogen peroxide, and we quantified the hydrogen peroxide in guard cells in the PIP25 overexpressing plants after addition of ABA. And here, after 5 and 10 or 10 minutes, you have, after 2 and 5 minutes, we have a huge accumulation of H2O2, which is much faster compared to the wild type. So meaning that PIP25 is also a hydrogen peroxide channel, and actually we demonstrated that by expressing PIP25 in the plant suspension cells, and we were able, using a hyper probe, to demonstrate that this the expression of this channel allow accumulation of H2O2 inside the cells. Or we did also some experiment in yeast. So let me conclude this part by this model. So after a wild water deficit, we have an ABA increase, signaling molecule that is going to activate PIP25. The activation of PIP25 can favor the inward hydrogen peroxide diffusion, and activation of membrane transport system and release of ions and solutes. But activation of PIP2.5 can also trigger the outward water flow by PIP2.5. I mean, the water is going out, and both processes decrease the trigger pressure, close the stomata, and reduce the transpiration. So it means that overexpressing PIP2.5 can be beneficial for plant growth in mild water deficit, allowing water conservation and also a larger leaf elongation rate. I did not show you the data, but it is the case. We have a longer elongation rate for the plant overexpressing PIP2.5. So it means that PIP2.5 could be a nice target to optimize the water use efficiency of plant and to try to conserve this water in stress condition. We have also plenty data concerning another guy, PIP1.6, and overexpression of this guy enhances both stomatal aperture, which is not the case for PIP2.5, and closure, and this is dependent of the carbon dioxide concentration. And right now, we are generating plants that are knockout for aquaporins, but specifically either in guard cells or in subsidiary cells. And we are currently analyzing these plants. Okay, so I am going to stop here. I would like to thank the people who performed the work. So today I show you data from Thomas Millet, Charles Haché, Robert Hayden, and many data coming from Lei Ding, a talented Chinese guy who is here. And of course, we also collaborate with plenty different labs. And the transgenic we obtained in Ghent, in Belgium, in Dirkinze laboratory. We are collaborating with INRAE in France for the laser microdissection and also to characterize the plants. And yes, I'm going to stop here. And thank you for your attention.
Thank you. Thank you so much. Understanding these molecular determinants, important for the water content in this context of climate change, is very, very useful. So I open the discussion. Questions, comments, common interest? Yes, there is a question.
Yeah, nice talk, thank you. So you showed that basically at the end of the day, aquaporins level decreases, yes, expression. What's happened to those which are already in the membrane? All of them gets degraded, removed from the plasma membrane to resynthesize a new one?
So actually we did not follow the dynamics of these proteins at the end of the day, but there are plenty possibilities. Either they are vesicularized, I mean they are endocytosis, to be either degraded or to be kept to come back later the next morning in the plasma membrane. I mean, we still have aquaporins in the membrane, but these proteins can be active or inactive. So we can also imagine that protein in the membrane that stay in the plasma membrane during the night could be also inactive due to post-translational modification. But we do not have any clues about the fate of the aquaporin that are. But I mean, here I show you actually Western blots. So it means that they are degraded. Otherwise, we should have see them.
Yeah.
Because, I mean, we are purifying microsomal fraction. It's not from purified plasma membrane. So, okay, so they should be degraded through proteasome or through autophagy. Yes, definitely.
Okay, so in your overexpressing line, then you would think that it's kept overnight or gets also...
We don't know, actually.
Okay.
We don't know.
Other questions? There's one there. Pavel?
So I have the question regarding this impact on stomata. Yeah? So because maybe I somehow overlooked this. Is it somehow specific for the one you tested, or you think any other could have like a similar impact from those, of course, plasma membrane aquaporins? So.
You are talking about the specific wall? That's what you mean?
Yeah, I mean the impact on the stomata closure. Yes. So is it like you've...
But as I showed, I mean, aquaporins can be involved in different functions. So for the water, for the hydrogen peroxide, and for also carbon dioxide. I did not show you, but PIP16 we analyzed is able to transport carbon dioxide. So it means different function. And the main question is, why do we need so many different aquaporins expressed in these cells? And so, for instance, the PIP1s, which is a group of plasma membrane aquaporins, are not able to reach the plasma membrane by themselves. They need to interact with PIP2s within heterotetramers. So we know that the PIP1 are carbon dioxide channels that are important to be in the plasma membrane. So we can suspect that specific PIP2s are required to transport this channel to the plasma membrane of the stomatal cells. So, of course, it's difficult to figure out, especially, the role of each isoform. But what is funny is, even by disrupting one aquaporin, PIP25, we are able to see phenotypes. So the idea is to perform that with all the different isoforms to know exactly the function. And the technique, the cell-specific CRISPR-Cas9 technology would be useful to do that. Especially because in this case, we only address the deregulation of aquaporin in these cells. For the PIP25 overexpressing on knockout plants, these plants are deregulated in all the plants. It means that we have more aquaporins in the root, more aquaporins in the leaf, and we know that this high expression of aquaporin can affect the stomatal functioning by hydraulic signals. So that's why we like to analyze, for instance, the stomata aperture and closure, taking into account only, peeling the epidermal cells. So what is happening is due to the activity of the aquaporin expressed in these cells, and not due to the expression in the roots or in the bundle sheath cells in the leaves that play also an important role. So it's quite tricky to figure out exactly the function of this, but we try to develop tools to address this.
Okay, thank you.
Okay, thank you so much. And now it's time to move from maize to mouse. To mouse, not yet Cenorhabditis. We are moving from maize to mouse, and I would like to invite Professor Ewelina Knapska from Laboratory of Emotions, Neurobiology, the Nensky Institute of Experimental Biology in Warsaw. Her research interest is in the neurobiological basis of emotions, and most probably she's going to tell us a little bit more today about potential therapies in behavioral disorders.
Okay, thank you very much for the kind introduction. And first of all, let me thank the organizers for inviting me here. It's really a great pleasure to be with you and celebrate with you. Thank you. And I'd like to tell you today about very interesting structure in the brain called central amygdala. So I'm switching the gears a bit toward the brain. Firstly, the brains of mice and rats, and then the human brain. The central amygdala is very interesting because this is a motivational hub in the brain, and as such is a potentially interesting target of therapies of motivational disorders. So what are these motivational disorders? First of all, it's depression. In depression, we observe the general deficits in motivation. People with depression perceive less pleasure from interacting with different types of rewards, including food. So this is one example. Another example are neurodevelopmental disorders in which the deficits in motivation are more specific. They are limited mostly to social interactions. And this needs to find food and consume food and to find interaction partners and interact with them are evolutionary conserved and can be observed in many social species, including rats and mice. And we can assume that the neuronal circuits or the brain structures that are responsible for those drives to get food and to interact with others are also evolutionarily conserved. So we can use mouse and rat models to study them. So this is what we do in my laboratory. But where in the brain to look for these structures or neuronal circuits that control our responses or rodent responses to rewards? Well, we have the reward system in the brain, which comprises many different structures that are heavily interconnected. One of the very important parts of this system is the amygdala. That it's important for processing emotions, both negative and positive emotions. And we are focusing on one part of it, which is called the central amygdala, visible here. And as I said, it's interesting because the classical studies showed that the central amygdala encodes motivational significance of stimuli, and that stimulation of the structure amplifies motivation for food and drug rewards. We know much less about motivation to social interactions. I will tell you more in a moment about it. But I'd like to stress that the central amygdala is so interesting because it's a sort of a hub structure, which means that it receives many projections from other structures in the brain, integrates information, and sends projections to so-called effector structures that directly control behavioral and physiological responses to rewards. And we can see this intense connectivity here. This is a mouse brain in which we injected a tracer that shows connectivity of one of the central amygdalae. There is a symmetrical structure. Mice and we have two of them. One of them is injected here, and to the right you can see you can see how dense these projections are. So the idea is that because these projections are so dense, when we manipulate the activity of the structure, we actually affect many other structures which work downstream of the central amygdala. So we thought that this is the interesting target, but it's not so easy to know which structure is important because the studies of the last decade, or let's say 15 years, showed that there are different neuronal circuits within one structure. The neuronal circuit is a group of neurons that are interconnected with each other and play the similar function or the same function. So it means that there can be different groups of neurons within the central amygdala that actually control different behaviors. So to be able to manipulate the activity of the structure in a reliable way and responsible way, we need to know these structures. And such manipulations were a sci-fi in the 90s. This is the picture of Captain Jean-Luc Picard from the Enterprise, from Star Trek series, early 90s. He is stimulated, he has a cortical stimulation. It's a non-invasive method of manipulating the brain activity. It was a sci-fi, but now it's not any longer. Now we can manipulate even the activity of the structures that are located deeply in the brain, like the central amygdala, in a non-invasive manner, using, for example, transcranial ultrasound stimulation. So such studies can have some potential for the treatment. Okay, so as I said, we are interested in the food-motivated behavior and social interactions, and in this context, we found that the central amygdala is activated by both types of behavior. We can see this activation here as black dots. There are the nuclei of neurons that were activated by behavioral stimulation. It's a marker called CFOS that is expressed in activated neurons. So we know that indeed there are some cells in the structure that are activated, but we do not know whether they are the same cells or different in both cases and what they actually do. So I'd like to tell you one of the stories that we are developing or have developed in my laboratory. This one is focused on the matrix metalloproteinase 9 dependent neuronal circuits, in short MMP9. MMP9 is a protein that is involved in synaptic plasticity. And we have, thanks to collaboration with Professor Leszek Kaczmarek's laboratory, we have many tools that allow us to manipulate this protein. This is one of the reasons why we focus on these circuits. And the studies I'm going to tell you about were carried out by three very talented people in the lab, Tomek Lebitko, Ksenia Mejza, and Alicja Puść. So CFOS is, MMP9 expression is controlled by the transcription factor, which is called AP1. And one of the parts of this transcription factor is CFOS. So we thought that because we saw the activity of CFOS in the central amygdala after our treatments, we thought that MMP9 may be also involved. We know that MMP9 is involved in synaptic plasticity, which may be measured as long-term potentiation. This is the measure of physiological plasticity. It is also visible in the changes of the shapes of the dendritic spines. There's morphological plasticity that can be observed in the brain. So, and in the physiological conditions, MMP9 activity is tightly controlled by its endogenous inhibitor, which is called TIMP1. Most of the studies before were carried out in different brain structures, so we decided firstly to look what is going on in the central amygdala, and we found that MMP9 activity is indeed there, but not in all neurons. So there are some subpopulations or neurons in the central amygdala that depend on MMP9 activity. It's also involved in synaptic plasticity. We've showed that long-term potentiation, the model of synaptic plasticity, is impaired when we manipulate inhibit MMP9 activity. So then we decided to look what happens when we manipulate MMP9 activity in the central amygdala, what happens with food-related behavior. And to measure food-related behavior, we use the IntelliCage. This is the cage in which mice live in groups. They are not disturbed by an experimenter. They are not stressed. We can observe their behavior for the long time. And to get water, they have to go to one of the four corners. There are two bottles in each of the corners. And when a mouse enters the corner, it can drink water by nose poking. The doors open, and a mouse gets access to a nipple of a bottle. We can add something to the bottle. I will tell you later about it. The mice live in group, but they are individually recognized. Every mouse has a transponder with a unique number, so we know who is in the corner at any time. When they enter, they are recognized by RFID antennas. We can also apply some unpleasant stimulation. Like here, this is air puff, they don't like it. So in our studies, we use such cages because they allow for long-term observation, and they give more reliable results than the classical tests in which we observe the behavior of animals for a short time. So we use the system to check the motivation for food, and our mice had to find a bottle, one of 8 bottles in the cage, that contained the solution of 10% sucrose, which is highly rewarding. They love it. Or they had to avoid a solution of quinine, which is very bitter, and then they don't like it. And we firstly tested the knockouts that didn't express MMP9, and we found that they don't learn, even for several days in a row, they don't learn where the bottle with a reward is. But they learned very well how to avoid this bitter, unpleasant taste. But it was the whole brain. So in the next step, we use the tool that allows us to gradually release TIMP1, inhibitor of MMP9. There are nanoparticles that gradually release this inhibitor in the central amygdala. When we inject it to the brain, we can control or inhibit the activity of MMP9 for several days. And we basically replicated the results that we had in the knockout animals. So it means that this effect is related to the central amygdala. It was a bit surprising because the central amygdala is also involved in aversive learning, but here we saw that they learn very well how to avoid unpleasant taste. We confirmed it looking at the activity of the MMP9, the level of one of its substrates. Indeed, it's activated or it's clived only after the appetitive training, but not the aversive one. Then we look even closer using what the IntelliCage offers us. And we ask the question whether what is actually impaired. Seeking behavior that allows the animals to get to the reward, or consumption. And what we found is that MMP9-dependent circuits actually control seeking behavior. When we inhibit MMP9, there is the decrease of motivation to get a reward. When we overexpress MMP9, it's increased motivation, but the consumption is not affected. When they already find the right bottle, they drink as much as the healthy, untreated animals. As I said, the central amygdala is also activated by social interaction. So next we check whether these circuits are involved in social interaction. We firstly used the cage in which the mice had to choose between a social partner or an animate object. There are two parts of the cage. Healthy mice spend more time with another mouse than with an animate object. We firstly tested knockout animals, and the animals that overexpressed MMP9, no differences. They preferred, as healthy animals or wild-type animals, they preferred another mouse. Then, as I said, we prefer to observe the behavior of animals for a longer time. So we used the system in which we developed in my laboratory some time ago. We called it EcoHab. Could we activate this movie to the left? Okay, so you have to believe me that Aha, okej, sorry. Czy mogę prosić o kliknięcie na film, ten po lewej stronie na dole? Dziękuję. Sorry. So now you can see the mice moving. This system is inspired by how the mice live in the natural conditions. They love to dig tunnels along which they build nests. So we have 4 cages, like nests, connected with tunnels. And the mice had transponders with unique numbers. When they go through the corridors, there are RFID antennas, so they are recognized by the system when they travel. We also are able to analyze their behavior in more details. That's why they have the spots on the fur. They are actually virtual spots. They are not real. So what we can measure is how much time they voluntarily spend with each other in the 4 compartments of the system, how often they follow each other in the corridors, how they are interested in social stimuli that we present behind the perforated partitions here. So we tested the mice that had inhibited MMP9 activity in the central amygdala, and we found that the behavior was not changed. So it seems that the MMP9-dependent central amygdala circuits are involved in modulation of food motivation, but not social interaction. And as I said, we thought that the central amygdala is a good target for manipulation of behavior, rescuing behavior. But to test this hypothesis, we had to perform an experiment, and we decided to test it using the mouse model of Fragile X syndrome, FMR1 knockout mice that replicate the phenotype of the human patients with this mutation. This is a single mutation that in humans results in intellectual disability and autism. And what was important for us that both patients and mice had increased level of MMP9, in particular in the central amygdala. So we firstly checked whether indeed these mice have the deficits that are described in patients, and they had severe problems in learning the location of the reward in the IntelliCage. They also had the severe problem, like impairment of social behavior. Then we brought the MMP9 activity level in the central amygdala to the physiological level. And we found the full rescue of the food-related behavior, but not social behavior. So it's actually confirmed our previous observation that these circuits are involved in food-related behavior, but not in social behaviors. We also confirmed that at the physiological level, the plasticity is rescued. In the central amygdala, we were able to rescue long-term potentiation and also morphology of spines. So it seems that indeed we can manipulate this tiny structure and the neuronal circuits within it and see the full rescue of quite complex behavior. So it seems that this structure, the central amygdala, is potentially interesting target for such manipulations. I mentioned that the central amygdala is also activated by social interactions. But the apparently MMP9 dependent neuronal circuits within the structure are not involved in the controlling social interactions. So we wanted to check, because there were no data before on it, we wanted to check what these cells that are activated, that we see that express CFOS, actually do in a context of social interaction. So what we did, we firstly introduced to these cells the genetic construct that express channel rhodopsin. There's the protein that allows us to activate using blue light. We can activate the cells that express this construct. So we introduced the construct to the central amygdala of rats this time. They express it, then they were subjected to social interaction, and on the next day, in the presence of a partner, we artificially reactivate the activity of these cells. And what we found was increase in social contact. So we thought that, okay, these cells are involved in social motivation, then their activity drives animals to start social interaction. So we perform long row of experiments. They were quite complex, so I don't have time to tell you the details of them, but I would also only tell that there are some circuits that can be identified by the connectivity with the different structures in the brain. In particular, the projection from the central amygdala to ventral tegmental area, which is the main source of dopamine in the brain, is specific to social interaction, but not to food-related behavior. So it seems that there are some circuits that are specific to social interactions. So this is the conclusion. So altogether, we have some circuits that are either specific to food-related behavior or to social interactions, which gives hope to specifically manipulate this circuit to obtain the rescue of some behaviors not affecting others at the same time. And at the very end, I would like to tell you that it seems that there is some translatability of our data. Till now, the data I showed you were obtained only in the mouse or rat brain, but of course, we are interested eventually in the human brain. So we look at the activation of the amygdala in the human brain. Usually, the amygdala is treated as a whole, but we managed to divide the amygdala into its two main parts, the basolateral and central part. And we found that the human central amygdala actually contributes to food-motivated behavior and also is involved in processing social emotional cues. So indeed, it seems that this neuronal circuits that are involved in processing such cues and producing behavior may be located there. And there is some evolution, they are evolutionary conserved. So to wrap up what I showed you, is that the rodent and human central amygdala is involved in processing food and social-related stimuli, that the food and social-related circuits only partially overlap, so we can combat specific deficits in motivation or social interactions without affecting other properly working circuits. This is very important in the human brain because it's like quite complex organ, and we want to affect the circuits that work properly, because then we can destroy some behavior instead of rescuing the one in which we are interested in. For example, in terms of rescuing social deficits, we want to increase motivation for food, because we will produce obesity on the top of improving social interactions. And at the moment, we are testing the possibility of stimulating such neuronal circuits in the brain. For now, in the rodent brain, it seems that this ultrasonic stimulation is able to increase the synaptic plasticity of some circuits, so it's a potential tool to use also in a human clinic. But this is for now just the basic research. Here I would like to thank all the people from my laboratory, especially the ones involved in the projects. I told you today about Ksenia Mejza, Karolina Rojek-Sito, Alicja Puścian, and Tomek Nikolajew, and people from Brain City, in which I worked, for the very fruitful discussions and support with many tools, especially to Leszek Kaczmarek and his laboratory, and Ania Barun, with whom we are working together on the central amygdala. Thank you very much for your attention.
It's time for questions. Something.
So, can I start? Yes. So thank you for a very interesting talk. So is the structure, the morphology of this amygdala affected upon this MMP9 knockout? Is it conditional knockout?
No, the one we used here was like full knockout. It was not conditional one. The morphology is affected, but very slightly. This is there is some change in the number of synapses and the shape of the dendritic spines, but there are some morphological changes, but they are very subtle.
Okay, thank you.
Hello, very nice talk.
Thank you.
Thank you. And the question is, what is the population which is expressing the matrix metalloproteinase 9 in amygdala? Is this the dopaminergic or the GABAergic, which is?
This is a very good question. This is something that we are trying to figure out now, because in the central amygdala there are. It's very likely that there are GABAergic cells, because this is.
I thought so too.
Yeah, the central amygdala is like 80% of cells are GABAergic, so it's actually like just like thinking about probability that they are probably GABAergic. There are several so-called markers in the neuronal circuits in the central amygdala. Like somatostatin or CRF, which is corticotropin releasing factor. So we are testing now whether there is some colocalization. We know that that there is no colocalization between somatostatin expressing cells, which is type of GABAergic cells, and MMP9. We are now testing for CRF positive cells, but it's like it's not obvious what these cells are at the moment. I mean, we have molecular marker, but it's like anatomically, there is nothing obvious. I mean, they don't differ much from other cells around.
Yeah, this would be very nice to know. It'd be the depression and other things.
Yes, one quick question.
I'm interested, have you observed any differences between animal genders?
In this experiments, no. I mean, like we tested both females and males, but there are, like in social interactions, there are some differences usually, you know, because there's especially mice are very hierarchical. So it's like the level, the status, like social status usually matters in social interactions. So it's possible that there are some differences here. So we try to control for it.
Okay, thank you again.
Thank you.
Now that's the momentum for Cenorhabditis elegans. I would like to invite for the stage Wojciech Pokrzywa from the Laboratory of Protein Metabolism from International Institute of Molecular and Cell Biology in Warsaw. Wojciech, you're welcome. And you are going to tell us something about adaptation of C. elegans to external and internal stimuli.
Yes. I hope that I will convince you that this exists. First, I would like to thank the organizers for inviting me and organizing this conference, especially with such a wonderful lineup of speakers. So I'm very pleased to be here and to present our research data using a well-known model organism, nematode Cenorhabditis elegans. Okay, in the lab we study different aspects of protein turnover at the molecular cellular level and at the organismal level, where we use worms as our model. What's protein turnover? It refers to continuous renewal from synthesis and removal of proteins by degradation. We are in the lab most interested in the last episode of the life of the protein, so it's degradation. So how cells can remove unwanted or damaged proteins. So it uses two main pathways, ubiquitin proteasome system and autophagy. Less commonly, however, cells can expel unwanted proteins or damage by extracellular vesicles, like well-known exosomes. And about this pathway will be my talk, because quite recently a new class of vesicles, the class of the biggest vesicles, was identified in C. elegans neurons by Monica Driscoll's group. They call these vesicles exophias. So they observed that C. elegans neurons, here is a neuron, it's a touch neuron, can expel proteins. Proteins here is non-targeted M-cherry, as well as organelles. Here are in green mitochondria, there's identified lysosomes there. In pretty big vesicles, up to 7 micrometer in diameter, as you can see, almost the size of a neuron itself. They also noticed that during transfer of this material, the vesicles remain connected to the neuron. And these are membrane-surrounded vesicles. I will show later a few characteristics of that. But what was the most important in this finding is that neurotoxic stress or expression of toxic proteins, disaggregation from proteins responsible for neurodegenerative diseases, is increasing the production of exophias. So therefore the group claimed that this is another branch of proteostasis pathway, to improve the protein clearance. The problem is that, however, that was not that much discussed in that paper, that these vesicles are only formed in very narrow window of worm's growth. Basically, when worms reach adulthood stage, in worms there are 4 larva stages, then there are adults. We call them adult day 0 when they start to lay eggs. So they noticed that the highest peak of these vesicles is only at day 2 or 3 adults, so there is a peak of the progeny production. So it seems that this is not really a constantly active proteostasis pathway, it's only active in a very narrow window. It's not that much discussed in that paper later, but I will come back in a moment to that. I just wanted to show you a few details of that. There will be, I guess, some questions, and nevertheless I wanted to just show you of the well-known exosomes versus exophias. Much bigger, they need a bit longer time to be released. They do not need any escort machinery for the formation. They sometimes be connected with a sending cell via this thin and elastic tube. It's most important that a part of the proteins, they also contain, they can uptake from the neurons, mitochondrial lysosomes, and they don't have this phosphatidylserine marks there. Okay, that was sufficient. Only paper at that time, Nature, big story, many questions remained unanswered. Are exospheres specific to neurons? How they are regulated? And it's really the disposal of toxic proteins to produce such a big vesicles of the size of neurons is the only function of them. So before we go further, I want to show you the C. elegans muscles. So this is a worm. In orange quadrants are so-called body wall muscle cell, single nuclei cells, a cross-section where there is a cuticle, hypodermis and basal lamina peeled away to reveal two body wall muscle cell, section through those body wall muscle cell, where you can see a sarcomes, basically, so M-line, dense body called in C. elegans, which are Z-disc invertebrates, actin filaments. So basically, striated muscles of C. elegans resemble vertebrate skeletal muscle in structure, function, and protein composition, so this is a pretty good model to study also muscle biology. Well, I said that because in the lab, and we identified that C. elegans muscle can also produce exospheres, which are much more abundant and also bigger than the neuronal counterparts with diameter up to 50 micrometer, with the size up to 50 micrometer in diameter, with a role in reproduction which is far beyond the toxic, the expel of toxic proteins. Okay, how I identified it? We use various tools, fluorescently labeled reporters that were originally also used in this study in neurons. We also just adapted them for the expression in the muscles. So this is how it looks. This is a body wall muscle cell. Here it's a vesicle. In green, mitochondria. We also use proteasome subunits because we initially studied exospheres as a branch of proteostasis network, so we're just curious what about proteasomes since there's lysosomes and so on. This is how it looks like. Then the electron microscopy images, a muscle cell adjacent to that is an exosphere. You can see mitochondria inside, not looking healthy, kind of swollen, and some other material. Okay. Exospheres are released from the body wall muscle cell via pinching of mechanism. It's presented here. And they also remain, some of them connected to the muscle via this elastic tube that permits a transfer of material from the body wall muscle cell. Okay, what's happened after to this vesicle? So when it's formed, around 20 minutes after, it's released to the body cavity and it's disintegrated into smaller vesicles. And here there was a twist in our research. So because it turns out that in contrast to neuronal counterparts, prototoxic insult like a heat stress or depletion of the core transcription factor, heat shock factor 1, did not change the level of these exospheres made by muscles. So it suggested to us that this proteostasis branch might be not the only or not the core function of these extracellular vesicles. So here another jump to another organ, basically reproductive system in worms. So worms has two sexes. They are self-fertilizing hermaphrodites and males, but males usually are not that much in the population, around 0.1%. So therefore I just concentrate on the hermaphrodites here. This is a gonad, two arms. It works like this, that the nuclei migrate from distal toward proximal part, initiate meiosis, cellularize, and differentiate into oocytes. Oocytes pass through spermatica, when they are fertilized by sperm that is produced earlier by worm or deposited by male. And then immediately after, embryo starts in utero development until the eggshell can be fully formed, and then embryo is released via vulva outside of hermaphrodite. I'm saying this because similar to neuronal counterparts, we observe that the muscle exospheres are only produced, the peak of the production is at this narrow window, at the peak of the progeny production by hermaphrodites as well. And we see that later in the life when worms gets older, there is also a bit back trend to make these vesicles. Importantly, males don't do them, mostly not at all. And same is for feminized hermaphrodites. So when we feminize, meaning that the hermaphrodite can produce oocytes, but cannot produce sperm, no fertilization, no embryo, no offspring, no vesicles at all. So this, and all the data suggested to us that either oocyte fertilization, but we know already say, or embryonic development in uterus is just triggering the production of these vesicles. And why embryo? Several experiments we have done, just show you two. First, we observe a correlation between number of embryo present in the uterus, developing embryos, and the number of exophages released by a mother. And also, worms are great for the genetics and so on. So just by depletion of mRNA of certain genes that are control egg-laying behaviors. So for example, by depleting this gene GOA1, this leads to hyperactive egg-laying behavior. There are fewer early-stage embryos present in the uterus, and this, as we expected, decreased the formation of these vesicles. And when we depleted the genes that leads to egg retention of the embryos in the uterus, this increased vesicle production. Okay, we know that embryos are triggering the whole thing, how this process benefits the offspring. So by muscle-specific transcriptomic analysis, we revealed a presence of significant level of vitellogenes. So what are vitellogenes? Are quite big proteins, so lipoproteins and main yolk precursor. And here we had a bit of a problem with our hypothesis at this time, because it was kind of crazy that maybe this part of vitellogenes is transported via exophase and could be then used to nourish the developing embryos. However, the state of the art was, and we were heavily criticized in one of the biggest journals, that we should have known that vitellogenes are only formed by intestine and that's all. What we are claiming here doesn't make any sense. Okay, so in order to just prove our hypothesis that might be right, we deplete vitellogen 1. So basically there are 6 vitellogenes in the muscle, lots of mRNA of vitellogenin 2 and 6. So we deplete vitellogenin 1, which is the main egg yolk precursor expressed solely in intestine, and we would expect that there would be a compensatory mechanism coming from the muscle that would just produce more of these vesicles. That was the case. And when we deplete the only receptor of vitellogenes and yolk at the level of oocytes that allows the uptake of yolk by the oocytes, depletion of this receptor leads to accumulation of yolk in the body cavity, so meaning that there is no need of the muscle to deliver any of it. It also, it immediately, what we expected, it just blocked the formation of the vesicles. But we need some more direct proof of that. So by CRISPR-Cas9, we just tagged a vitellogenin 2 by the GFP, and for the first time we showed that this is very nicely expressed in the muscles. Here are muscles. Here it's an exophage, and they are filling exophage very well and present in the embryo. All right, but we wanted also to know if there is any correlation between the number of exophages released by the muscle and this vitellogenin in embryos of the mother with active process. This process called exophagogenesis that was named by the authors. So how to do that? From the synchronized population of worms, even in synchronized population, we already observed that there are animals that produce very few of exophages and some of them that produce plenty in the narrow window when we observe them by the microscope. So we just split these animals into group. We analyze the mother, phenotypes of the mother and its physiology, but also we look at this progeny. So as we expected, the progeny, so basically the embryos from the mother with highly active exophagogenesis, it contains much more of the vitellogenin 2. All right, still the question whether it would have any beneficial effect to have a mother with such an active process in its muscle. It turns out that yes. So as I said, worms undergo these 4 larva stages when they become an adult. So, and it is known from previous studies that yolk-rich eggs supports post-embryonic development, not only in worms, post-embryonic development and survival of the offspring of the mother. So we look on this offspring from the mother with highly active exophagogenesis to the other, and we notice that the offspring from the mother reach the adult stage far beyond the controls, so meaning that such offspring could take over certain niche before the offspring from the worms with not that much active process. Okay, so we published this story, quite nice. That's also the cover of the emboss. So here are the muscles. This is how we visualize it. Exophanes, and we are directing toward the oocyte there. So imagination, but nevertheless the model is, also in the story we showed that the embryos are able to send some signals, metabolites which we haven't identified at the time, that triggers body wall muscle to produce exophanes. Exophanes are transporting, among other also vitellogenin, and from the body cavity this is taken up by the oocyte, and it has an impact on the growth of behavior of the progeny. Okay, many questions still remain unanswered, but based on this foundation, we decided to switch our study and look at the modulation of exophagogenesis by environmental factors, more precisely social, but even more precisely by pheromones. Why we decided to do that? Because this is what I told you, exophanes can mediate the transport of maternal resources. And it is known that pheromones can control social behaviors and this maternal provisioning. So our hypothesis was that exophagogenesis is modulated and by intertissue interactions and metabolite-mediated social cues. By intertissue interactions, we mean by the neurons, by chemosensory neurons sensing potentially pheromones and sending some signals to the muscle to regulate the process. And that was indeed the case, but we started the study after the results of this experiment. So, you know, it's well known that hermaphrodite that mated with males, it produces much more progeny, offspring, because it has also much more sperm available. It has its own sperm and also sperm deposited by male. So in such a scenario, we expect that a mother that mated with male would have a more demand for the material from the muscle to support their embryos and will produce more of these vesicles. That was the case. But most surprising was that it doesn't need to mate. It's enough that it sends males. So how we have performed such an experiment, we kept males on a dish for 2 days. We removed them. So basically we gave them 2 days to leave some traces of certain, pheromones, possibly another secretum. On such a plate, we place our reporter lines, and then we score exophanes, and it's enough just that the mother would smell that there was a presence of males, and it's already boosted the whole process of vesicle production. In contrast, when such a mother was placed in a crowded environment of other hermaphrodites, also their smell was completely enough to reduce the exophagogenesis process. Okay, so what metabolites might be implicated in this communication coming from male and other hermaphrodites that ultimately lends to muscles and stop the vesicle or boost the vesicle production? These are ascarocytes. Ascarocytes in nematodes are pheromone counterparts. They are based from the sugar ascarulose, which is then connected to the fatty acid-like side chains and decorated with many primary metabolites. Many different ascarocytes, for a number of them, the function is unknown, but what is known that they control sex-specific behaviors of worms, so mating, looking for each other, also escaping, and particular embryo development. And these ascarocytes are sensed by G-protein coupled receptors, and they initiate or trigger a conserved signaling pathway like insulin and IGF-1 signaling, and so on. So, cut the story short, we identified 2 ascarocytes in collaboration with Frank Schroeder by certain genetic screenings and also by these synthetic ascarocytes. We showed that ascarocyte 10, which is also in large amount produced or secreted by males, it's alone can boost the production of the vesicles by the muscle in dose-dependent manner, and coming from the hermaphrodite ascarocytes 18, just additional that to the plate, slow down the production of the vesicles by the muscles. So there were just many, the picture is big, many figures and so on, but I wanted to come to conclusion because of time, but it looks complicated, but I will just briefly explain you what we identified. When it comes from the activation, there is ascarocyte 10, which is released by the males. This binds to one of the G-protein coupled receptor and very specific neurons we identified. And this signaling can directly and indirectly, through accumulation of embryo in the uterus, and also directly to the muscle, can stimulate the process of vesicle production. Vice versa, when there is too crowded environment, too many hermaphrodites, high concentration of ascarocyte 18, we haven't identified the receptor neuron, yes, but the interesting is that this group of neurons is activating another neurons, which are these PQR, EQR neurons that are exposed to the body cavity neurons that basically function was not that much known. And these neurons are triggering the production of two neuropeptides, and these neuropeptides are directly blocking muscle, and the muscle don't produce exophase. So it's very elegant model by the intertissue connection, and ultimately that the offspring could benefit it or not from that scenario. Summary could be, why these things happen really. So, as I told you, these males that are inducing the production of exophase, that comes at certain somatic cost, because the resources from the muscle are used, and we see that the matter, for example, its mobility decline heavily, but nevertheless the progeny is a much better quality. But in the scenario of crowd hermaphrodite population, this is a strategy to survive. There might be a certain signals that there might be too crowded, not that much food. Please stop at the moment, sacrificing your tissue. So this research basically ties the reproduction somatic traits to certain resource management. Most important question is, whether this is evolutionary concept. And it turns out that yes, 2 years ago it was shown that mice cardiomyocyte can produce exophase. And have some substantial role in cardiac homeostasis. A very nice study by this group, but still it seems that this extrusion phenomenon is very poorly understood metabolic management pathway, but worth of exploration. Okay, at the end, I really want to thank people involved, particularly the first part, so identification of exophase. Great collaboration with Agnieszka Hacińska's lab, and particularly with a postdoc at this time in this lab, Michał Turek. Michał currently is a principal investigator in Institute of Biochemistry and Biophysics of Polish Academy of Sciences. Is also a fantastic group, and we collaborate with Michał on the second project that was about this ascarocytes pheromone and control of muscle through olfactory system. And yeah, I'm thanking you for this attention.
Thank you so much. Before we go to the quick question section, a small remark from our online viewers. They kindly ask the speakers to be closer to the microphones.
All right.
Helps.
A bit too late, but...
Yes, we have online viewers. So the discussion is open. Questions? So maybe I will start. Sorry, as a plant biologist, I have not that much knowledge about the sex of Caenorhabditis. However, I would like to ask you for a comment concerning the yolk content, potentially the yolk content for the percentage of the hermaphrodites in the progeny. Or I would say the impact of the yolk for the sex equilibrium in the progeny.
I don't know any studies that, sex, males basically are not produced, okay, only shows up in certain adverse environmental conditions.
Okay.
So heat stress, oxidative stress, and so on. Then there's a specific segregation of chromosomes, and then male shows up. So this is not that, you have a specific type of eggs, rich or not rich with yolk, and then could you a result in different sex.
So there's no predetermination?
No.
Okay, that's truly an environmental impact.
Yeah.
Okay, thank you. Yeah, there's a question there.
Thank you very much for this great presentation. I have a question. For how many different cell types it was identified that the exospheres can be created? Except for neurons and muscles.
So only neurons and muscles.
Okay, but only these cell types were like researched, or it wasn't
So looking
observed?
excuse me, just looking on one of very recent posters from the group that identified them originally, Monica Driscoll, they are also showing that intestine cells can do that as well. So it seems to, although I haven't seen the data, but nevertheless it seems to that might be still broader mechanisms for broader, different kind of cells. Still, it's funny that all of them relies only on this narrow window of the reproduction. Then there is boost of the production, and it's gone. So I also like the idea that it might be, that they are participating in certain switching, certain metabolism of the organism, which is pre and post reproductive. Now participating in all of these switches, and are preparing an organism for the aging, basically, and soon death might be, but only on basically at the moment they publish two cell types. So neurons, body wall muscle, cardiac, and human neurons in vitro into the culture.
Thank you very much.
Okay, thank you so much.
Thank you.
And now we smoothly swim towards the two selected talks from the abstracts. And when I say that we smoothly swim, it's really literary, because now we are going to have a talk about zebrafish as a model organism. Our speaker is Savani Anbalagan from the Institute of Molecular Biology and Biotechnology of Adam Mickiewicz University in Poznań. And he's going to tell us something about neurovascular morphogenesis using the zebrafish as a model.
Thank you. Thanks a lot. Thanks a lot for the organizers for selecting my abstract for this talk today. And the first thing is the acknowledgement. So this work was led by a master thesis in bioinformatics student, Miłosz Chodkowski, and in collaboration with Assistant Professor Andrzej Jelezinski. And another important person in my career is my postdoctoral mentor, Gil. Without him, I will not be working on ligands and receptors at all. And a special acknowledgement also to our lab tech, Emilia Wysocka, who was very instrumental for me to establish our lab here in Adam Mickiewicz University. And also my mentors who showed the right dose at the right time of my career. So without them, I will not be standing here. We are close to Christmas. So I first start with the book of Genesis. As you can see here, the painting by Michelangelo, where he's showing the creation of Adam, his interpretation of creation of Adam. And I point your attention to here, where the God is giving the spark of life to and creating Adam. Of course, you could ask why God is a man and why first he decided to create Adam, right? Those questions are for debate. But look at these hands, right? So this, for a cell, it's something similar. It's called cellular communication. At any given time, there are some hundreds of ligands and receptors that are being expressed in human cells. And they communicate using several hundreds of pathways. They talk to each other. So these are, ligand receptor mediated cellular communication pathways. And so any organism that you will choose, there will be always ligands and receptors. Be it C. elegans, be it E. coli, be it zebrafish, be it humans and mice. Yeah? Mating in yeast, mating in C. elegans, mating in zebrafish, and egg laying. Everything is because of ligands and receptors. If you take a zebrafish mutant that lacks FSHR, follicular stimulating hormone receptor, and you cut open you see the ovary, and the ovary will be not formed properly, and these females will not have any offsprings. So again, one receptor mutant. Okay. So our group, we try to understand the role of ligands and receptors in neurohypophysis, tylna szysatka, posterior pituitary gland. It's not really gland. So the two major neuropeptides that are being released from this tissue oxytocin and vasopressin. These are nonapeptides, 9 amino acid cyclic peptides bound together by one disulfide bond. And originally, they were found by Henry Dale, where he was subjecting animal uterine dissections using the extracts of neurohypophysis, and he found that these uterine tissues will show uterine contractions and relaxation. That was, Henry Dale, 1906. And then later, they eventually synthesized this synthetic oxytocin, and then came human patients in maternity wards in hospitals, pregnant women in hospitals, inserted with some sort of electrodes to measure their uterine contractions. You see here 9 women who were favoring delivery, 3 women adverse to delivery. And that is 1950, synthetic oxytocin, clinical trials, yeah? Any case, I really don't wish to be a woman in that position. The thing is, if you look at Western literature and we think this is the entire story of the function of neurohypophysis, I think we are all wrong. Because there's just one side of the story that is published in PubMed, okay? There are these indigenous African plants, and the extract from these plants, it can also induce uterine contractions. It just was not published. It's called as Calata calata, Olandala officinalis, and for several hundreds of years, even before there was a department of embryology or maternity ward in such institutions, the Africans already know how to induce labor. And that is this cyclic peptides. So next time when I have a daughter and a son into Polish, and I read to her the Murzynek Bambo poem. She asked me who is Murzynek Bambo, and I say, you know who is Murzynek Bambo? His great-great-grandparents already know how to induce uterine contractions. Okay? That is Murzynek Bambo. In any case, if you want to read more about the story, I just made a new link, tinyurl.com mbambo. Okay? You could read more about these cyclic neuropeptides in African indigenous medicines. So in any case, the studying neurohypophysis can help understand the beauty of life, which is birth. Okay? So we study using zebrafish. It's a vertebrate model organism. And the neurohypophysis has multiple cell types, but the 3 major cell types is these glial pituocytes, these endothelia, and these axonal projections that arrive into the neurohypophysis. So what we are asking is, what can be these ligands and receptors that lead to the proper formation of neurohypophysis, using zebrafish as a model organism. And so back in 2016, this is with the help of a bioinformatician at the Weizmann Institute, we made something like this. We didn't have, at that time I think we didn't have any of these RNA-seq data and so on, so I was data mining using mouse neurohypophysis, rat neurohypophysis data, hypothalamus data, and I made a map like this. And of course, we had an hypothesis for these 3 ligands, and we published. Okay, we showed that these 3 genes, VGF or TGF or this RoboSlit, they are important for the proper formation of this neurovascular interface. That is 2016. The publication was in 2018 and 19. The thing is, our understanding of neurohypophysis development is limited by the tools that we use. Okay, if today in 2020, last year, if we need to make similar analysis on many different zebrafish data, we are, as a zebrafish researcher, we are being forced to use tools that were not built for zebrafish, that was built by mammalian researchers for mammalian proteins, ligands, and receptors, but not for us. See, at least there is a Drosophila one. So think about all the other model organisms, people who work on other model organisms, they don't have such tools. And we were also one of them. We didn't have a tool. The problem is, if we will use these tools, of course we will be able to see what are all the genes that are in this, that are common with humans and mice, but we'll be ignoring the genes that are only in zebrafish, which don't have any orthologs in humans or mice. If somebody says that they are interested to study zebrafish regeneration, then they are only looking at the genes which are in humans and mice, but not in zebrafish. Are you really trying to understand zebrafish regeneration? Maybe it is some of the genes that are in zebrafish that make them more pro-regeneration. In any case, so the point is the tools built for mammalian ligand receptor analysis are not ideal for researchers like us. So, you know what? We decided, let's build one. Let's build one for zebrafish. And that's what we did with the help of Mivosh and Anjay. So we made a new tool from scratch. It's published. It's in the GitHub called as DaniaTalk. It's open source. Anybody who has some basic coding skills can use it, apply it. The heart of this tool lies in a machine learning algorithm called as DeepBlock2 that is published by a Danish group. So this machine learning algorithm has been trained on several thousands of proteins and their cellular localization data, where a protein can localize. So just with this tool, now you can give any protein sequence, it will predict whether the protein will go to the plasma membrane, to the nucleus, to the ER, to the mitochondria, etc. If you know where a protein is localized, then maybe you could sort of guess, okay, this protein, maybe it acts as a receptor, or maybe it is acting as a ligand. That's what we did. We predicted the entire zebrafish proteome using this DeepBlock, and we sort of guessed where and all a protein can go in a cell. And once we had this list, then we sort of curated a list of potential zebrafish ligands and receptors. So many of them conserved. Some of them are not, but please, this is a sort of incomplete data because this is depending on the database that we use to find orthologs. So that database is not complete. So I expect there will be more genes that are conserved with humans. The thing is, as I told you before, that there are a lot of genes only in zebrafish that are not in humans. Here's a few examples. These are chemokine genes that are only in zebrafish, but not in humans. There are no orthologs in humans. Okay, ligands in zebrafish, but not in humans. Receptor in zebrafish, but not in humans. So now, how should somebody go and try to find which is a ligand or which is the receptor for these proteins? So our tool can sort of guess it based on the database that we use. So here are these sort of orphan or ortholog-less ligands, and we think we know which could be a potential receptor. But again, somebody should do experiments to prove it. We didn't do experiments. We are just sort of finding pre-published databases. We are trying to drag things and hence highlighting it for people who may be interested in genes or proteins such as those. We also, again, zebrafish is a very, very powerful vertebrate model organism. The power of zebrafish is also in the use of it in drug screens. And so because it's conserved, so you could model it to study many, many different human diseases and disorders. And so here is just a small list of diseases that can be potentially studied using zebrafish: autism, schizophrenia, diabetes, many, many syndromes, human syndromes. Yeah. As I said earlier, you could use drug screens. And for people who have money, they have this QIAGEN Ingenuity Pathway Analysis. You put your data, and then it will tell you which drugs you can use it. So we just decided to make an open source one for zebrafish researchers. We took the Drugs Central database. It has all the drugs that are approved that can target human proteins, and we found those orthologs, and we think there are some thousands of receptor targeting drugs that are available in humans that can be also used in zebrafish, and we give it all. If you use our tool, it will just list everything out, the same way as a QIAGEN Ingenuity pathway analysis tool. Finally, it's the interactome, so we use the string database. It has a lot of physical protein interaction, also interlog protein interactions, and we came up with this. We also, to satisfy the reviewers' needs, we also integrated all the existing human ligand receptor databases. So what we now give is this 4,000 new ligand receptor pairs, which previously did not exist. So if zebrafish researchers, before they'll be only looking at the sort of these human ligand receptor pairs, but now we say them, hey, there are another 4,000 more, which maybe is of interest to you. This is all fine. I told you that in the beginning I'm interested in neurohypothesis. So now we could take every single available single cell data. Now we already have zebrafish single cell data of hypothalamus, neurohypothesis, and so on. And here is a map what we made. So these are all the potential ligand receptor interactions that may happen in the neurohypothesis. Some of them we already published, this SLIT3, ROBO2, but now we know the story is a bit more complex because of the existence of the single cell data, because SLIT3 is not only in the glia, but SLIT3 is also in the neurons. So it just complicates things. This is all fine, so we made a tool, but so what? So what? Okay. To answer this, I will start with this. There's this the Little Prince. Again, I read to my daughter, and it's only with the heart one can see rightly what is essential is invisible to the eye, right? So here is zebrafish heart, embryonic heart, okay, and here is a single cell data of zebrafish heart, embryonic heart, okay. I see the single cell data, but I don't see. It's just for me data. It's just cells and lot of single cell data. But for me, it's nice information. You could publish in Cell and Nature of such data. But so what? I'm just rewriting here. It is only with Daniel talk that can one see rightly. From this single cell data, we go to something like that. Okay. And this is a preprint of this from Professor Dr. Cecilia Vinata group in IMCB Warsaw. So, I guess, I thank the organizers again for this opportunity. I thank you for staying for so late and to listening to my talk. I invite you, if you next time you're applying for your animal ethics application, please, you may be interested to check out my new sort of a practice and policy manuscript where I argue that we still need animal research for research in drug safety studies and understanding civilizational diseases. So thank you very much.
Thank you so much. Okay, thank you so much. We have a time for one quick question. Yes, please. There is one there.
Thank you, Stefani, for your talk. I have a question concerning paralogs. How do you handle paralogs? Because zebrafish genome went through several duplications, so I know that paralogs are a bit problematic, especially if one of them encodes protein while the other one is not doing this. And there is also this evolution of the genome and change of biotypes, where you can have non-coding ortholog in one species, like for example in zebrafish, but it becomes protein coding gene in human. So if you could explain a bit, that would be helpful.
Sure. Were you our reviewer number 3? So in any case, this work is already published. So these questions were also asked by the reviewer number 3. We use the Genome Alliance database to find out. It already has a list of all potential orthologs that also includes paralogs. For example, SLIT1 in mice and humans, in zebrafish it's SLIT1A, SLIT1B. Okay? So if you use our tool, you will already have all those inside our tool. The second thing is, because we use the string database is, it also has interlogs. So it's not just about the protein-protein interaction from zebrafish, but many, many other organisms that have similar proteins and those interaction data. So again, this is all predictions. Somebody has to do experiments and to test what we predict. Okay?
Thank you.
Thank you.
Okay, thank you so much.
Thank you.
So we are moving to our final speaker of the session, Łukasz Przybył from Laboratory of Mammalian Model Organism at this Institute of Bioorganic Chemistry here in Poznań. And we are coming back to mouse and the mouse model in Huntington disease.
Yes, thank you, Michał, for this kind introduction, and thanks to organizers for giving me the opportunity to share with you the data we acquired for the last few years. Ladies and gentlemen, welcome to the last session, or last lecture of this session. I hope not to keep you away from your food-motivated behavior and stimulation of your amygdala as we will go on. So, today I would like to share with you our two new Huntington's disease mouse models and to convince you that we can prove a mutant RNA contribution to the phenotype. So, few words about the Huntington's disease. I would like to take you back to the last lecture of the previous session of Paweł Świtoński, where we already been to the polyglutamine diseases. So Huntington's disease begins between the 30 and 50 years of age. It's a rare disease, starts with uncontrolled movement called also chorea, which is another description of the disease, with abnormal body postures. Changes in behavior, emotion, judgment, and cognition lead to the impaired coordination, slurred speech, and difficulty feeding or swallowing. So the death is certain. We have no cure up to now for the disease, although many attempts are being made with the therapies. And the death comes to the patients within 15 to 20 years following diagnosis. And it's mostly due to pneumonia, cardiovascular events, and a lot of suicide. So in a normal condition, we can see the Huntington. Okay. The Huntington has several dozens of CAG repeats that are being translated in glutamine tracts in the protein, and we have a healthy neuron with normal brain. While in Huntington's disease, those CAG repeats are being elongated to several dozens, and we have a polyQ leads to the neurodegeneration and atrophy of the brain. So there are several disrupted cellular pathways that in medium spiny neurons of the mutant Huntington. So they can lead to the proteolytic cleavage, proteasome inhibition, and leads to the aggregates of the mutant Huntington. Then we have abnormal protein-protein interactions, ER stress, mitochondrial dysfunction, and transcriptional dysregulation. That all leads to the communication between neurons and altered neurotransmitters release and the receptor activity. There are a few proposed mutant RNA pathways that can lead to the pathogenesis of Huntington's disease. They include the nuclear stress, but mainly it's non-canonical translation, lost in transcription, and nuclear stress and sequestration of RNA, mutated RNA, and then sequestration of different proteins. There are a few attempts there are models available now on the market that recapitulate the Huntington's disease. They are the models that carry human and terminal fragment. They are quick and pretty pathogenic. There are full-length transgenic mice, including YAC 128 that we've worked before, and we have knock-in models with expanded CG repeats inserted into mouse exon 1 Huntington gene. We have plenty of more. There are not only mice, there are worms, there are Drosophila models, there are zebrafish models, as we've heard before from Savani, and rat models, piglets, sheep, and as well monkeys. So the main goal of this project was to evaluate the in vivo effects of induced by mutant RNA. We wanted to directly compare behavioral caused by mutant RNA only with behavior caused by both of this factors, so mutant RNA and mutant protein, and to identify networks that are deregulated on RNA level. So to show you, this is our thought behind the model. So we have a ROSA promoter or CAG promoter to choose one from. We have a neo and stop cassette, transcriptional stop cassette, ATG codon, and around 100 CAG repeats, HA tag to visualize the protein MS2 for identification of RNA. And this is a transcribed and translated model with the proper ATG, while we have only the transcribed model which lacks ATG, so the protein is not being translated. The beauty of the system is that we have this what I showed you is a parental line. Upon the Cre recombinase breeding with a mouse, it cuts off the stop cassettes, and the transcript are ongoing. So you can imagine many purposes of using of our model. So we have followed the mice throughout almost 2 years, and with a checkpoint checks every 4 months. We've started with 31 animals per group that we had to take out for molecular analysis. We have performed the whole battery of behavioral tests, including the rotor rod, static rod, cylinder test, grip strength, open field, Actimod, and phenotype scoring on the end of the project, of the experiment. We have analyzed the blood, the qPCR, obviously with organ weight, immunoprecipitation, and we have performed and encountered the mouse neuropathology panel. We have checked as well the microbiome and immune status of these mice, but we have no time to show you all of this. So firstly, the proof of concept. So we see that our model, lacking the translation of the protein, has a higher number of CAG repeats, although they do not change in time. We have proven the presence of the protein in our translated model, this HD100Q, while the lack in wild type or untranslated model. We have visualized the transcripts on mouse embryonic fibroblasts coming out of this model. So firstly, the behavioral test that we have performed, we have seen that the rotor rod, when we put the mice on the rotor rod, we've seen that they start to lose their motoric abilities already as early as month 4 of age. On static rod, that comes a little bit later, but we can see in almost every time point that there is a difference between the mouse. We have conducted the Hindland test and gait analysis, and in the middle and the end of the experiment, and taken it together. We have performed a phenotype scoring at the end of the experiment, taken 4 different tests like ledge test, hind limb clasping, gait, and kyphosis, and taken together in a measurement of the phenotype for those mice. With the use of Actimod system, that is 100 laser beams that track the mice every minute, we were able to perform a 24-hour measurement of the behavior of those mice. And what we have seen is that actually during the day, so this is the daytime, they do not really differ between, but when the lights go off, our mice start to be hyperactive. This was only possible with the use of this state-of-the-art system, and this activity shift was present as well in both time points. We have used the same system, but not within the home cage manner, but in an open field, and it came out that our mice are more anxious during the 15 minutes presence in the cage. So we have performed, as I mentioned before, the neuropathology panel using the nanostring, and what came out, we have tested the stratum of the 21 months old animals, and we have come up with 30 and 34 differentially expressed genes specific to the models when referred to the wild type, while 4 genes were overlapping between the models. The nanostring covered almost 800 genes, and using the ingenuity pathway analysis, we have picked up the 20 most significantly changed pathways related to neurodegeneration, which included the chaperone-mediated autophagy, the retinoic acid receptor activation, IL-8 signaling, neuroinflammation occurring in both models, but we had a clear signaling as well, and we had 2 different pathways that have been changed specifically to the model. So in translator model with the polyQ tail, we've seen the axonal guidance signaling and Huntington's disease, while in non-translated model, we've seen the Krebs signaling in neurons, leading probably to apoptosis. The EPA gave us the few genes that had interacted within pathways, that they had inflammatory markers as well as vascularization markers here, and adhesion molecules. So to conclude, we have generated 2 unique mouse models of Huntington's disease that distinguish between pathogenic roles of HTT transcript and protein. So in our model that is not being translated, we have seen the separation of the RNA, so we have seen only the RNA influence on the model, while in our HDQ we have this coexistence of the RNA and protein toxicity. We have fully characterized them using a vast spectrum of behavioral and molecular tests. We have a model that is slowly progressive, that is reflecting human pathological condition, and it's a first direct proof for significant phenotypic effects related to CAG RNA in context of Huntington's disease. And I hope that these models can be used for cell-specific expression studies using the Cree-specific mice. I would like to acknowledge the main leaders of the project, so 2 Magdas, Woźna Wysocka and Jarosław Ciesiołka, Paweł Świtoński and Agnieszka Fischer from the Department of Medical Biotechnology, with a few other people, including Joanna Suszyńska-Zajczyk, that performed and some of the behavioral testing. And the whole experiment was done in animal facility of the Center for Advanced Technologies on Morasko campus. And worth mentioning, and I have to mention that, is that this idea of the RNA toxicity and the first idea of the model was from deceased Professor Krzyżosiak. taken a little bit of opportunity, I would like to advertise ourselves. So we are a laboratory of mammalian model organism. I encourage you to visit our website. We can see what we are able or incapable to do. And I know that animal experiments can be hard, but with our help, they don't have to be. Thank you for your attention.
Thank you so much. So now that's the moment you can ask actually our speaker about how he can help you. Something bothers you, it's interesting for you. Yes.
Thanks, Łukasz. So how did you score the phenotype? Is there any rank or something? Because you showed the test and then you showed the graph where you scored the phenotype.
Yeah, so that has been done blindly by two independent experimentators, researchers, and you score in a scale from 0 to 3 the 4 different parameters. So you see the hind lip clasping, then there are certain moves of the leg that is being done, or one leg, both legs, or taken together, grasping. And you can take out of it a certain number, so you scale it, and then you sum it up, you divide it, you have a score.
Okay. So it's kind of, how to say, it's
subjective.
Subjective.
Yes, but doing blindly by the two independent experimentators, it can be
Yeah. Okay.
seen.
Thanks.
One quick question. Oh, there's one here.
Thanks, Łukasz, for nice work. I would like to ask, what do you think, or do you have any speculations on how your finding can open the door or new ways to find therapies for Huntington disease, maybe antisense RNA, or how would you see that?
Yes, so our model reflects kind of slow progression of the phenotype. So if you're struggling with showing any specific effects of your therapy, for example, in R61 or R62 models that are really quick and develop the disease within weeks or, few dozens of days, then our model would be more fitting. The other use of so this would be for the translated model, but for the untranslated model, you can have a proof of concept of what you're trying to achieve by targeting the RNA, possibly. So it will never be, it doesn't have a protein, so it doesn't reflect the human condition, right?
Okay, it's a pity, actually. This is the end of the session. But there's also good news. There's quite a lot of social activities this evening. So let's applaud ourselves for being here. And thanks our speaker for the session. And I wish you a good evening.